University, which is, I think, a very right platform to explore their knowledge and gain something new for the future endeavors. But I would like to share my thoughts about the science, technology, and basically my experience, how this technology helped me to innovate the new things in the molecular diagnostic. So, what is science? Basically, science is the experimental pathway where we can study the structures and behavior and physical of the world through various observations and various experiments. So, basically, it is a knowledge based analysis. And what is the technology? The technology is the application of scientific knowledge. So, science and technology should go hand in hand to get a good success and to get a better future. Next slide, please. Yeah, thank you. Uh, so, as you know, in this last decade, the technology has developed very well. We cannot think the life without technology. It's help us to live very easily and to grow up very easily. The lifespan has been increased a lot in the last decade. The medical advances help us to live without pain, with all the treatments on the medical devices in place. The information technology has grown up in such a way that sitting here right now, I'm giving the talk with this and sharing my thoughts with the student. It's of course the technology and nothing else. So smart use of technology is an integral part of success in business today, as I feel. See, what is the number one industry? Of course, it is the information technology. The cloud sharing, artificial intelligence, machine learning has the great impact on our industry, and it has a market value of greater than 135%. This IT industry ruling the world. And what is the second industry? I feel it is the healthcare industry, because that the technology makes life very happy, very easy with a moderate modern things. The technology in the healthcare, with this science and technology in medicine, the life is very very easy, where patient can easily undergo the diagnostic tools, then they can get a easy treatment, they can get the online appointments, no, not need not to go to the doctors. With the visual tools, they can contact with the healthcare providers. And of course, the invasive technologies now went into the non-invasive procedures, where with a less pain and easy healing, we can undergo the diagnostic testings, diagnose what disease we are suffering, and go for a proper treatment. The technology is the catalyst for innovation, is it my feeling? See, through various experiments of technology, using the technological tools, which is a strive for innovation, we can gain the new things in place. And with the artificial intelligence now, which is ruling the world, is what the future of the technology also. Next slide, please, ma'am. So before going to the role of science and technology in medicine, I would like to share my experience in technology. As the madam previously told, I am a virologist. So I have completed my master's in virology and learn how the virus, which is an invisible enemy, but ruling the world. So this is a very minute particle. We cannot see even with a naked eye, but it's totally capturing the world, and we need to fight against it together. So as you know that we have suffered with a like life threatening pandemic, SARS coronavirus 2. So sharing the thoughts about it. So previously also, if you say how technology developed about it, when the SARS virus first uh, de- came in the world in 2002. The whole genome sequencing was done completely during 1 year of time span, and then actual treatment and depth analysis started. But with these technological inputs, technological tools, and advancement in technology, with this SARS coronavirus 2 pandemic, the scientist was able to sequence the whole genome of the virus within month and came up with the beautiful diagnostic tools. So basically, I worked on the molecular diagnostic parameters where our vision is to provide the public to provide the various tools in a cost effective manner to the uh, Indian society, where the infectious diseases can come for diagnosis, then oncomarkers where we are targeting the molecular detection of it, then genetic disorders. And I feel that this is all advancement in diagnostic is just due to the technology in medicine. So let's go through what are the role of science and technology in medicine. I would like to share my thoughts about it. As you know, the cancer is the second leading cause of death in the world. It is a deadliest, we can say, the not a disease, but it is a deadliest thing we never feel in our dreams also. So previously, when someone diagnosed with the cancer, it was like, now it's the end of the life. But with the technological tools and advancement, precision medicine has come up. What happened previously, that the diagnosis was very late, so on time information was lacking. Then the treatment was very costly and not to the accurate mark. So as you know, the chemotherapy is the way to like fight with the cancers, but chemotherapy has a lot of toxicity and side effects. But with the precision medicine in the market, it has it has really overcome with these side effects. So what is a precision medicine? It is which allows the physician to select medicines and therapies to treat diseases like cancer, which is based on individual genetic makeup. So, as I told earlier, also, the technology has helped to develop molecular diagnostic, which is the actual biotechnological era. And these tools help to provide the personalized medicine based on the genetic makeup. What is a genetic makeup? As you know, each individual has a specific genetic makeup, but the 99% are common nomenclature, and only some in 0.05 or something percent are specific for human. Now whatever the common things, common chromosomes, common genes are there, when there is a mutation in that genetic makeup, it may trigger the immortal cell growth, which lead to the cancer. If we take the example of leukemia, especially the chronic myeloid leukemia. So, when the first patient was diagnosed with the leukemia long back in the Philadelphia, there was no treatment, no diagnosis, no prognosis, nothing else. But then scientists with a high curiosity started working on it, check for the chromosomal aberrations, and see and observe that there is a translocation in two chromosomes, and thus affecting the genetic makeup. When they go insides of this translocation, they understood that there is a translocation which are in turn changes the expression pathways and leading to immortal cell growth of the B cells. So how to overcome it? So we need to stop the pathway which are leading to this immortal expression of the specific molecule, and in turn which are giving the cells a non-stoppable growth. So over the period, scientists have come up with these observations with the technology, and cytogenetic techniques came up. Due to the cytogenetic techniques, now we can easily diagnose this translocation at the chromosomal level and by karyotyping, and further with molecular diagnostic tools we can easily diagnose if the specific translocation leading to the BCR ABL different pattern. So, and as per the evidence, almost 20 to 30% of the leukemia patients have this mutation. And with this proper diagnostic, we can give the precision medicine where the tyrosine kinase inhibitor drugs are given, which are very, very specific to that translocation and thus help to extend the life in a good way. So, initially, if the CML was diagnosed, then the lifespan was just 2 to 3 months. We can, it's deadlier, I know, but with this advancement, with the so much of research using the various tools in the medicine and so much of knowledge by observation experience, now we came up with the precision medicine where the CML patient can expect a long lifespan in a years, and in rare case the cure can be possible. So the precision medicine, I feel, is the outcome of the technological advancement. Going forward, see, the telehealth is also helping us in a various ways. Telehealth describes a quickly developing technology that allows patients to receive medical care through their digital devices instead of waiting for face-to-face appointments. And especially this telehealth helped us during this lockdown period in the 2020. See, during the lockdown, no one can go outside. We need to sit inside, keeping the social distance, even, and all the focus was on the only and only COVID-19. But it does not mean that the uh, people around us cannot suffer from the other diseases with the some background health issues, and with the climate changes, routine routine diseases come up. So for that, to get a treatment, the face to face appointments were not possible last whole year, but this telehealth helped us to sit at home and contact the doctors using the digital devices and get diagnosed with a proper expertise, and for which help us to the further management of the clinical things. So telehealth is a outcome of technology in a recent years. The next one is artificial organs. See, initially, so much, I mean, when the medical health is concerned, then the failure rate of liver, kidneys were high, and it can cause the death. But with the artificial organ technology, where we can even like in silico prepare the blood vessels, synthetic ovaries, and even pancreas in an artificial way, and we can replace it in a human body without any without any the no immunological system opposition, and it can help life saving to many patients every year. Further, about a robotic surgery is used to minimally invasive, which are minimally invasive and help the doctors to do the surgeries with precision and control the things very easily. So I think this is the future of the technology. The small but not least, the smart inhaler. The inhaler are the main treatment for the asthma, and with this proper utility, almost 90% of the asthma patient can be cured. Though it is not in use in a proper way, the smart inhaler is playing a crucial role. See, the technology helped us in so many ways to control the health pattern, to give a proper medicines, but the thing is we should be aware of these things also, and the acceptance is also there. So I feel that in Indian society the unawareness is high about the medicines, about the healthcare, but as the aware we, and I think we are here to make them aware. So in addition to our profession, we should be aware of the technology and using the technology in a proper way, so that we can is there. Technology will surely help us to get connected very easily. Henceforth, some network issue is there. I'm really sorry for that. Can I get a next slide, please? Yeah, thank you. So now I want to share my thoughts about how these modern technologies help us to tackle the COVID-19 pandemic. I would like to share that I worked a lot in COVID-19, and how this journey went for me, I would like to share my journey with you. See, in 2009, in December, the first patient was detected in Wuhan, in China, with a SARS coronavirus 2 the thing. Later on, the patients were diagnosed in US and Europe, and that time only we thought that this disease is a transmissible disease and may hit India also, and we should be prepared with some diagnostic tools, which is a first step of management. So we started working on it. I was pregnant at that time, but of course, with this technological improvement, though initially I was going to the laboratory, in the last few days I could not go to the laboratory, but I started work from home. It was a challenge for me, but I have taken this challenge, and with this my knowledge, my expertise, and using this digital era, I help my team to work through day and night. I guide my team to do the scientific experiments, do the research, and come up with the 100% accurate COVID-19 test kit. So, uh, how this technology help to combat the COVID-19 pandemic. With this technology, the scientists were able to come up with the molecular diagnostic tools as a first aid of the management, which is followed by the supplementary treatment, because you know that there were no specific drug, antiviral drug to treat the COVID-19, and of course there were no vaccine. But with the additional supplements, the doctors were helped people to control the disease, along with these molecular diagnostic tools which were really unavailable in remote areas. The technology helped with a digital era and different things to screen the patients in an initial way, in a initial stage, and help them to the management. At a technological level, for diagnostic kits, see, in 2002, as I previously told you, scientists took more than a year to sequence the viral genome, but in 2009 it took less than a month, which helped in turn understanding the structure of virus and further creating a vaccines. Now you know that the vaccine is already in the market and helping to control the disease very well. Mm, next slide. Yeah, thank you. So, science and technology sector constituting of data science, machine learning and artificial intelligence, and in turn contributing towards the COVID-19. So what is artificial intelligence, which is now a focus of technology in current days? So facial recognition cameras to track the infected patients with a travel history, then robots to deliver food, drones to disinfect the area and the hospital places, the patrol and broadcast audio messages to public encouraging them to stay at home. All these different measures using the artificial intelligence technology helped to control the disease. In turn, the artificial technology also helped to develop drugs and coronavirus vaccine using the supercomputers. So, these modern technologies help us during COVID-19 pandemic very well. Next slide, please, ma'am. Yeah. See, as I told you, the diagnostic start, but yes, the uh, I mean, the pandemic was so high and the prevalence rate in incidence rate was so high that each and every patient cannot get this the diagnostic tool on time. In remote areas, there were no infrastructure also to go through the molecular diagnostic techniques. So, at that level, the radiological images help doctors to diagnose the patients in a very initial stage. So, CT scan, X ray, and disease tracking helped in combating the COVID-19 pandemic. Further, the prediction of health condition, it is also important. So, the CRP protein, which is a C-receptive protein, which shows the deviations and help to understand the pathophysical, physiological progress of COVID-19 help us as a secondary analysis tool for doctors. Further, the technology helped scientists to predict the COVID 19 protein structure, also, which in turn helped to devise a vaccine and drug discovery. So, overall, if we see together, the technology has helped us a lot during this COVID 19 pandemic for diagnosis, for curing, for clinical management, epidemiological management, and so on. So, we should be thankful for the technology to help us in a way. Next slide, please. Yeah. So this is out of the way of pandemic, but I would like to share a few things with you also. See, the medicine in so the infectious diseases is the one part where the technology helped us a lot. But along with that, the genetic disorder is also very important to understand, diagnosed in very early stage, so that some medication is possible. So this is one of the example, very recent example in Maharashtra, where this is a Tira. She just 6 months old, and during birth the weight was perfect and all other parameters was really perfect. But within month or two, their parents observed that the reflex action was very high when they feed with the milk. Secondly, the muscular movements was very restricted. It was very difficult for her to move. So with the help of technology, they did the investigation, the doctors did the investigation, and the spinal muscular dystrophy was diagnosed, the type 1 dystrophy. It is very rare disease, but it is life threatening disease, very and very dangerous disease, where the it affects the central nervous system, peripheral nervous system, and voluntary muscle movement. As the disease progresses, children find it difficult to carry out basic activities like sitting, swallowing, even breathing. So it is the biggest genetic cause of infant death worldwide, affecting 1 in 10,000 babies. So right now we cannot say it as a rare disease. The prevalence is also getting high these days. So what should we do? What should we do to treat this, to diagnose this? So technology has helped us to diagnostics in early stage using a genetic molecular tools. So the single mutation in gene is reflecting to the spinal muscular dystrophy, SMN gene, where basically it is a allelic mutation, allelic discrimination, where only single allele got mutated, and it is due to the hereditary bit. So if parents has both, so is a carrier, then the 25% chance is there that the baby get the SMA disorder. So this 5 months baby diagnosed with SMA, and see, the only single drug is available which is called Zolesma, and it is very costly, 16 crore rupees. But using the digital era, their parents ask help through various fundraisers to collect the money. And I'm really happy to announce that they collected 16 crores. It's not very less amount, it's a huge amount. And the drug is one time therapy, it is a gene therapy basically. So technology helped these parents in two ways. During the using the digital ways, they ask help and get the help also. And secondly, due to the advancement in medicine, they got the drug which is very specific for this disease and can cure the disease. So, gene therapy is one of the tool where science and technology helped us to overcome with these deadliest disorders, where even the gene can be replaced in human body and our signaling pathway can be adjusted in a way that they even with these dangerous disorders the person can survive with a healthy outcome. So next slide please. So, coming right now, I have shared my thoughts about how technology are helping us these days. So, coming to the future perspective, how technology will be useful for us. Even we cannot think a single day or a single minute without this technology. So, technology and humans is in hand to hand for betterment of the healthcare. In medicine and healthcare, digital technology help us to overcome the diseases, to get a healthcare system a better one, help medical professional and patient for effective solutions for diseases. So we can expect that using this technology we will overcome the battle against various cancers, then AIDS, and of course with Ebola. As you know, Ebola outbreak was there in Africa few years back, and it is the life threatening disease. Till to date, we don't have any medication or vaccine on Ebola, and it is the deadliest virus. So I am sure that this technology will help to get some outcome with this uh, pand- Ebola pandemics or Ebola outbreaks, and of course the cancer, which is a second leading cause of death all over the world. So technology already it has helped us a lot, but in future also technology will help us to overcome the cancer. So next slide please. Few of the things which I feel that technology, with the use of technology, these things can come up in a future. So one of these is a tricorder. This is a medical tricorder, is a handheld portable scanning device to be used by consumers to self-diagnose medical conditions within second and take basic vital measurement. So right now we need to go to the doctor, undergone so much of investigations to detect what is affecting our life and health. But in future with this a very small device and using this digital era, I think the person can able to scan and self diagnose the medical conditions with some basic measurements to overcome it. Secondly, the revolution of drug development. Right now, to develop any drug, which is maybe antiviral drug, maybe the to like treat the cancer through the chemotherapy. So drug development is a long process. Clinical trials are included. Step 1, stage 2, stage 3. So many trials were included to get a finalize the composition and the utility of the drug and efficacy and safety also. But with the technological advancement, uh, we can get a revolutionary drug development where the solution therapeutic solutions will be get in a record time and speed up the time to market, all while saving cost and lives. So using the technology, I think the cost will get down for the cost. Whatever the treatments are costly right now, we will get in a cost economical way. The secondly, the disease diagnosis will be very easy, management will be easy, and in turn it will help us to live our life very smoothly. So next slide please. So I would like to thank you to giving me the opportunity to share my thoughts about the technology and medicine. I would also like to congratulate all the students in Shastra University uh, for being there and I'm wish them all the best. I would like to share my thoughts for them, that be bold, be curious, and be informed. You are the future of the technology. As in, one time Einstein said that I'm not intelligent, but I'm curious enough to get through. So I would like to tell you that be curious. Curious is the way for innovation, and technology is catalyst for innovation. So be curious to understand the science, go into the technology, its application, and be confident, because the confidence is the way to success. All the best. Thank you so much.
Thank you so much, Mrs. Meenal. That was really indeed a fitting inaugural address for science, technology, medicine, and the nexus. Thank you so much. And you're a living example of being bold, curious, and informed, and how what it can give you if you're being bold. Ma'am, we have one quick question.
Yeah, sure.
Ma'am, this is from Ms. Hema Bhagavati. Ma'am, are there any products from your organization to measure the level of antibodies in people who have been exposed to SARS-CoV virus, and also to track the body's immune response after COVID-2 vaccination?
Yes, yes. I would like to share the things. We are in a molecular diagnostic, but as the COVID pandemic came up, we were also in the serological diagnostic also. So, MyLab Discovery Solutions have the solutions which are antigen detection kits and antibody detection kits. So, if person has suffered from the SARS coronavirus 2, then IgG antibody detection can tell us whether it has a previous infection or not, and IgM antibodies can tell us the if it is a current infection, that is a acute infection. So we have a rapid test, rapid lateral flow assay, where the we can get the result in just 15 to 20 minutes. So it's called the rapid. So we can detect the IgG antibodies and IgM antibodies to check for the condition of the person, whether it has a pre-exposure of this virus. Secondly, after vaccination, we need to understand where this vaccination is really effective for that person. So how we can do this? By focusing on the neutralizing antibodies, basically. So for COVID-19, when it affects the person, then the immunity will make up the neutralizing antibodies, which are fight with the virus and overcome the disease. So if we can detect neutralizing antibodies in that specific person, then we are sure that the vaccination is efficacious. So we have a rapid detection test where neutralizing antibodies can be detected in just 20 minutes. So yes, we have the solution. I would like to I'm feeling happy to tell you that we have the solution to check the efficacy of vaccination.
Thank you so much, ma'am. We have few more questions, but since because of the time, thank you so much for your time
Thank
and
you
your
so
thoughts.
much for giving me this opportunity. Thank you so much, and all the best to all students. Wish you good luck.
Thank you, ma'am.
Thank you.
So next, we will quickly move on to the session. I would like to invite Dr. R.B. Chandramohan from Shastra University to session the chair and kindly introduce our speaker, Dr. Praveen Vemula from INSTEM Bangalore. Over to you, sir.
Good morning, everyone. I'm very pleased to welcome Dr. Praveen Vemula. He is an associate professor at the Institute of Stem Cell Science and Regenerative Medicine, INSTEM Bangalore. He has completed Masters in Chemistry from Osmania University and obtained PhD from Indian Institute of Science, Bangalore. He has done postdoc at City University of New York and second postdoc at Harvard Medical School, MIT. And since 2013, he has been a faculty in INSTEM, Bangalore. He is an expert in developing chemical technologies for medical applications. His work spans the fields of biomaterials, drug delivery, medical devices, and chemical biology. He has published more than 70 peer-reviewed papers and has given 150 national and international invited lectures, including 2 TEDx talks. I think we should all have a look at those TEDx talks too. He has more than 25 patents issued or pending, and he has been crucial for starting up several companies. Several technologies developed in his lab have formed the foundation for multiple products onto the market and currently under development. Some of the companies are Sepio Health. It's an anti-pesticide technology company in India. Artus Therapeutics, a drug discovery company in US. Skintific, a skincare company in France. Alveo Therapeutics, an inflammation targeting company in US. And Color Threads, innovative textile company in India. There are many things. I think if you look at his lab profile, and we also know him personally at Shastra, he has visited us several times. We had a talk, we had a conference, international conference in 2014, Emerging Trends in Nanotechnology. Since then, we have been very acquainted with him, especially some of our students who went for semester abroad program know him personally, and that is how I also got to know him at that time. I'm very glad and happy to invite Dr. Praveen Vemula for the talk. Thank you very much.
Thank you so much. Thank you, Dr. Chandra, for kind words. I think it's a very long association with Shastra. I think I have more than a decade association with Shastra now. Welcome to the conference. And at the outset, I'd like to thank organizers, Dr. Adeline and Dr. Prasanna, for this, at least giving me the opportunity to share some of our work with attendees. And I think we had a great talk by Dr. Minal. So now what we are going to do is let's discuss about some of the conceptual materials what we are developing, that is like a disease responsive biomaterials. So it's been recognized one of the emerging concepts in the treatment of autoimmune diseases and inflammatory disorders. So as I said, I'm a faculty at INSTEM, which is located in Bangalore. My lab is primarily focused on clinical translational research, where we run two major themes in the laboratory. One is biomaterials and another chemical biology program. And typically, biomaterials are used to develop new concepts or new materials to, eliminate all the limitations that may have with the current drugs and improve their efficacy. But whereas in the chemical biology program, it's a completely new drug discovery program. And as name suggests, biomaterials could be both types, they could be used in a therapeutic applications, or you can also use them as a prophylactic materials to prevent the disease to occur. In the drug discovery program, again, we can have both types of drug discovery, which is small molecular based, traditional medicinal chemistry approach based, and new chemical entities can be developed, or either novel biologics based drugs can be developed. So today, primarily, I'll talk about our effort in developing therapeutic biomaterials. And because as if we see, inflammation is inherent part of most of the diseases you take, arthritis, psoriasis, lupus, and so on. But if you see, despite of having a very potent anti-inflammatory drugs are available in the market today, the overall therapeutic outcome from this disease treatment of these diseases is very poor to moderate. The primary reason for this is if you understand the nature of the disease or nature of inflammation in these diseases is highly fluctuated, because it's never like a steady state, right? It's you have an increased period of the disease, you have a steady state, and you have a remission period, occasional flare-ups, and so on. But unfortunately, today we do not have any diagnostics tool or non-invasive diagnostics tool to measure the patient how much inflammation a patient has, right? So because of that, it becomes very difficult for the clinicians to prescribe the right amount of drugs and the duration period of the treatment. So that's where we thought if we can develop the new concept, which could be give the appropriate drug delivery for all the inflammatory disorders, then we will have much more impact in this field. So, so when we decided to develop new concept in the drug discovery, if you go back and see, like, almost 70 years of the drug delivery, the two major breakthroughs concepts have been developed which change the way we deliver the drugs, right? So one is, developing this excipient based formulations. This could be polymers or small molecule based. But choosing appropriate excipient drug combination, you give the single dose and keep the drug in a therapeutic window for a certain period of time. When it goes to subtherapeutic level, you give another dose of drug and so on. So that's how, like, a doctor suggests you whether you have to take a medication once in a day or twice in a day or so on. And this is great for the treatment of where you have to take medication for a few days. But when people have to take the medication for chronic conditions where you have to take medication for, like, several weeks to several months, it becomes hard to take the medication without missing any doses, right? Because if you take more frequently, it becomes toxic. But if you take less frequently, it becomes least effective. So that's why late 70s, people have come up with a concept called sustained drug delivery systems, where people have developed this variety of biomaterials like, hydrogels, polymers, and biomaterials like, nanoparticle, microparticle, and so on, where you can encapsulate drugs and give a single dose administration, and they sustain manner they release and keep the drug in a therapeutic window for much longer period of time. So these both ways of delivering drugs revolutionize the way you treat the patients, right? But unfortunately, if you see inflammatory disorders, as I mentioned, the level of disease itself highly fluctuated and it changed from patient to patient, right? So then either way of delivering these drugs are not effective because you end up with either overdosing the patient or underdosing the patients. So ideal way of delivering for treatment of these disorders is, like, it should be identical to the level of disease, right? For example, when the disease comes, they should release the drug. When disease goes down, remission period, it should stop releasing. If flare-ups comes, they should release. Since we don't even know how to measure non-invasive manner, it's impossible to design a material which can sense and do this, right? So only way to do this is, like, instead of we pre-tune the kinetics, if disease can autonomous way it can control and decide when to release the drug and how much to release the drug, then we can have the great output from treatment of these disorders. So that's why we thought of developing where the disease response drug delivery could the way to treat these inflammatory disorders. So how we develop this is, like, almost 5-6 years we spend on time to develop these systems. And in this concept, what we do is first you identify a biomarker which is specific to these diseases. So this biomarker could be, like, an enzyme, a cytokine, a peptide, or protein, and so on, which will be produced only during the disease. So in case of inflammation, it is proteolytic enzymes. Now you design a injectable or implantable drug delivery system. In the absence of this biomarker, it shouldn't release the drug non-specific manner. But when get in contact or with the biomarker should able to degrade and release the drug. And release drug goes and alleviate the disease that stop producing the biomarker that stop releasing the drug. In this way, your disease itself is controlling of when to release the drug and how much to release the drug. So that's the overall concept what we have developed. And I'll show you quickly run through how this could be used for multiple medical applications. Okay, like protecting transplanted organs and the treatment of arthritis and IBDs and so on. So when you talk about organ transplantation, you have, like, solid or internal organ transplantation, which are life-saving organs like, heart, lung, liver, kidney, and so on. But the second class of organ transplantation called vascular composite tissue allotransplantation, known as VCA. Here, like, what we do is, like, entire tissue, composite tissue you will transplant, like, full hand, full face, full leg can be transplanted. Here, all color-coded parts are already been successfully transplanted. Practically every part of your body can be transplanted by using these microsurgeries. Today, specifically, we'll talk about the hand transplantation program, where now surgeries have developed so much that people can do, like, a single and double hand transplantations very efficiently. And the post-transplantations, these patients can regain their function and they can do normal chores, and they can get the sense of touch and feel and so on, right? So it sounds amazing, right? Like the kind of impact it has. But the question is like, is everything else okay with these patients? But unfortunately, answer is no, because as you know, our immune system is our friend, right? Like it is designed to identify foreign material, foreign tissue, and reject it and neutralize it, right? And protect us. But in these patients, the same old friend becomes foe because your immune system identifies your transplanted tissue as a foreign material, try to reject it, right? So that's why the post-transplantation, all these patients have to take immunosuppressant drugs to control your immune system throughout the life. And because of that, they have to take like, large doses every single day that lead to the condition called global immunosuppression. They become immunocompromised. And even a small infection could be very lethal for this patient. They can potentially lose their life. So that's where we thought, like, if we can develop a system where you can protect the transplanted graft, but without causing any global immunosuppression with all other non-specific toxicity, then we can have the major impact on this. So then that's where like the people started using these drugs and using biomaterials to encapsulate and injected them. But unfortunately, overall therapeutic outcome wasn't great because, again, your immune activation associated inflammation is highly fluctuated. And this material was continuously releasing the drug, so it wasn't very effective. So that's where we thought having this inflammation responsive material may have the great output. So, and it is well known that like, inflammatory conditions lead to production of this hydrolytic enzymes, especially MMPs and so on, in a high concentrations. Then we thought, can we develop a system which can be responsive to release the drug only in response to these enzymes? Okay. And how our conceptual approach is like, we design a variety of small molecules called lipids, amphiphilic molecules, and these molecules have the ability to self-assemble together to make these nanostructures. And during the self-assembly process, one can encapsulate the choice of your drug, here, say immunosuppressants, and they don't release the drug in a non-specific manner, but they can get exposed to proteolytic enzymes that are associated with inflammation, can degrade and release the drug in an on-demand manner. So that's the overall concept what we have developed. And to make these amplifiers, what we have done is we screened like a few hundreds of compounds called GRAS agents, generally recognized safe agents by FDA. And we came up with the 4 molecules which have the ability to form this self-assembled structure. One of them triglycerol monostearate, and we optimize conditions to generate hydrogels based on these molecules. And we selected the immunosuppressant drugs called tacrolimus, which is given for hand transplantation patients. And we optimized conditions to encapsulate these drugs and generated these injectable hydrogels. Once we have that, we went and tested their ability to release the drug in response to the proteolytic enzymes. As you can see, in the absence of any proteolytic enzymes, it releases like 5 to 10% of drug non-specific manner, but after that, it doesn't release at all. In fact, we tested up to 3 months, you don't see that non-specific release. But in the presence of proteolytic enzymes, you can see the degradation of that and release the drug in a responsive manner, right? Then but like these are very stabilized enzymes. Then we wanted to see in a real inflammatory like condition can do the similar manner they can release the drug. So to test that, you take the macrophages, which are and you activate them by using lipopolysaccharide. And this activated macrophages resembles inflammatory like condition, they are known to produce proteolytic enzymes. Then we wanted to see this in situ generated proteolytic enzymes by activated macrophages can release the drugs or not. So to test that, we took the conditioned media from activated macrophages and also healthy non-activated macrophages, which resembles healthy condition, and exposed the gels to see the release of the drugs. As you can see, the conditioned media that was taken from the non-activated macrophages did not have much release because it's almost similar to the buffer. But whereas the conditioned media that was taken from the activated macrophages was able to degrade and release the drug. Not only that, at the different time points, if we give a fresh pulse of this activated conditioned media, you see the dose responsive manner, the release of the drug, which is clearly suggesting that this material has the ability to respond to the inflammatory conditions and release the drug in a on-demand manner. So once we have that, we went and tested their ability to protect the transplanted organs without causing other toxicity. So for that, what we have done is we did entire hand limb transplantation from completely mismatched rats, from brown Norway rat to Lewis rat. And the post-transplantation, you can take these injectable hydrogels and keep them as, inject them as a subcutaneous depose, and you measure the survival of the graft. Okay, I'll just quickly summarize the results. And for example, if you see here, post-transplantation, if you don't give any treatment, right, or you just inject only gel without having any drug encapsulated within it, as you can see, within 11 days, you see the rejection of the graft. You can see, severe inflammation and grade 2 rejection. Now, as a control, what you take only drug, immunosuppressant drug, and do the one injection without putting into gel, right? In that case, again, you see like in 33 days, they start rejecting the grafts, and you see severe damage of the muscle and tissue damage you see, and grade 3 rejection we see. Now, what you do is like take the same dose of drug and encapsulate into the gel and do the one injection. In that case, you can see like there is no sign of inflammation, no sign of rejection, hair grows back, and in fact, the animal gains its function, and now it's able to move around by without having any stress. And not only that, what we tested their entire systemic immune system, their immune system is still active. And also we tested their entire internal organ system to see the non-specific toxicity that is typically occurs with the standard therapy. And we haven't found any non-specific toxicity as well. So which is clearly suggesting the advantage of having this disease responsive material to protect the transplanted graft. Then we wanted to see how long we can protect them. So to do that, as I mentioned, if you do the single injection of the just drug, they survive for only 33 days graft. At the same dose, you put it in the gel and do one injection post-transplantation, survival goes to 190 days. And we have done extensive pharmacokinetics to see that drug availability and so on. So based on that, if you do the periodic injections, for example, if you do one injection after the surgery on day 1, and second injection after 3 months, if you do, survival went to 260 days, and without having any other side effects and so on. So which is clearly suggesting that if you do this periodic injection, as long as animal is alive, it can protect the grafts, right? So that's a great improvement in the current situation. Then we wanted to see whether we can prevent the opportunistic infection that occurs because of in the standard therapy, because of their entire immune system gets compromised, right? So to test that, what we have done is 2 groups of animals got transplanted. In one group of animals received or gel therapy, where like you do 4 injections, like with a interval of 70 days, and you terminate with the 280 days study. And in other case, second group of animals receive systemic tacrolimus, which is much lower dose than the standard therapy. Then terminate in 280 days. In both cases, if you see, the survival of the graft is same. But interestingly, all the animals that received standard therapy developed opportunistic infections. But whereas none of the animals that received this on-demand drug release system developed any opportunistic infections, which is clearly suggesting the advantage of having this on-demand delivery systems to protect the grafts without having any side effects that is clearly we see in clinic today. Then we also went and demonstrated this efficacy in large animals like swine models as well, and we see complete protection in these animals as well. Now we are putting a clinician together to go for the clinical trials pretty soon. So that brings to the second example. So how we can use this inflammation responsive drug delivery systems for the treatment of inflammatory arthritis. As you know, arthritis is primarily affects all the joints, and people have to take these very potent anti-inflammatory drugs to high doses systemically in order to have the efficacy at the joints, right? But unfortunately, they comes with the serious side effects, systemic toxicity and because of their high systemic toxicity, there are multiple drugs like Viax and Bextra was recalled from the market by FDA. So to overcome this systemic toxicity, people have started injecting directly in the joints through intra-articular injections. But like, unfortunately, it wasn't very effective, because if you see, when you inject these drugs, like, they can diffuse from there. Like, there is a series of drugs have been tried for these local injections, but like, maximum they can stay is like, maybe like 10 minutes to 5 hours, right? And they diffuse from there. And according to protocol, you can do only 2 injections per year, intraarticular injections. Otherwise, we end up damaging the cartilage and so on. So that's why then people have started encapsulating these drugs into a variety of microparticles, nanoparticles, and gels and so on. But unfortunately, the overall therapeutic outcome wasn't great, because as I mentioned, this inflammation is highly fluctuated. It can come and go. But irrespective of having disease or not, they release drug and disappear, right? So because of that, it wasn't very effective. That's where we thought having this inflammation responsive material might have the great advantage in the treatment of arthritis. So again, we took the drug called triamcinolone acetonide, which is prescribed drug for arthritis. And again, we optimized conditions to generate this injectable hydrogels. And then we went and tested their ability to release the drug in response to the proteolytic enzymes. As you can see, a very good responsive manner, they release the drug, not only with the isolated enzymes, but even for the inflammation also, they are nicely responsive, right? But more critically, we wanted to see, because these inflamed arthritis patients, their entire synovial fluid in the joints get inflamed, right? Then we wanted to see this inflamed synovial fluid also can release the drug in a responsive manner. So to test that, we collected the synovial fluid from arthritic patients as well as in healthy volunteers and expose these gels to those synovial fluids to see the release profile. As you can see, if you see this yellow line, okay, where synovial fluid that was collected from healthy volunteers were given, in such case, you don't see much difference of the release kinetics compared to the buffer. Buffer line, that black line is just underneath this yellow line, right? So you don't see the induced release at all. But when you take the synovial fluid from arthritis patients, when you expose the gels, you can see the release of the drug much higher doses. So which is clearly suggesting that the inflammation occurs in the synovial fluid can actually release the drug in an on-demand manner. Then the next question comes, in the absence of this inflammation, can they remain stable at the site of injection so that whenever the disease comes, they can release the drug? To test that, what we have done is these gels were fluorescently labeled. In a healthy animals, they were injected in the knee and followed for a couple of weeks, and you don't see any release of the drug. And as you can see, like despite of this animal is moving mechanical movement, this gel is confined to the place of injection. It's remained there. And then we can induce the arthritis in these animals by injecting KBXN serum. And when you inject KBXN serum about 37 microliters, they're known to produce mild arthritis from day 6 onwards. And as you can see, when you inject this serum after day 6, you can see slowly depletion of the signal. But like instead of 37 microliters, if you inject double dose of that is like 75 microliters of serum, it is known to produce severe arthritis from day 2 onwards. So when we did that, as you can see, after 2 days, and a rapid depletion of the signal, which is clearly suggesting that these gels have the ability to remain at the site of injection without degrading if you don't have any inflammation. But whenever the disease comes, that can react and release the drug in an on-demand manner. So once we proved that, we went and tested their overall therapeutic outcome of that. So once we induced the disease, then we injected both either free drug or the same dose of drug encapsulated into the gels and injected. When you measure the therapeutic outcome, one of the way to measure is the swelling of the palm. The least swell is the better therapeutic outcome. You can see when the drug was encapsulated in the gel has much better therapeutic outcome compared to free drug that was injected. And the same manner, if you see the clinical score also represents same manner. So which is clearly suggesting that having this injectable or inflammation responsive material has a much better therapeutic outcome in the treatment of arthritis. So that brings to the third examples where how we can use this for the treatment of IBDs. But what we have done is we added another layer of design here, because it's not only inflammation responsive, but inflammation targeting material, which is very important because, as you know, like inflammatory bowel disease, IBD, comes in two variants, Crohn's disease and colitis disease. And where in these both cases, you have the patches of inflammation, right? It's not entire tissue becomes inflamed, but you have the patches of inflammation you see. And today, all the drugs what we take, and same dose goes to the inflamed tissue as well as the healthy tissue, right? Your adjacent healthy tissue. But unfortunately, these drugs have the side effects. It's irony that these drugs, if it goes to healthy tissue, it can cause inflammation. So basically what we are doing is we are converting healthy tissue into inflamed tissue into healthy tissue. That's why the overall therapeutic outcome is very poor. That's why like people take medications for like, 6 months to years when they get this colitis and Crohn's disease. So the best way of treatment of or delivering drugs for this IBD is if you have a material which can be stable in your GI tract, but which can go and sense the where the inflammation is there and specifically adhere to those tissue and remain there and release the drug. In that way, only inflamed tissue can get the drug, but not the healthy tissue. But the question is how one can induce this specific glue-like property for which can bind to only inflamed tissue, right? So if you see, when you have this inflammation, and because it tend to break the barrier and you start the infection there, right? To curb the infection, your immune system start producing this high concentration of bactericidal proteins and antibacterial peptides. And all of them are cationic in nature. So because of that, if you see, entire tissue becomes like a cationic charged bed, right? So then the question is, can we use a simple Coulombic interaction? That means like if we generate a particle which are like, complementary charge, negative surface charge, can they able to go and bind to the cationic proteins which are resides at the inflamed tissue and release the drug? So that's the overall concept what we have been developing. Okay. So then again, we selected another amplifier which can self-assemble to form these micro-sized fibers and an encapsulated drug. Again, we chose the drug dexamethasone, which is prescribed drug for IBDs. But this, upon the self-assembly, it also should render the negative surface charge, right? And also they should able to release the drug in a response to inflammation. So by putting all this together, we chose the amphiphile ascorbyl palmitate because it also has a negative charge. So the overall fibers can give negative surface charge. As you can see here, all these micron fibers, all of them have high negative surface charge. Then once we went and tested again their ability to release the drug in response to inflammation and so on. Then we wanted to see, will they able to bind to the protein that are overexpressed at the site of inflammation? To test that, we took the transferrin, which is known to overexpress at the site of inflamed tissue, and coated on the plate. As a control, you take the mucin, which is healthy mucus protease, and coat on the plate and incubate with these fibers, which are again fluorescently labeled, and wash them and see how much fibers attached. And as you can see, as we expected, all these negative surface charge particles efficiently bind to the transferrin protein, which are cationic, right? But it's not very surprising because you have a simple Coulombic interactions play a role when you have the cationic proteins and you have the anionic charged particles, they should be able to bind it. But the challenge comes, can it be done and can you achieve such level of specific binding in vivo condition, right? So to test that, what we have done is we took healthy animal and you give them dextran sodium sulfate for 7 days, then that generates the develops the colitis. And for these animals, you give these fluorescently labeled particles, and after administration of the particle, after 24 hours, you surgically remove these colons, okay, and image them, okay. As you can see, all these are colons that were taken from healthy animals, you don't see any sign of fluorescence particle. That means because you have high bowel movement, right? So all the particles might have already excreted away. But all the colons that were collected from the colitis animals, you can see patches of red signals, which means these particles able to identify the inflamed tissue and bind there. Despite of this high bowel movement, they still remain there, right? Which is clearly suggesting that these particles have the ability to go and specifically bind to the inflamed tissue and remain there. Then we wanted to see, once we have this level of targeting efficiency, what is the overall therapeutic outcome, right? And as you can see, once you develop the disease, you give the either free drug or encapsulated into this inflammation targeting material. And the overall therapeutic outcome by histopathology score, you can see much better when you encapsulate it into this particle compared to the free drug, right? But the second question comes, uh, although we know that this can bind to rodent tissue, but can we get the specific adherence to human colitis tissue as well, human inflamed tissue as well? So to test that, what we have done is we collected the tissue from the biopsy through colonoscopy from the patients who comes from the colonoscopy for colitis. From same patients, we collected the tissue from the site of inflamed colon. From the same patient, we also collected tissue from the healthy colon as well. And the ex vivo, you incubate with these fibers and wash them extensively to see how much fibers they still remain. And as you can see, all the fibers, they are able to bind to inflamed tissue, but whereas in healthy tissue, they don't bind much, okay? Even if an individual patient, if you check, for example, this patient in the normal tissue, if it binds X amount, same patient, inflamed tissue has much higher binding. And the same manner, every single patient we tested, we see the same trend. So, which is clearly suggesting that indeed this material had the ability to bind to the human inflamed tissue as well efficiently. So we assume that probably we will have much better therapeutic outcome and in a manner to target the inflamed tissue in human as well. So now we are putting company together to take this forward, and we're going for the clinical trials pretty soon. So those are our effort in our therapeutic biomaterials. So I thought, given taking inspiration from Milan efforts to develop very reliable diagnostic kits to fight with the current pandemic. So we thought what we can do to contribute to current pandemic. So that comes into our prophylactic biomaterials, how to prevent the diseases, right? So I quickly tell you one example in a couple of minutes, how we have developed germicidal antiviral masks to protect viral infections. As we know, now during this pandemic and all, wearing mask and all, it becomes a norm, and it becomes a regular habit. But all the masks what we wear, are they safe, right? If you see all type of masks, including N95 masks, surgical masks, or cloth masks, all these masks act as a filtration devices, right? They filter the germs to prevent the infection, right? But there are even clinical studies have been shown recently that all the viruses, even including SARS-CoV-2, which is causing COVID-19 pandemic, can remain active on this mask, and this cloth, it can remain active for up to 7 days, right? So just we wanted to see how it is true. So for example, this is like a simple agar plate assay, where you can see when you don't have any bacteria, and if you give an X doses of bacteria, you can see the growth of bacteria here, right? Then to test that, what we have done is we asked people to wear N95 mask, surgical mask, and cloth mask for 6 hours. And these masks were, touched on this agar plate for, a minute or so, and removed, and see the bacterial growth. As you can see here, all these masks have contains high amount of load of germs, and as you can see, the huge bacterial growth on this, which is clearly suggesting that this mask can actually filter them, but they still remain active on the these cloths. So because then when people touch to adjust and all, people can still get the infection, right? So that's where we thought, can we develop an antiviral material which can prevent such accumulation of the bacteria and virus? So what we have done is we quickly developed a new class of molecule called quaternary ammonium salts, and we developed a process to make this part of integral part of fabric, and this activated fabric can be stitched into any kind of PPEs. And how they work is like, these materials, once it is there on the cloth, if any virus comes, it interacts with the membrane of this virus and rupture the entire membrane, the viruses or bacteria, and deactivate them. That's how they do that. So then we developed and we formed this, then we quickly wanted to test. As you can see, your regular masks and all, like, even after wearing just 6 hours, you can see how much amount of bacteria gets accumulated, right? Now, we developed the technology from the company, licensed to the technologies to color threads, and they licensed to the Van Heusen and so on. And once they developed the mask, right? And we asked same mask to wear people like either 6 hours or even 18 hours. And same manner we tested with other, and you can see there is no sign of any bacterial growth at all. So which is clearly suggesting that, these masks have the ability to deactivate any germs that it comes in contact. And in fact, we did tested with even SARS-CoV-2 as well in a approved labs and all, like we see the same results. So that's where our quick contribution towards the current pandemic situation. And I thought, like, if you see the line of speakers today symposium, there's a great emphasis great emphasis put it on microbiome, like, including oral microbiome and all, like how it could be used in medical applications. Then I thought, like, we quickly share some of our recent effort, how we can use a gut microbiome as inspiration to develop the drugs. Okay, so this is like our effort in developing new chemical entities as a drugs for treatment of inflammatory disorders. As I said, IBD comes in two variants, right? Chronic, the colitis and Crohn's disease. And today's standard therapy, if we see for these disorders, are either steroidal or non-steroidal based anti-inflammatory agents or immunosuppressants. But, like, both type of drugs, they just manages the disease, but they don't cure it, right? Because that's how, like, people have to take like years when the disease comes. So we thought, like, can we develop much more efficient drugs, not only just managing the disease, but like having the cure, right? But even to develop new drugs, we thought, like, where to start from, right? Classical medicinal chemistry approach is like, you take like millions of libraries of random chemicals and screen them and find the heat and do the medicinal chemistry, right? But what we thought, like, why don't we understand the local environment where the disease occurs, take the cues from there and develop the drugs, right? And as you know, like, our intestine is harbors like, billions of bacteria. It's called microbiome. And there is a huge studies are known that this bacteria microbiome has the both beneficial and detrimental effect, because they're known to have the huge beneficial effects to the body, but at the same time, when you have the dysbiosis of this microbiome, can lead to huge side effects as well. And there is a growing interest or growing evidence clearly suggesting that your microbiome controls the function of major your pivotal organs as well. So, but how they control your body function, right? How they modulate is like, any diet what you take, this bacteria, they break down into metabolites, and these are small molecular metabolites, active metabolite they generate, and through this metabolite they control the function. So we thought, if we can understand this function of this metabolite very clearly, we can take that information and develop the drugs. That's the approach what we are taking. And as we know, when you take the phytochemical rich diet, from pomegranate or like cherries and berries, they're known to have the beneficial effect, because when you take this diet, they start breaking down and form this polyphenol based elagitins. And in the colon, like when you have this microbiome, they start degrading these polyphenols and make further metabolites called irolithins. Okay, and what we have done is we have synthesized all these metabolites in laboratory, and individually we also tested their efficacy, and we found that urolithin A is the most active metabolite because traditionally these are known to have anti-inflammatory properties, right? So we thought, like, can we synthesize this molecule and use this as a drug, right? And we tested that in vivo, but unfortunately nothing worked. In fact, several other people have been trying in the field to get the in vivo efficacy, but it doesn't work. The reason is, if you see this molecule, right, like it's a two phenol rings, it's a biphenol ring connected with a cyclic ester, it's a lactone. But like when you take it in a oral route, like in your severe pH and digestive enzyme can easily break it. Once they break it, it breaks and it becomes like a propeller structure, then it's not active. That's why you don't see the efficacy. That's where we thought, can we stabilize this and target to the inflammation side? As I mentioned before, we have the experience to develop this nanoparticles which can target the site of inflammation. So we developed the injectable, or like oral nanoparticles which can encapsulate this drug, or like a urolithin A, which can prevent from the degradation in the stomach and can go to the inflammatory site specifically and release there. When we did that, you develop the disease. As I mentioned, if you expose to DSS, the animals develop the disease, and you give the different doses, the 4 milligrams per kg, where either free drug was given orally or the same dose of the drug given through nanoparticles. And then if you measure the overall therapeutic outcome, as you can see, because when you have the colitis, your entire colon shrinks because of the inflammation. As you can see, this is a healthy animal, but when you have the disease, colon, you lose the colon. And but when you give the only drug alone, or just blank nanoparticle, you don't see any protection. But when you have the same dose of drug given in the nanoparticle, you can see complete protection of the colon. And even if you see the histology of the colon, this is a healthy animal gut, where you can see a nicely have the villi structure and crypts and so on. But during the disease conditions, completely breaks off. We don't see loss of these structures. But whereas in the presence of this nanoparticle urate, it's completely prevented that, and it's completely protected the same structure. And then to test the severity or most efficiency of this, what we have done is, after completely developing the disease, we gave one single dose of this nanoparticle, single dose. And even when you give the single dose, you can see a complete protection of the animal. It's almost reverse the disease, right? And because you can see gut morphology is as good as a healthy one. So we are super excited about it, right? Like, with a single dose, it can cure, almost like reverse the phenotype. But at the same time, it really bothered us because, see, this compound is known as a anti-inflammatory, right? If it is anti-inflammatory one, inflammation is just an effect, right? And because colitis is a chronic, if it is taking care of only inflammation, how it can completely reverse the phenotype? That question was bothering. If you see your gut morphology, right? This is your epithelial cells barrier. You have all these epithelial cells tightly stitches together with these proteins called tight junction proteins. And that's how they form the barrier, and they protect all the commensal bacteria coming through and exposing, right? And that is in your healthy conditions. But in colitis, what happens? They lose tight junctional proteins, the stitches they stitch together, these epithelial cells. Because of loss of these tight junctional proteins, you have this leakage or barrier leak. Because of that, all the bacteria keeps getting exposing. That's where your immune system start fighting against it. So that's why you have the continuous persistent immune activation and the chronic inflammation. So we thought this compound, if it is just not only just anti-inflammatory, if it is reversing phenotype, means probably this might be somehow stimulating your gut barrier to restore it. That means like it is stitching back, so that like there is no barrier leak, so that you don't see further inflammation. So then we want to go and test this hypothesis. And not only that, as you know, this parent compound, it's highly unstable. So we wanted to make the stable analog and do the medicinal chemistry. Systematically, we did this medicinal chemistry. One way to do is we remove this cyclic ester and made the cyclic ether, and which is completely stable in pH and so on. So this is a typical synthesis. As you can see, parent compound is completely unstable if you put it in pH and 2 or digestive enzymes, whereas synthetic analog is not. Then we went and tested their ability to increase the tight junctional proteins. As you can see, in the gut epithelial cells, when you expose to the compounds, then you measure the tight junctional proteins such as claudin 4. You can see when you give these compounds, you see increase of tight junctional protein production, and which is true for all other tight junctional proteins like claudin 4, aclodin, zonoclodin, and so on. And then we went and tested their ability to in vivo efficacy as well. So again, after developing this is when you treat them, we gave these both compounds. One way to measure the in vivo barrier function, okay, because we wanted to see whether gut is getting repair or not. So one way to do is, take this fixidextron, it's a polymer, fluorescent label polymer, give them oral gavas through animal, and collect blood and measure in serum how much this polymer came. Okay, if you give this to a healthy animal, you don't see much amount of this polymer coming into the serum because you have intact gut and there is no leakage. And this is polymer is a big molecule, so you don't see. But when you have the disease, you can see huge amount of polymer comes into the circulation. That means like there was a leakage in the barrier dysfunction happened. That's why you see. But when you treated with these compounds, you see complete the restoration of barrier. You don't see that much amount of polymer coming into the circulation, which is clearly suggesting that indeed this material has the ability to seal the broken barrier and prevent that. And we went and tested their in vivo anti-inflammatory properties as well. You can see they significantly reduce the production of pro-inflammatory cytokines such as IL-6 and IL-1B and so on. And again, if you see the gut morphology, it completely protects new synthetic analog as well. But not only in the acute disease, but even in the chronic model we tested, where periodically exposed to the DSS several weeks. And even in that case, when you give the treatment, again, the barrier protection, you see 100% protection when you treat with these compounds. Again, we went and tested quantified the tight junctional protein expression in the colon tissue. You can see much higher when you treat with these compounds. And again, they are anti-inflammatory and they protected the they protected the gut morphology as well. So, which is clearly suggesting that overall this compound has the ability to stimulate your gut epithelial cells to stitch back and repair the barrier dysfunction, so that have the much better therapeutic outcome. So that's where we are. Just to summarize the overall, and in therapeutic biomaterials, we could develop this disease responsive biomaterial concept, and which could be used for multiple medical applications like protecting transplanted organs and treatment of inflammatory arthritis and IBDs. And prophylactic biomaterial, in fact, I haven't spoke today, is like we also developed a series of materials which can protect farmers, Indian farmers, from pesticide-induced toxicity. Maybe some other time we can speak about it. In the new chemical entities, like in drug discovery, we by understanding the microbiome and their metabolites role, we could able to develop small molecular drugs to fight IBDs. So, but our lab is not only interested in just conceptual molecules, but we also want to go to the end-to-end journey, because we want to translate this technology into the clinic. But this is the classical depiction of valley of death that exists in the translational science, because it's ironic that majority of the breakthrough science, they remain in the laboratory, they are not able to reach to the people, because that your discovery should be converted into technologies to reach to the people. So however, this gap can be bridged by practicing very efficient entrepreneurship program. our lab also equally interested in developing entrepreneurship program here. So as that effort, what we have done is putting all this disease responsive biomaterial platform. We formed a startup company in Boston, Alavia Therapeutics, and now it's been 4 years now. We have done like mostly all the IND enabling studies have been done. Now, very soon the phase 1 clinical trials is going to start now. In the prophylactic biomaterial, as I mentioned, like, anti-pesticide therapeutics, which we didn't speak today, but based on that, also we formed another company in Bangalore, where we are going to start human clinical studies supposed to be last year, but like we couldn't do last year. Probably this year we'll be doing. And again, based on our antiviral or germicidal fabric technology, there's a company in Colored Threads in Tirupur transfer the technologies, and they partner with the Vanus, and then we launched antiviral mask and hoodies, and your entire track suits and pants and t-shirts are already being launched in the market. And for the based on your drug discovery program, again we formed a company in Boston. It's called Arthur Therapeutics, and as we speak, like all the preclinical IND enabling studies are being done there. So our idea is to go to the phase 1 clinical trials from these drugs from either end of this year or probably next year. So with that, I'd like to stop here. I'd like to thank all the people who did this exciting work from this entire organ transplantation work done by Ashish. Is also a Shastra registration student. He will be coming and presenting pretty soon there. And there are very talented postdocs and group of PhD students and JRFs worked with the various platform technologies. This entire credit goes to them. And I was blessed with the very productive collaborators. They are source of the knowledge where we learn a lot. And I'd like to thank various funding agencies for their generous funding. And I would like to thank all of you for your kind attention. I'll be happy to take any questions and have any discussion so far. Thank you so much.
Thank you very much, Professor Praveen Kumar. There are quite a number of questions. If you look in the Q&A, can you see them? I can read it otherwise.
I'll check that.
Yes. There are some of them are very technical questions. So.
Okay, so maybe I'll stop sharing probably then.
Yes, probably. But you can also, we have only 6 minutes or something.
Sure,
you can answer later too, typing.
Okay, I think the first question from Pankaj Yadav says, how challenging is to apply the outcome of your work, particularly the mice work, limb transplantation limb into human system?
Yes.
Also, can you give the idea of efficacy? Yes, you see, not only for this system, but like if you take any translation science, translating from preclinical model to clinical model, it's been a great challenge, right? But that's where it becomes like how meticulously each step you optimize in each stage and bring to the clinic. So we see a great sign. I think we are super confident. The reason is this has been from day one, we partner with clinical collaborators. There's a clinicians who do hand transplantation regularly. In fact, there's a father of hand transplantation who started. They are the part of our team. And so they know exactly what works, what they don't work, and what clinical condition they need. So by keeping that feedback always, we try to develop the systems. And now we have optimized so much that even the very robust rejection system in swine models, because if we can achieve the protection in swine models, there's a huge chance that it can work in human as well, and we could see that in our swine models. So we are super optimistic in that. But surprises always there. One need to be aware of that too. So the second question, it says, do you give the IT head? Oh, sorry. This is from Emma Bhagavati. I could understand that the TA gel remains the site of inflammation and from the animal model, It's also clear that the drugs effectively released in response to the severity of the disease. How long does the TA gel remain at the site of arthritis subsides? Yeah, so it depends upon the, again, like the disease. And if you don't have any inflammation, we see almost like 3 months in vivo, they remain there. Okay. But when you have the inflammation, it's again like a depend upon the level of inflammation. It controls how fast it goes, how slow. So what we are doing is like, but then it varies from patient to patient, right? Because they have different inflammation. So to monitor that, now what we are doing is like we are labeling these gels with a fluorescent dyes, where you can do non-invasive just imaging. Then anytime you can image and see how much gel is remain there. So depend upon that, whenever you see the depletion of the gel, one can get the fresh dose of that. Hope that answers your question. So the next one, do you give the IT hydrogel microfibers by oral or intravenous injections? No, these are not intravenous injections. These are oral and the rectal. Right now, either rectal or oral give. But like these systems also can be, it will work for your IV also, but like these are not designed for that, but that will work as well. How is the hydrogel is being eliminated? What is the clearance of the hydrogel? With the single administration of hydrogel would serve as a better option. Oh, great. So what happens, like, hydrogel, again, we design into a small molecule which self-assemble to form these hydrogels. And these molecules, the one way to pick a molecule from this FDA approved GRAS agents are, because these molecules, how they degrade, how they eliminate is very well studied in human system already. So that's why once they get degrade, it's again, like it just generates a fatty acid and they triglycerol moiety, which is clearly excreted from the body. So whether it's required single administration or the multiple, we believe it needs the frequent administration. I think in animals, we see once in 3 months it works. But like, based on our dose calculations and the clinician's output, the current guess is like, they might need once in a month, one dose of injection, you might need it. So what is the solvent for the polymers? Do they have a tendency to agree? I think this is a little bit general. Depend upon, what material you take, what polymer you take, one need to optimize. But if you're using for the biomedical application, you tend to optimize everything in water, aqueous based system. Maybe some of them, like, maybe friendly solvents like, DMSO, a part of your composition can have the DMSO or other alcohols, so on. Another question is, there is any chance by which the drug can be designed to release the beyond a certain threshold of inflammatory response, as inflammation aids the normal function of this pretty good question. Yes, that is optimized, because you see, even without having any inflammation, right? Even at the base level, we do have a very low level of proteolytic enzymes in the body, right? So that's why these materials in the composition, I didn't go through like entire data, but like they are optimized in such a way that they have to cross a certain threshold of the concentration of this proteolytic enzymes. Then only they start degrading and release the drug. So that has been optimized. And so that based on the composition, what we take, that is, but that's been optimized. Yes.
Yes, sir. Thank you. I have one question. Sorry.
Sure.
Say if we are injecting hydrogels into the synovial fluid, and if it is especially to a human who's into the knee of a human whose weight is is to be held on the knees, will there be any problems to the cartilage that is present in the knee because of the hydrogels, physical damage?
Yeah, in fact, that's a great point. It is not. The reason is we kept this in a mind. In fact, the gels even have the dual purpose, because your mechanical strength of the gel plays a role. Because if it is stiff material, it can actually cause damage of cartilage. But these materials are designed in that such a way that, in fact, it can even act as a lubricant. it's a very soft material. Your mechanical properties are adjusted in such a way that it doesn't cause any mechanical impairment to the cartilage. So we don't see, I don't think there would be any adverse effect. And because even in animals, when we see like, without disease in healthy animals, in several weeks, they remain stable there. And because they keep moving and running around, like, that mechanical strength also, it doesn't really cause the degradation or anything. So in fact, what we have done, okay, I didn't show the data again. So these were implanted for like few weeks. And then we sacrifice the animals, and we section the tissue, like the knee joints, and through histology to see where is there any damage or any inflammation, what is the response to this material. And we didn't see any such adverse effect at all.
Thank you very much, sir. I think we are running over time. So I would just thank you very much for enlightening us with the conditional release of drugs in space and time. And I think we all will draw a lot of inspiration from you about the energy with which we have to work to take from lab to the market and to the required or the needy people. Thank you very much, sir. It's very pleasing to have you out with us.
My pleasure. Thank you so much. My pleasure. Thanks.
Sahna, over to you.
Yeah, thank you, Dr. Praveen and Dr. Chandramohan for this for sessioning the chair. Now I invite Dr. Bhuvana Kamath to introduce our next speaker and chair the session. Dr.
Thank
Bhuvana,
you, Sahana. Yeah, thank you, Sahana. It gives me great pleasure to introduce our next speaker, Dr. Karishma Kaushik from the Institute of Bioinformatics and Biotechnology, Savitribai Phule, Pune University. Dr. Kaushik comes with a very strong medical background. She has completed her MBBS from Dr. D.Y. Patil Medical College, Pune. Then she's completed her MD in clinical microbiology from the Armed Forces Medical College, Pune. From her medical career, she has then gone to pursue her research career, which led to her PhD degree in molecular genetics and microbiology from the University of Texas. She has been awarded several laurels and fellowships. One, to name a few, are the Ramalinga Swamy Fellowship and the Innovative Young Biotechnologist Award. Her current research area is in human relevant infection biology, to which she has several publications, mostly relating to chronic wound infections and biofilms. Her research looks into bioengineered platforms, antimicrobial approaches, and alternatives to animal testing to answer the many questions around infection biology. Not only is her passion in research, but Dr. Kaushik is actively involved in several community engagement programs. Her group has recently founded the science outreach program called Talk to a Scientist, where she's ensured her passion for research is not limited to her group, but is effectively communicated and shared with society. She is also featured in Nature Careers, where she has shared her insights on work-life balance. So with no more delay, I'd like to invite Dr. Karishma Kaushik to give her talk on what is a pathogen without a host, incorporating host elements in infection biology studies to make them human relevant. Over to you, ma'am.
Thank you so much, Bhuvana. And before I start, I have to say that was the best introduction I have ever been given. So I need this recording before anything else. I need to hear this often. Thank you very much for it. It was wonderful. Very, very beautifully put. Thank you.
Yeah, thank you. And I think your CV deserves that.
Yeah, that's very sweet of you. Thank you.
Over to you.
And thank you, everyone. Quorum Sensing Lab, Shastra University, and your collaborators in Hong Kong, the SPARK program, for this opportunity. As Bhuvana mentioned, I moved back to India in 2018 on the Ramalinga Swamy grant. And we are a young but very dynamic research group. And we look forward to a long innings in Indian science and contributing to science on global platform. So this is where you can find me, my email, website, and Twitter. Please feel free to get in touch. We are always excited about engaging with the community and evaluating opportunities that could come our way. All right, so I'm going to give you a snippet of the projects in the group, some of which have been published, some of which we are wanting to publish soon. So inputs from you will be welcome. Inputs, suggestions, questions, we look forward to all of that. Okay, so I study infections. I'm a clinical microbiologist in another lifetime during my MD. And we typically, we've studied infections in the lab, like microbes growing in these tubes. And when through my PhD career, I realized that this is a hugely reductionist approach because there's no host. We are studying it in very optimum circumstances where there is no host. So a huge focus of our group, one of our focus areas, which is a major focus, is to incorporate host elements in studying infections and to try and do this on the bench. So we don't need complex animal models, but we do this on the bench using in vitro approaches. So let me take you through this story of 2.5 years that we've set up our group, just to shake things up a little bit to make sure that the audience is all still awake and listening to me. Have we all had a wound sometime in our life? Yes, everyone's fallen down. Hopefully not in the near future, but yes, thank you. I like the raised hands. Thank you. So everyone's had a wound. Most often than not, that wound has healed and we have just been left with a scar. But we may be the few for whom the wound healed, but there are circumstances, which is ever increasing situations where these wounds do not heal and they become what we call chronic or non-healing wounds, which is a burden to the healthcare system because of their huge morbidity. And believe it or not, this is considered a silent pandemic. Silent because we don't see this very often, like the pandemic we are witnessing or going through right now. This is a burden on the healthcare system. It's a drain on our resources. And this is only being fueled with an increase in longevity, life expectancy, diabetes, obesity, sedentary lifestyle, immunocompromised states. It's only increasing this problem of non-healing and chronic wounds. So when we got a wound and it healed, it went through this beautiful orchestrated sequential process of formation of a clot, immune cells infiltrating the site of injury, proliferation of granulation tissue, which is our host cells, eventually closing the gap and leaving a scab. The critical stages in this wound healing process is this inflammation to proliferation phase. So what can happen in this phase that leads to non-healing wounds, and now it's almost synonymous with non-healing wounds, is formation of biofilms. So preaching to the choir maybe, but one line introduction of biofilms, they are these extraordinarily beautiful, structured, three-dimensional microbial communities, sometimes bacteria with different species, sometimes bacteria and fungi, which are typically seen in the wound. In the context of wound infections, these are seen as aggregates embedded in host tissue. So here you see the blue DAPI stained host nuclei, and you see these red, fluorescent red bacterial aggregates. Here you see them as green aggregates. And this is some of our work trying to develop mixed species aggregates. Pseudomonas is green and staph is your red cocci, similar to what you see in the wound. So these biofilms, bacterial biofilms, are considered the most important cause of non-healing wounds. And Pseudomonas aeruginosa and Staphylococcus aureus, often found together, are the most common wound pathogens implicated in this non-healing state. So wound infections, wound biofilms are most often polymicrobial. Okay, so however, the biofilms obviously, unlike the way we study them in 96 well plates, in polystyrene 96 well plates, they don't exist in isolation in the wound bed. They are closely interfaced with host elements. And you can think of the wound like a two-compartment system. The upper compartment having host elements like fibroblasts, spindle-shaped cells, cobblestone-shaped keratinocytes. So this is your granulation tissue that I talked about, the proliferation. These cells secrete matrix like collagen, elastin, fibronectin, fibrinogen. Biofilms form on this bed of host and matrix elements, and the entire wound is also subject to mechanical or biophysical forces like shear stress, tension, compression. This upper compartment of the wound bed interfaces with the capillary, through which you have immune cells like neutrophils and macrophages in the wound tissue and lined by endothelial cells. So you can really think of the wound bed as a two-compartment system. And you know where I'm going with this in trying to recapitulate these two-compartment system in the lab. So bottom line is biofilms exist in a milieu of all of this. They're not isolated. And this milieu is active, it's dynamic, and they closely influence each other. For example, biofilms secrete small molecules, metabolites, presence of oxygen gradients, production of reactive oxygen species, enzymes like proteases, especially MMPs, various virulence factors produced by bacteria, all of them influence each other and result in a characteristic cellular, biochemical, molecular milieu. So you can also see where I'm going with this in trying to recap this milieu in an in vitro condition. So obviously biofilms are not isolated. They exist in approximation with host elements, and together they actively influence each other to give us a wound milieu. All right. So what is the problem then? We know so much about them, it seems, right? I get very excited when talking about wound biofilms. The problem is we treat them in a very generic approach, one size fits all. What do? We bombard them with antibiotics, anything that we can lay our hands on. Unfortunately, so there's a heavy reliance on antibiotics. Why it works is uncertain. More importantly, why it fails is even more uncertain. And so it's not a very deterministic approach to treating wound biofilms. As a result, that ends up with long-term treatment, emergence of resistance, and a further burden on all the healthcare resources and everything that I talked about. So while there have been lots of lab work, this is a review where we looked at all the non-conventional antimicrobial approaches to look at the wound biofilm microenvironment, and there are lots of lead molecules. There's blue light therapy, pH modulation in the wound bed, nanoparticles impregnated with various antimicrobial compounds. And the problem with this is a lot of this works in this early preclinical stage. And like Praveen mentioned, and I think it's a good segue into my talk, the big leap is moving from preclinical to actually making it work in clinical practice. And here you have almost this valley of death, this cliff where all these molecules show promising results in early stages and then never make it further down the drug development pipeline. So one of the reasons for this is all these molecules are evaluated under very optimum laboratory conditions and not really under host or human relevant conditions. And this, we think, is a major gap that needs to be filled. So how have people approached filling this gap? I'll talk about that shortly. And the rest of my talk will focus on chronic wound biofilms as a case study to incorporating host elements in vitro studies. So how have people tried to fill this gap of trying to study wound biofilms or wound infection states in the lab? So let's look at so obviously there's a mixture of in vivo, ex vivo, in vitro approaches. Let's start with in vivo. So we know that these are your animal systems. Most often we use small animal models like rodents, mice, sometimes porcine models, but rearing pigs in the lab is a huge challenge. So apart from well-known ethical limitations with using animals in the lab, there are certain challenges very unique to the infecting an animal, which is most animal houses in India don't allow you to infect an animal. Even if they do have the facilities to rear animals, they would prefer you not to infect them. They wouldn't give you permission to infect them. So this is one challenge. Then there's the ethical challenges. Then there are technical challenges of actually being able to work with these systems. However, there is a major scientific challenge when it comes to using murine models for wound studies. And that is that mice don't heal wounds the same way as humans. And I don't mean this in a philosophical way. I mean this in a real way. Mice heal wounds by contracting the edges of the wound. They do not proliferate the wound with granulation tissue the way we do. So obviously, there is a fundamental difference in approaching wound infections in mice and in humans. And therefore, more than philosophy, mice are not humans, and therefore they pose limitations in translating these approaches. Of course, they are not high throughput, and there are huge cost implications as well in trying to infect these mice and work with them. So in vivo studies have their well-known limitations specific to wound biofilms as well. One way to overcome them, some studies, this is a collaboration we are developing with UK as well, where we are trying to use ex vivo human skin that is discarded from cosmetic surgeries, ex vivo porcine skin. Again, technically, this is challenging. This is a static system. It's not very high throughput, all right? And it does, static meaning it doesn't account for biophysical and mechanical forces that bathe the wound bed is exposed to. All right, so ex vivo also has limitations in procurement, execution, and high throughput, especially when you're thinking of screening all these molecules under host relevant conditions, right? Screening all these potential antibiofilm molecules under relevant conditions. Then we come to in vitro. Now, what's the problem with this? This can be done in the lab. They're pretty high throughput, but they're far too reductionist. One thing is, most often than not, they use just serum or plasma in a tube, maybe with some matrix here, as you see, to mimic the wound bed. or they use transwell systems, or they use these drip reactors where you grow biofilms, or then they fabricate microchannels with microfluidic systems. Huge challenge. I know when I came to India in 2018, I tried to do microfluidics for some of the work we were doing, and the dust just killed the project even before it could take off. We didn't have a sophisticated nanofabrication facility in the university, and that was that. So this is how people have tried to overcome ex vivo, in vivo limitations by doing all of this under in vitro systems. As I mentioned, they have various limitations, apart from the fact that they are far too simple. So they do not quite closely mimic clinically relevant conditions. So we see that there is a gap, and this is work that we did. We went to PubMed, we put in all these search terms, we found, is there a gap? This was one of the first projects we started in the group when I came back in 2018-19. I said, let's first convince ourselves there's a gap. So there is an increasing recognition that we need to study wounds because wounds get infected. Biofilms are well known now to be implicated in this, but there is very few new models being developed to study these chronic wound biofilms. Heavy reliance is on in vivo models, or then these in vitro reductionist models. So you can see this gap that exists here. This is the huge gap that exists basically. So we said, can we fill this gap? Since we focus on infection states, can we fill this gap by making in vitro approaches more human relevant? And we've approached this through different ways. Broadly, what all our work would lead to is first serve as alternatives to in vivo and ex vivo testing. They would be high throughput platforms that can be done in the lab and therefore can be used for preclinical screening. They can also be manipulated, enable selective and precise control. So we can push forward to personalized infection treatments, precision-based antimicrobial therapeutics. So let me take you on some of the work that we've done in the lab. So the first thing we did is we want to develop an in vitro device that can host, pun intended, host elements, microbes, matrix. So I told you my challenges with microfluidics. We said, all right, let's think big. Wounds are not micro, they are macro. So let's go big. We said, let's develop a macrofluidic device. And we came up with this design. Some undergrads worked on this. And we found that we could create a device of PCL, which is a biocompatible polymer that allows the growth of host and microbial elements. So we have a wound area. This was approximated from clinical studies that, an average wound area under different conditions. So we host a wound area right in the middle. You have inlet to flow in the cells, the matrix, the microbes, as well as fluid to support the growth of this co-culture. And we have an outlet which we can harness for downstream applications, chemical, biochemical assays, etc. So then we had to hook this up to a pump. Obviously, I said, one of the major issues with static systems is they don't allow you to apply shear stress and tension and compression. So we got another bunch of undergrads from the engineering department at the university and said, now design a peristaltic pump. This was way back in 2019. I was operating on a tight budget. The other 3-4 grants had not come through, and I saw a cost of peristaltic pump was 50,000 rupees, and I said, I don't want to spend this money. Can we do this somehow, some other way? And so we literally created a pump from scratch. They wrote the code, they developed the electronics for it. This pump is the size of a shoebox. So I said, this is the box. You can make it the size of a shoebox so it fits in a CO2 incubator. So the pump gets hooked up with silicone tubing to the inlet outlet gets hooked to some collection device, and the entire thing can fit in the CO2 incubator. All right, so we developed this. This is working. The next question is, what do we grow in this wound bed? And so we said, all right, so before that, we also said we have to grow something in the wound bed and we have to apply fluid that flows through and it maintains this co-culture once we establish it. And based on the dimensions of the tubing, the flow rate enabled by the pump, and the size of the wound area, we calculated to get a shear stress pretty close to what has been reported in vivo. So our device is able to recapitulate shear stress in the wound bed. All right, of course, this depends on the flow rate, the material that you're flowing through, and the wound area dimensions, which are now standard, which we are not going to change. Nevertheless, we can recap shear stress. So I would, take this to a place where I hope you're thinking of applying host cells and microbes, applying shear stress to them and seeing what the result is. Okay, so we said, fine, we've developed this. Now we need to grow cells in the wound bed. And as I said, the wound area is filled with two kinds of host cells, fibroblasts and keratinocytes. There is a challenge with growing these two cells together. So you can see here the spindle-shaped fibroblasts look beautiful and cobblestone-shaped keratinocytes. The challenge is, though they coexist in the human skin, that means you have fibroblasts in the dermis and keratinocytes in the epidermis, the upper layer, growing them together in the lab is actually a challenge. The reason is keratinocytes typically grow under serum-free conditions and fibroblasts require serum for growth. All right, as luck would have it, this was their incompatibility with regards to serum. So we looked at approaches that had been done to grow them together. What were they? So as you notice that when you're starting a young group, I'm trying to develop smaller projects that can get us publications and get the group morale high, and undergrads and master students working with us get something out of their time, like in the form of a proper publication. So we said, all right, what has been done? How have we grown? How have people grown this in the lab? And approaches included, first growing fibroblasts, then layering keratinocytes, often needing to irradiate fibroblasts to release factors to support the growth of keratinocytes. The problem with irradiation is you're killing the cells, so then they're no longer suitable for migration and proliferation assays. They're dead systems. So these feeder systems involve irradiation. So we found that either they were far too complex in the terms of this layering and irradiation, and they didn't allow live readouts of proliferation and migration, which is important for wound studies. So we came up with a protocol saying that let's look at two cells, okay, human dermal fibroblasts and HECAT, which is a keratinocyte cell line that happens to be immortalized but is well known to mimic primary keratinocytes. So we developed using standard media widely available. Any vendor can supply this to you. We developed protocols to co-culture them, which under simple, easy to obtain, easy to execute conditions. So what we did is we developed two media formulations. One contains fibroblast growth medium and keratinocyte growth medium in a 1:1 ratio. And the other contains minimal medium supplemented with fibroblast and keratinocyte growth factors. What this did is reduced the serum from 10% needed for fibroblasts, as I'm in the earlier slide, to 1 to 2%. This reduced it almost 5 to 10 times, reduced the requirement of serum. And at that low serum, keratinocytes, the HECAT keratinocyte cell line was able to proliferate with fibroblasts. And so we tried two approaches to mimic the wound bed, because you can see in the center of the wound, they are enmeshed, the two cell types, enmeshed in each other. But on the edges, you have keratinocytes overlaying fibroblasts. So to mimic these two conditions, we adopted a layering approach where we first added fibroblasts, then we added keratinocytes. And then you can see over 5 days, keratinocytes are forming beautiful cobblestone colonies in the fibroblast, between fibroblast sheets. The other approach, we said, let's put them both together. Can we co-seed them? So we co-seeded them. And again, they could grow beautifully. You see the keratinocytes colonies in the fibroblast sheets. So both are approaches which are easy to obtain, accessible, low cost, reduce the requirement of fetal serum in the system, were able to support the growth of these two important wound cell types under two different conditions, layering as well as co-seeding. All right, so we have developed our wound bed. And just to tell you that the cells were live, if you made a scratch with a pipette tip, they could migrate, they filled the gap under both conditions, under the growth media as well as under the growth supplements. We were able to fill the gap. So we had a wound bed that was living and could be used to study migration and closure. Okay, so you do realize that now we want to take this and co-culture it in that wound area, in that macrofluidic device. Okay, so we said we've got this sorted. We got one of our early publications, which was a great morale booster for the group. And then we said, all right, but what are we going to flow through this? We can flow through MEM and growth supplements, but that's not human relevant. That's not host relevant. And our whole focus is to build host relevant that I've been telling you since 11 o'clock and that you're patiently listening to. So, all right, so we said, then what are we going to flow through? And then I told Snehal and Vandana, who were the first two members of the group, I said, let's develop a wound milieu. We know through clinical reports what wound fluid composition is. We know this. Let us see if we can develop this in the lab so that we can kind of be that bridge between just using simple serum or plasma, or much worse, only LB and in vivo systems, right? We are bridging this gap again. So far, when people try to study these pathogens, they use LB or tryptic soy broth, the brain heart infusion broth, which are these protein-rich media. But the last time I checked, the wound was not bathed in LB. All right, the wound has wound fluid. So we looked at clinical reports. We said people have done this, so we don't need to do this. Let's look at what they did. And they listed the composition of wound fluid. So based on that, we determined that we can create a milieu in our lab. What would it consist of? 70% FBS would account for most of the factors in wound fluid. So we said, all right, let's just go with 70% FBS. What were the factors that were missing in FBS that should be present in wound fluid? Were these. One was matrix elements like collagen, fibrinogen, fibronectin. So we said, let's add these in concentrations that were found in the wound fluid. The second thing were important molecules, important biochemical factors like lactic acid and lactoferrin. So we said, let's add these. The rest of the factors were largely recapitulated in that 70% FBS. So based on known clinical wound fluid composition, we came up with this milieu. The first thing we said is, okay, we made the milieu, but does it even support the growth of these pathogens? We have no idea. So we said, let's start with simple growth curves. So we did planktonic growth of Pseudomonas and staph, and this is alone. And you see in LB, no Nobel Prize winning work, but in LB, Pseudomonas and staph are growing very well. So we were at, all right, our system is working. In serum alone, because we wanted to compare the wound milieu to serum alone, in serum alone, Pseudomonas appears to grow well, whereas staph appears less than LB, but it's growing. But staph appears to be retarded in growth. And if you notice in the wound milieu that we just created, and I told you the composition of, this difference in growth between Pseudomonas and staph is even more exaggerated. If you see, Pseudomonas grows well in wound milieu and staph is even more retarded. So growing alone, monospecies cultures gave us this inkling that Pseudomonas appears to have a growth advantage to staph in the wound milieu. But what would happen if we grew these two together? What would happen to this growth advantage? So this is staph growing alone in the wound milieu, Pseudomonas growing alone. This is in LB and FBS. This is when they grow together. If you notice, when grown together, the growth of staph appears to be inhibited. And how did we read this? We read this by colony counts in selective agar. So you find that Pseudomonas grows the same, but staph's growth appears to be inhibited. And you see this here as well. In the wound milieu, Pseudomonas is actually getting a bit of a growth advantage, as I talked about. It's growing slightly better, because if it was the same, it should have been 1. So it's growing slightly better, and staph is inhibited. So this importantly told us that even when grown together in the wound milieu, we are recapping in vivo conditions which show that Pseudomonas outcompete staph. This is well known in wounds, that even though they coexist, Pseudomonas outcompete staph. So we are recapping this in the wound milieu when they are grown together. What could this be because of? Well, it could be Pseudomonas' effect that it kills staph, that is well known. It could also be the effect of other factors in the milieu like lactoferrin and lactic acid, which are known to inhibit staph. So this is in planktonic state. But again, I sold so much of the story to you saying it's biofilms in wounds. So we said, can the milieu support the growth of biofilms? All right, so we set this up in 96 well plates, your standard XTT assay. You see Pseudomonas and staph in LB and FBS are fine. They also form biofilms in wound milieu. There is some inhibition when you compare expected to co-species, but the biofilms formed are metabolically active. So the second takeaway is that the wound milieu supports the growth of metabolically active, viable biofilms, which is in accordance to what you see in the host conditions, right? These are metabolically living biofilm aggregates. And it also recaps interspecies interactions, which are well known, where even under biofilm conditions, Pseudomonas kills staph, and this appears to be recapped here. Okay. The third important readout we wanted is what happens to antibiotic tolerance in the wound milieu? What happens to antibiotic tolerance? So you notice that for both Pseudomonas and staph, wound milieu shows increased tolerance to standard antibiotics, tobramycin and vancomycin respectively, compared with LB or FBS. Now, what happens for Pseudomonas in wound milieu versus FBS, you see pretty much the same. So I think to us, this means that serum, that 70% serum is playing a very big role in increasing the antibiotic tolerance of Pseudomonas. And for staph, you notice that it's much higher than what you see in FBS, which points to the role of all those matrix factors possibly in increasing the tolerance of Staph aureus to vancomycin. You also notice it's very high in LB, and the only explanation we have for this is that it forms very thick biofilms in LB. It's known to, and that we see based on our metabolic activity and further our confocal studies. All right, so we are recapping the increased antibiotic tolerance in the in vitro wound milieu for both pathogens against two commonly used antibiotics. So this is a good sign because we know that in the wound state, these biofilms are tolerant to antibiotic treatments. Okay, so the next exciting thing was to actually see this under the microscope. So when we looked at biofilms grown in 96 well plates in situ, that we just grew them, pushed them under the confocal, and we used a tile scan approach where it scans the entire biomass. No washing done before putting it under the microscope because any washing is going to disrupt the structure of the biofilm. Just push it under the microscope. You see in LB, staph forms good biofilms, in FBS, in wound milieu, but wound milieu is thinner than LB and FBS. This is important because it may mean that when we are testing all these antimicrobials that I talked about, these various lead compounds, in LB, we are overestimating the biofilm. So it means we are probably giving the antimicrobial a very harsh situation to work against, stacking the odds against that antimicrobial. So the wound milieu actually shows nearly 50% thinner biofilms compared to LB. Now, importantly, these plates were surface treated so that we parse out the role of the milieu and reduce the role of surface attachment because, well, again, the last time we checked, the wounds didn't have polystyrene in them. So we wanted to reduce the role of surface attachment and bring out the role of the milieu. What that did, and this was surprising to us, we never expected this, what that did is Pseudomonas didn't form very good biofilms in LB. The only explanation we have is in the absence of surface attachment, in the absence of host proteins, the proteins present in LB, which is these refined soy protein or yeast extract, is not sufficient to support biofilm formation. The only reason it was working in the earlier assays because the surface attachment was still there. Whereas with FBS and wound milieu, you have a lot of host proteins like collagen, fibrinogen, fibronectin, albumin in FBS that would support the growth. So the role of the host milieu became more important in Pseudomonas biofilms, and you could see these dense red maps. What was more interesting for us at this stage was once we got this to work, which was several months of work, what was more interesting, Snehal and Vandana would like me to tell you that every tile scan takes 40 minutes. And so you're pretty much waiting for it to work till it all stitches together and tells you whether it's worked or not. So now once they said this, oh, this has worked. What do you think I told them? I said, all right, next step, let's grow them both together. And they were like, oh, we knew if we went to her, we were going to get more work. So the next step was to grow both the pathogens together under the three conditions, LB, FBS, and wound milieu. And all right, Pseudomonas was known to form poor biofilms in LB. This was the earlier slide. It continued to do so, but even the staph was not seen. Why? We think in Pseudomonas killed staph. And in LB, this effect is well known, well documented. This effect appears to be rescued in the presence of serum. And albumin in serum is well-published work shown to rescue this killing effect of Pseudomonas on staph. So we can see a nicely structured biofilm, green staph and red Pseudomonas, and you see some spatial organization, which is interesting, which we haven't looked into, but you always see Pseudomonas on top and staph at the bottom. And these were inoculated together and mixed. So why this spatial segregation is happening, we need to look into it. But FBS rescues the killing that you see in LB, and you do get a stable coexistent biofilm. And what happens when you add factors in wound milieu, like especially lactic acid and lactoferrin? Again, you see an out competition of Pseudomonas. You do see staph. You do see staph if you look at it very closely in the inset images, but much less than FBS. And you see Pseudomonas outcompeting staph. So there's a predominance of Pseudomonas in the co-species biofilm, which we again recapitulate something that you see in the wound milieu where they coexist, but Pseudomonas predominates. So what are the next steps for us? What we are working on right now, we have some revisions for this paper to get done. But subsequent to that, what we are working on is trying to integrate this all in our, where we started, what was funded by Ramalinga Swamy, the macrofluidic wound device. So you have the device which will grow the wound cells, the wound bed, which we optimized under those reduced serum conditions. The pump will flow the wound milieu, not MEM, because that's not in the wound. It will flow our human relevant wound milieu, and we'll be able to collect downstream material for chemical and molecular analysis. We will also be able to infect, the next step actually is to infect this wound bed with Pseudomonas and staph to get host microbe co-culture under shear stress in the presence of wound milieu in the presence of both host cells. And we've already started this, and you can see Pseudomonas studying the surface of the fibroblast, but we need to look at this in more detail. So this will finally be our wound biofilm on a chip. It would not only, well as the wound milieu alone can serve as a high throughput preclinical platform, even this could serve as a screening platform and also allow us to study host pathogen interactions in ways that have not been studied before, thereby integrating in vivo, in vitro, in the lab. All right, those are the next steps. So since 2018, many young people have come into our lab. Vandana Snehal took a lead on several of these projects, and we have undergrads, including visiting students who've taken it forward. Right now, the lab is being held by Radhika and Nizam. They are keeping it going. Post Ramalingaswamy, we got funding from IYBA for another project, which I haven't talked about here. And I have an International Exchange and India Alliance International grant. So I can only say thank you for this opportunity, and happy to take questions, suggestions, and look forward to more engagement with all of you. Thank you.
Thank you, ma'am, for that wonderful talk and truly interdisciplinary. I think you have definitely given, in a subtle way, informed all the engineering students in Shastra that having learned mathematics, physics, along with biotechnology subjects is equally important.
Yes, certainly. In fact, Aniket, who made the pump, is a biotechnology student at IBB. But because he did engineering, because he did physics, he was able to execute, because he could write code, he could execute the creation of the pump. So that's a wonderful point, actually, Bhuvana. Thank you.
Yeah, truly interdisciplinary, because today's research has become more interdisciplinary, and that's lovely to see that. There's a couple of questions that's in the question panel. I think from Regina Huang, it says, in the presence of the wound, there are different cytokines in the wound fluids. Have you considered adding different cytokines in the culture medium to observe migration and proliferation?
It's a very, very good point. Cytokines, matrix metalloproteinases, we have not added them. I mean, to be very frank, I'll tell you, they are far too expensive. If you are needing to flow, and we need to flow it in ml, that's a volume. They are far too expensive to add, but maybe in a limited study, we could add them in. I think this is a good point. We will look into that. Cytokines, like I said, even MMPs, we have not added in, but they are both critical to wound outcome. We have not added them. Yes.
Okay, so there's another question from VK. Since hypoxia plays a vital role in wound healing, especially in case of DFU, does these wound bed have hypoxic condition too?
Excellent question. We don't know, VK, but what we are going to do as a next step, and I don't know if you were the reviewer for our manuscript, but this suggestion was given by the reviewer for our bio archive, is we will start with the wound milieu. We have never looked at the wound milieu under hypoxic conditions. So we are going to start with that and then see what we can do in the wound device, the wound bed. We will start with looking at the growth of the two pathogens in the wound milieu under hypoxic conditions. And this was a suggestion given by the reviewer. Because the milieu is very, it recapitulates the chemical composition of the wound fluid. It would be interesting to look at it from the from under hypoxic anaerobic microaerophilic conditions. This is something we are going to do as a next step. But in the device per se, actually, I don't know what the oxygenation is. We haven't measured it. Something we need to see.
Okay, that's the next question is on to do more with, I think, immunocompromised conditions. So the question is, some microorganisms do cause infection in some individuals, but not for some. And diseases like aggressive periodontitis, little biofilms are seen, but still the damage is heavy. Inflammatory response are much more to infections and causing self-damage to tissues. Can it be classified as autoimmune or immunocompromised disease?
So I think diabetic wounds are immunocompromised states. Yes. There is clearly, it's well known that there is neutrophil malfunction, poor functioning. So yes, I mean, diabetic wounds are certainly immunocompromised states.
Okay, I don't see any more new questions, so I take the opportunity to ask you one question.
Please.
two, if you don't mind, because we do have some time.
Yes.
My first question is, it's very nice that you have mimicked your wound milieu,
Yes.
but I assume this is for like in the case of a normal person wound milieu.
Yeah.
How would it be? I know you'll be considering this for looking at immunocompromised wound milieu, right? Because the biofilms are more in immunocompromised people than in
Yes,
normal.
non-healing wounds is more in immunocompromised people, like diabetics, obesity. Yes, yes.
Okay. In the meantime, we got another question from Arwal. There is a concept of growing polymicrobial systems directly in vitro without isolation in simulated media. Would a similar experiment design benefit the unique macrofluidic device and IVWM that has been developed by your lab? What would be the challenges that would be expected in case of, or would in polymicrobial systems like these?
So what you're trying to say, Aral, if I'm getting you right, is like you take it from a clinical swab. Just take the sample from the swab, directly polymicrobial without isolating individual colonies. What are you trying to say? I'm not sure.
Arwal, you could directly ask that question.
Yeah, yeah. Yes, ma'am, that is right. That's what she means. So, see, well, I don't know. It will be too uncharacterized. There will be fungi as well growing. And what we can do next step is incorporate fungi, which again the reviewer recommended. We can also incorporate commensal microbes, which are known to exist in the wound biofilm as well. But directly from a wound into the device, it would be far too challenging because we are currently having challenges with growing 2 host cells and 2 microbes. If we add fungi and even more bacteria, I don't think the stable co-culture will be obtained at all. It will probably just kill off all the host cells. That is the challenge with that. But step by step, we want to definitely get Pseudomonas and staph stable, and then maybe add a fungal element and maybe add the commensal bacteria.
Okay. Due to this high relevance to the QSL group, I'm actually would like to invite a question from panelists, maybe Princy ma'am or Dr. Prasanna himself.
Anytime, anytime. I'm getting nervous now.
Thank you for the fantastic lecture, Dr. Karishma.
Thank you, Dr. Prasanna.
It was very informative, and I think something which is very, very timely and required for us to translate well plates to something which is more biologically relevant. And I totally understand the rationale behind which Ardwell raised that question, but that probably needs a bit more of a detailed discussion and cannot be done shortly. But we will get back to you on that. Because
sure.
there's any possibility to grow such polymicrobial systems under host relevant conditions, and something which is grossly lacking in oral microbiome research.
All right, okay.
That's very, very important to look at.
And Dr. Prasanna, we do have another project on oral biofilms in the context of tobacco use, and we can even get in touch with that. I mean, I could not talk about everything, so I said, but we will get in touch with that as well.
I think sometimes even scratching the surface is a lot more enough to stimulate minds rather than
Right. All right. At least leave people to go back thinking that we can't study pathogens without a host. We need to find ways to include those elements.
Sure.
Yeah. Thank you.
Thank you.
Okay. May I ask one question?
Yes, please, Bhuvana. You started asking earlier.
Yeah. No, the only thing is I'm just looking at 3D printing technology. Would that help in mimicking better wound designs?
Oh, yeah. So this device is 3D printed, Bhuvana.
Oh, okay. Yeah.
Yes,
okay.
it is 3D printed. That polycaprolactone. Yeah, it's very cheap. It costs us 1,000 rupees. It gets printed in 20 minutes. We have the design in AutoCAD.
Yes.
So we've moved from microfabrication because of the lack of facilities, and said a 3D printed reusable device would be the way to go. So we can modify this as per our requirements.
Okay.
Subsequently, like, look, I only focused on the upper compartment because even that is proving to be a challenge. If we want to involve the immune compartment, we can 3D print it into a 2-compartment system as well.
Okay.
Of course, the challenges of maintaining 7-8 types of cells in co-culture exist, but that's the way we want to go.
Okay, that's, I think, thank you for the extremely wonderful talk. And I do see that not only to study disease, I mean, your wound infection and healing, but I actually think there would be a very good market, or maybe other collaborators who will be looking at studying, antimicrobial, they may be having certain antimicrobial systems or drugs developed. And this wound healing, wound on a chip,
Right.
would be a very a good path forward for them to put it
Yes,
in.
most certainly. Most certainly. That is what we also want to see, how the biofilms grow in the on the host cells. This has never been shown before. They are
Yes.
probably not going to be mats like we see them in the lab. They're probably going to be clumps at
Yeah.
the junctions of the cells. Let us see. Let us see. It's an exciting adventure.
Yes, thank you so much
Yeah.
for the wonderful talk. I definitely agree that the students would be inspired by your talk. And I over to Shastra for the next sessions.
We have 10 more minutes, but we have one more question from Arvel. Dr.
Oh.
Bhuvana, if you can.
Oh, yes, sorry. Another question, Dr. Karishma. It's, when designing 3D printed platforms for lab research, how do you ensure biocompatibility? Various organisms have shown different adhesive properties based on material. Also, is there any problems with sterilization as 3D printed materials are heat sensitive?
I'm so sorry, I'm muted. I just said wonderful, Arval. Arval is putting me to the test. Absolutely fantastic. So first question, Arval, only PCL was compatible, polycaprolactone. So we use that. That allows cells to grow. Cells are growing on that. We know that. The earlier one we did for PCL is expensive. The earlier one we did PLA is cheap, but not biocompatible. So we optimized with PLA, but now once optimized, we are printing everything in PCL. Now about the sterilization, yes, we can't autoclave it. We cannot autoclave it. So what we are doing, this is a challenge. And what you have asked is right up Nizam's heart, because he deals with this every day. So what we do is take the device after it's used, overnight incubate in 70% ethanol, dry it out, UV expose, physically clean the channels and grooves, right? And then again, keep it exposed to UV. We have been having contamination issues. We need to get this better done. But we can't put it, it's not as easy as sticking it in an autoclave. You're right. It's ethanol and UV sterilization that we are doing. Yes, this is a challenge. This is a challenge.
Okay. So I've been asked to engage all of us for another 5 more minutes.
Yes.
So Dr. Karishma, let
Yes.
me take you from the technical session to a more non-technical question,
Yes.
which is, recently we finished, International Women's Day, and there's been a lot of talk in women in science, women in STEM. So what would be your advice for, I personally think it's not just advice for young women, but it's just advice for both young women and men, because I don't see bias from my end.
Yes.
So what would your advice be for the young generation in this area, for young women in science and overall STEM?
So I'm no advice is a very strong word. I will just say my suggestions from what I have learned.
Yes.
Bhuvana, I think one thing is we should all we should take our so one thing is developing good science. We should be very confident about the science that we do, right? We should genuinely enjoy it, and know more about it, because the best thing, the strongest thing you bring into a room is your science. But after that, I also think, especially for young women, and especially for young South Asian women who are typically taught as we grow up to not be very visible, it's hard for us to be on social media and other platforms because we've grown up with a lot of conditioning of not being very vocal, not being very visible. I think today, networking, building connections, collaborations, being out there is very important in career building. So it's along with the science that you pursue, is very important to know people, that you are to tell people, for people to know that you're doing this and that you're serious about this, and be proactive in engaging with the community, social media, whatever tool you use. I can absolutely vouch for the fact that our India Biofilm Society was hugely fueled by connections on Twitter.
Mhm.
The collaborative grant we got with Joey Shepherd in Sheffield through Royal Society was through Twitter. It was on Twitter I saw a post that she works on a similar wound model and we collaborated. So it's no longer enough to just be doing science and doing it in an ivory tower. One has to be a little out there, for want of a better word. And this is particularly hard because we've grown up saying don't put too many photos on social media. It's a conditioning that we've all grown up with. So as safe as you want, as much as you want to share, but some presence is important for career building, for professional growth. So maybe that would be my take to young women in particular and young people in general.
Yeah, that's a very, very good advice, I would say. And yes, to all the young people out there, this is Dr. Karishma Kaushik for you, who has seen different spans of areas, right from medical, then to molecular genetics, and then she's even seen the building of a pump. I mean, that's really inspiring.
Yeah, thank you so much, Bhavana.
She's seen a full circle of research and truly interdisciplinary research. And coming even from my personal experience, I would say that that's really the research of the future, where you have to be interdisciplinary. Like, gone are the days where you just say, I do physics, my area is
exactly. Very
those
true.
days are.
Very, very true. And I mean, more exciting and very exciting for me has been the fact that I could return to India and do all of this. That has really been the icing on the cake.
Yeah, that's truly inspiring too, because I do see that extremely inspiring.
It was a very busy personal and professional phase to move back to India and start a research group and apply for grants and manage the science at the same time.
Yeah.
But 3 years down the line, it's been extremely rewarding. I will definitely say that. And we can't compare apples to apples. Ecosystems the world over are different. There are different gains in doing it in your own country.
Yeah.
So I'm excited about that. Yeah.
Yes. I look forward to connecting you and getting more advice from you on that.
Thank you.
Maybe on another platform. So,
certainly.
Yeah. I think, thank you so much. I think I'll stop talking and I'll pass it on to Shastra for the next one.
Thank you. Thank you.
Yeah, thank you so much, Dr. Bhavana. And thank you, Dr. Karishma. It's always a pleasure having you. And as per your philosophy, you have finished right on time, even before that. So thank you so much for that. So next up, we have Dr. Prasanna to introduce Professor Purnima Kumar.
Thanks, Sahana. We may have to wait a few minutes before Professor Purnima joins.
Sure, sir. Sure.
Because she has been stuck in a personal emergency. So
Sure.
just give us a few minutes and then we should be back. Yeah.
Sure, sir. Sure. So in the meantime, the attendees can post their questions to either Dr. Karishma or Dr. Praveen. They're available for taking up questions.
Sahana, just wanted to check, are panelists able to type in the Q&A?
No, that option is not coming here. So you can use the chat box.
I'm not able to ask some questions too. I realize we don't have that option.
Yeah.
That's okay.
Yeah. You can ask now, Dr. Bhavana. If you have questions to Dr. Praveen.
If Dr. Praveen is available, I'd just like to ask one question, maybe to fill up some time. My question was actually on the hydrogels. Are they chemically synthesized hydrogels, or are you also looking at peptide, self-assembled peptide-based hydrogels as well?
These are not peptide-based. These are small amphiphile. They're synthetically made ones. So, in fact, like even peptides, also people now every peptide sequence one can synthesize. So that's not an issue. But these are chemically designed small molecule synthetic molecules. Right?
Okay. Yeah, that was my. Thank you.
Thank you.
Okay, I see Professor Purnima Kumar is here. Okay. All right. So the next speaker for the day is a true rock star in the field of oral microbiome research. We are extremely delighted to have Professor Purnima Kumar with us today. Professor Purnima Kumar received her dental training in Annamala University in India and her specialty training and her PhD degree from the Ohio State. Now a professor at the Ohio State University, Professor Kumar is one of the most sought after speakers on her cutting edge research on the critical intersections between oral microbiome and disease. With more than 100 high impact papers, including publications in Science, Professor Purnima Kumar runs a robust research program funded by the NIH and corporate partners. Last year, she awestruck all of us by systematically demonstrating her robust findings that e-cigarettes are more dangerous to the microbiome than usual cigarettes. Professor Purnima Kumar, all our attention now belongs to you. Thank you so much.
Thank you, everyone. I am sitting in a hospital, so the internet will cut in and out. So I apologize in advance for whatever it is, that's happening here. But let's get started without much ado. It would be this one. Let's see. Oh, okay. So are you able to see my screen? Yes, good. All right. So today I really want to introduce the concept, and I'm sorry, every phone around me is going to keep dinging because I'm expecting calls on different ends. So if you hear all of these dings and bells and whistles, it's entirely my fault, and it's intentional. So today I'm hoping to bring to you an understanding of why we host bacteria in our mouths or elsewhere in our body, and how these bacterial systems actually they're not just passive travelers in our body. They're actually by design. They're a beautiful design community, and this design, when this design is perturbed, then you result you end up in a disease state. And there is no one single disease state. This disease state can take multiple forms, like Medusa said, depending on what stage and what age of the disease you're in. Not human age, but disease age. So there are multiple factors and multiple things. But I'm really hoping that I can leave you with an understanding that there are many multiple factors that control what lives in your mouth and why we host these communities. So if I could leave you with 4 take-home points, the first take-home point with that, we have an ecosystem in the mouth. It is not simply bacteria. It's not something that you brush off in your mouth. It's an ecosystem, and it's evolutionarily conserved. There's a whole evolutionary pattern or even a biological blueprint that is associated with this. And that this biological blueprint is determined by you, your genotype, who you are, an Indian female, in the second decade of life, the second half of life, whatever, those kinds of things, right? So that becomes important. Not just that, but that this woman is living in the United States. She is a non-smoker. She is a non-alcoholic. She is a vegetarian. So these things kind of determine what colonized me and how stably they colonized me. The other thing I would like for all of you to take away is that there are the oral microbiome responds very specifically to these genotype environment interactions. And by understanding what these interactions are and what the response to these interactions are, we can probably there's room to create salivary diagnostics or any kind of diagnostics. And more importantly, you can assess risk for disease based on these fingerprints, as we call them, these genome or microbial fingerprints that are created by these genotype environment interactions. So I'd like to introduce you to the research group I work with. There's a very extensive network, another ecosystem. And I like to borrow the African word Ubuntu. Ubuntu means I am because we are. And what I'm sitting here is the face of about 50 or 60 people I work with very closely, people who are far more intelligent than me, who are smarter than me, who are more driven than me, and who are more curious than me, and more invested in saving human lives or improving human life through science. So this is my group. My research group, as you can see, is here. These are my students. Many of them are dual degree. They're getting both their dental training and their PhD. Some of them are just getting their dental training, but while they're in dental school, they're also doing research. Some of them are only getting their PhD. Some of them are doing their clinical specialty training. It's called a residency in the United States. So doing their clinical residency and getting a PhD. And some of them, like Mabel here, like Nate here, like Khalid here, and Michelle here, and Najla here, they're doing what's known as a sandwich PhD. They're doing 2 years of their PhD in their home country, 3 years with me, and 2 years back in their home country. So they're doing what's known as a sandwich PhD. And these international collaborations have truly been the cornerstone of what we have been able to achieve and how quickly we've been able to move the science forward. Along with them, I also have other collaborators. We work on what we call the her biome, which is the female microbiome. And I interact with many people in the medical field to do that. Dr. Lucas is an infectious disease specialist. Dr. Ching is a urologist. And Dr. Cheryl Justice is a PhD who works on E. coli infections. I work with two bariatric surgeons, Drs. Needleman and Dr. Noria. And I work with a diabetologist and endocrinologist, Dr. O'Donnell, in understanding how obesity and diabetes can impact the oral microbiome and how the oral microbiome can impact these two as well. I also work with a lawyer because of tobacco and e-cigarettes and the legal ramifications involved with this. And because we do a lot of population studies, I work with a biostatistician who's also my mentor. Dr. Nagaraja has done a lot of work with me, and he's mentored me through a lot of science. The last team I would like to introduce you to is this bioremediation team. We not only study disease, but also how to mitigate disease and how to ameliorate the symptoms of disease. And to do that, we study what's known as bioremediation, which is we study black raspberry extracts and how these black raspberry extracts can help to modulate the oral microbiome. And to do this, I work with food scientists and oncologists in an effort to study that. I'm going to start about talking risk. When we talk about risk for disease is a 4-letter word. It determines treatment algorithms. A patient high risk will have a completely different treatment than a patient who's low risk who will have a completely different treatment algorithm. In our country, it determines insurance payouts. People with high risk, the pre-existing conditions, right? People who are high risk have a different insurance payout when compared to people who are low risk. It determines public health policy. Right? And that is very, very important. And risk has 2 forms, primary and secondary. So what is risk? When you think about risk, I'm just going to take a very simple example. It is a chance or a factor that can lead to a negative outcome, which means if you're texting and driving, what is the risk of you getting into a traffic accident? So let us see how risk is assessed before we actually get into the whole system of why I'm talking about this today. Risk is assessed based on the facts we know, right? So this is the fatality analysis reporting system in 2010. There's a website. So I went into that and it gives you certain data. It says, what are the odds of you getting into a traffic accident when you're texting and driving? It depends on the frequency of the text. Each time you text, it increases your chance by 2.3%. It's also driven by the duration of the text. Each second of texting increases your chance by 0.9%. It increases by the type of a text, whether you're sending an image, or you're sending a text, or you're receiving an image. It also depends on the location where you're driving. It is a highway or a side street or a country. But notice that there are no numbers here, right? So some variables that drive risk are categorical. Some variables that drive risk can be assigned a numerical risk class. That is really what I'm trying to tell you here, right? So if you take all of these factors, similar to this, in periodontal gum disease, when you think of gum disease or caries, dental caries, cavities, any of these things, what you can find is there are specific metrics that data input that go to assess risk. And when we think about this, there are several different factors. In periodontal disease, gum disease, right? There are 5 different factors. So the 5 different factors are bleeding on probing, probe depths, tooth loss, whatever it is. There are 5 different factors that are used. And based on that, you can have a patient who is high risk for gum disease or low risk for gum disease. There are other methods also, different brands. Like, you know, as soon as something gets, it will become commoditized, and once it's commoditized, there's branding, there's tweaking of those algorithms. There are all of these different risk factors that are available to us. So, right, people said, okay, are these factors able to assess risk efficiently? And so this provisor system that I just showed you, they did a study where they looked at people who were low risk, that is risk score of 2, risk score of 3, which is moderate risk, 4 and 5, which are high risk. And they looked at their bone loss around their teeth, the bone loss around the teeth, right? So that the tooth gets loose or not. They looked at the bone loss around their teeth at 3 years, 9 years, and 15 years. And they found that people who were assessed as high risk at baseline actually did have a lot more bone loss at 15 years than who were assessed at low risk at baseline. Great. And then they looked at 2 different risk assessment systems, the provisor and the UniFi that I just showed you. And they said there's a very good agreement between these two risk assessment systems. So no matter what system you use, it's doing a very good job. So then you're like, okay, so what's the problem? Why are we sitting here having this discussion? Because they did one more study, which is very meaningful. What they did was they took 100 patients, right? No, sorry, they took 15 patients, 3 in each group. And I'll walk you, this last chart, I'll walk you through it in a second. They took 15 patients, 3 in each risk group 1, 2, 3, 4, and 5. They took 15 patients and they gave it to 100 dentists. And they said, okay, assess the risk for this patient, blinded, right? So they asked the people to assess. So when you're looking at this chart, here is the risk score, 1, 2, 3, 4, and 5. And here is the real risk score assessed, right? So the blue white diamonds, can you see that here? Here are the white diamonds for 1, white diamonds for 2, white diamonds for 3, white for 4, and white for 5. So this is the true risk. But then what they found was when 100 dentists assessed their risk, when the risk was low, people overestimated risk. The majority of clinicians overestimated risk. But when the risk was high, they underestimated risk using this provisor system. So then you might ask, what's wrong? Why are people doing this? You have a risk assessment system. Why can't you do it efficiently? Because if you take the same story and put it back into the fatality analysis system that I showed you, and take an example, I'm going to say, this is Purnima. I'm texting and driving. This is my second text today. I've been texting for 10 seconds and I'm driving on I-95, which is a highway in the United States. Each text is supposed to, according to the fatality analysis system, each text is supposed to increase my chance by 2.3%. So my risk goes up by 4.6. Each second increases by 0.9. So my risk, I've been texting for 10 seconds, goes up by 9. Sending one text is 1.2. I'm driving on a highway, but you can't put a number on it. So let's let it go. So essentially my risk according to the calculator is 14.8%. So 1 in 6 times I'm supposed to get into an accident. But when I put this data into the fatality analysis system, it tells me my risk is 47%. Where is 14.8 and where is 47? So then that system will tell you the reason why your risk estimation is completely different from our risk estimation is because we have not taken into consideration the speed at which you were driving, other distractions, whether you were putting on makeup or eating or drinking a juice, the make of your vehicle, whether you're driving a Tesla or electric car or whatever, your driving conditions for snow, smog, fog, rain, whatever. What is your gender? Are you woman, male? Females are supposed to be more risk averse than men, and many factors that we don't yet know. So this is a system that has thousands and thousands of data points by everybody driving all over the country in one of the largest driving countries in the world. And still there are so many unknown holes in the system. When you take that example and you convert it to the microbiome, or you convert it to the oral cavity and oral health, what are we doing? We are taking 5 factors, we are putting in 5 factors, and we're asking to estimate risk. So of course, people are not going to be able to estimate it well, right? You need a lot of factors. Really, there is more. Yes, I am a microbiologist, I'm a microbial ecologist, and we believe that when you look in the mirror every day, you see a person who is 43% human. only 43% human. You're saying, hey, where did you get that number from? This comes from a Nature study where they showed that we're made up of 30 trillion human cells and 39 trillion microbial cells, which makes us 43% human. You can say, okay, let's look at genes. Forget cells. Who cares about cells? There are 20,000 human genes and 2 to 20 million microbial genes, which makes you genetically 1% human. So if you are only looking at human genes or human factors to assess risk or predict anything, you are neglecting 99% of your genome content or 57% of your cellular content, the information, right? Your cellular informatics or 99% of your genomic informatics in your ability to make decisions. And so we believe that the way forward is harnessing the microbiome to be able to estimate risk. The way we do this is, I am a clinician, I'm a surgeon. And so I believe that the best experimental animal is the human being. I don't do animal studies. I don't do rat, mouse, dog models. I don't do those. I study human beings because I like my research to be directly translatable to humans. And studying a human gives me the breadth of the variability that exists in human studies. I sequence most of my work is done on sequencing. So we collect samples from these humans, oral samples from these humans. We sequence their DNA and sometimes their RNA and sometimes their protein. And based on the type of genetic material we're sequencing, we compare it to specific databases and we develop a microbial fingerprint for each subject, right? So I'm going to run this. So what you're going to see today is based on a very global, big picture view. I will not talk to you about specific species. I will not talk to you about specific genes. I'm going to give you a big umbrella, the view from Mount Everest kind of a picture about what's going on. And so the way we do this is we take all of this multidimensional data and we compress that into 1, 2, or maybe 3 dimensions, 3 axes, right? X, Y, and Z axis. We compress that data, and based on that data compression, I don't want to say data compression, it's actually dimension reduction. By reducing the dimensions of our data, we can plot each subject on a map, on a 3-dimensional map here. So these dots represent subjects, and the distance between the dots represents the similarity in their microbial communities. It can be their type of species, similarity in the types of species, similarity in the genomic content, in their gene expression, in their proteomes, whatever you want to talk about. But it represents some distance between the similarity between 2 subjects, 2 individuals based on some metric that we're measuring. So then we ask, are the differences between 2 individuals greater than or less than the differences between 2 groups of individuals? So we are asking if the between subject similarity is greater or less than the between group similarity. And based on those distances between groups, we play a game known as rock, paper, scissors, right? Have you played that? Do you know what I'm talking about? Rock, paper, scissors. So scissors can beat paper because scissors can cut paper, but rock can crush a scissor and can crush a paper. So rock can beat both scissors and paper, but paper can cover rock. So paper beats rock. So this is the game, right? What wins in this situation? So we're going to play rock, paper, scissors today. Before we go there, we're going to talk about why. What is beneficial to us to host these systems? So to do that study, this is what we've done. We sequenced both the RNA, I'm going to go back. So we sequenced the RNA and the DNA of a microbiome of healthy people, clinically healthy people, oral samples. And we took those sequences, the data from those sequences, and we mapped it onto this template that you're seeing in the background. This template that looks like a road map is actually a metabolic pathway map. It's known as the KEC map, and it tells you the different metabolic pathways that create a lifestyle for a community or a species, right? So this is basically a lifestyle map, if you will. And we mapped all the genes that contribute to a lifestyle of a community when you're healthy. And the lifestyle of this community is that this community does smart work. So it uses aerobic respiration. This here, what you're seeing here in green, the circle here is the tricarboxylic acid cycle. So this is the TCA cycle. You have the green, and it uses oxidative phosphorylation to metabolize simple proteins and carbohydrates. From there, because it uses such a simple process and it's highly energy efficient and it's doing smart work, there are very few byproducts that are created. Excuse me for a second. I'm going to pause myself. Excuse me for a second. This is the hospital calling. Sorry, false alarm. They called me for the wrong patient. So because it does this kind of smart work, there are very, very few byproducts that are created. And body hates byproducts, right? You hate many of the byproducts that are produced, which are very antigenic. And the kind of species that do this kind of smart work and live this very Zen, very Buddha kind of a lifestyle are the ones that also produce very few antigenic products like endotoxins, cell walls, glycans, peptidoglycans. They are low immunogenicity. So it costs us nothing to host this microbial ecosystem. Really, our cost to hosting the system is very low. The other reason we host this microbial ecosystem is because we're very familiar with it. What do I mean by that? You're looking at a group of species here. Don't try to read the names of these species. They're meaningless to you. What we're seeing here is a group of species that were found in babies less than 1 month of age. And then here's the next group of species that were found in these babies when they got their first baby tooth, that is 6 to 9 months of age. This is when they are 9 to 10, 11 years, no, 7 to 10 years of age. So they're losing their baby teeth and they're getting new fresh set of teeth. And this is when they are 15, 16, 17 years of age, when they only have adult teeth. So what you can see here is the group of species that they acquire when they are a baby, right? Within 1 month of age, 85% of these species persist all the way to adulthood. Even, what they have before they get teeth stays through 2 changes of teeth. The next set of species here are the ones that need a tooth surface to colonize. You'll see strep mutans here, you'll see Porphyromonas gingivalis, all of those species that need a tooth surface to colonize. And 45% of those species are colonized continue to adulthood, right? So about half of them. So when you think about it, the adult oral microbiome is established before your first birthday. Before your first birthday. Why is that important? Because it takes your immune system 2 years to mature. Before your immune system is mature and active and ready, these bacteria have colonized, they have been training your immune system. And so this is a very familiar system. And this familiarity allows you to drink water safely without changing your microbiome. Otherwise, think about it. Every time you eat, every time you drink water, every time you move from USA to India and you eat chaat masala, your microbiome is going to change. Your oral microbiome is going to change, which is terrible. Think about getting oral ulcers every 10 days. Think about, getting sickness. Think about getting all kinds of diseases by microbial shifts just because you're doing geographic shifts or you're drinking or eating. So what I want to explain is that this system in the mouth is a very stable, very, I like to say this is like, the Puri Jagannath Rath. It doesn't move, it is there. It is forever there. It is very difficult to move that Rath. Rath Yatra takes too many people to move it. Just like that, it's a juggernaut. It doesn't change very easily. And the change doesn't happen because the species that are established are the ones that were the ones that train the immune system to accept them. So that level of immune tolerance is what allows these species to stay in your mouth. So that becomes very important, right? I put a box here. I'll explain to you in a second why this box is important, okay? Third reason why we have these species is because these species are very, very species specific, are very, very site specific. So when you look at this is samples from women. We've taken samples from women 0 days, literally 1 day old women, 1 day old girl children to all the way to 18-year-old girl children. So the entire spectrum, right? And we find that no matter what the distance between the vagina and the rectum is, whether it is 3 millimeters or whether it is 6 centimeters, the vagina and the rectal microbiomes are very distinct, very distinct. And even though the rectum and the oral cavity are two ends of the same GI tract, those microbiomes are very distinct either. But these three mucosal microbiomes separate out from the skin microbiome for each person. So there's a very specific habitat specificity to that, right? Like all ecosystems, this is very habitat specific. We've seen this with dental implants. This is a map, same thing, the principal component analysis map of healthy teeth, microbiome of teeth with periodontitis, microbiome of healthy implants in blue, microbiome of implants with disease in red. You can see that the difference between implants and health and disease is much less than the difference between any implant and a tooth. Now, if I take these teeth out of this map and only map the implants, those two will separate out. So healthy implants and disease implants are very different microbiomes. But when you put the tooth into that mixture, the difference between the implants gets so much minimized that the tooth becomes the biggest driver. So these are very specific. We also did another study, what we call the hip-lip study, where we studied implants from the hip and the mouth of the same person. So 2 titanium implants, 2 different locations, and we found that it doesn't matter. Those implants are still different microbiomes. So it's not material specific, it's habitat specific. So then what makes you, you? Why is Purnima's microbiome specific and not the same as Prasanna's microbiome, right? So that's a very important question, microbially speaking. So this is like, fingerprint, microbial fingerprint. The first thing we looked at is genetics. So in Ohio, we have a city called Twinsburg, where every year in the first weekend in August, we have Twins Days Festival, which means that 9,000 pairs of twins show up there. We've gone there for 5 years. We've collected multiple samples from them. And what we did was, the hypothesis here is, if you have twins, if you're monozygotic twins, identical twins, then you're genetically identical. And therefore, your microbiomes must be more similar than twins who are not genetically identical, right? If genetics does contribute to shaping your microbiome. So this is a similarity index you're looking at. We found that, yes, monozygotic twins are much more similar than dizygotic twins before their teeth erupt and when they get their baby teeth. But that difference vanishes once the next set of teeth starts coming in and lifestyle changes take over and when they get their adult teeth. So genetics is very important in early bacterial colonization, but you cannot see that if you're only sampling adults, right? That gender, blue is men, pink is women. We did not find any differences between men and women. Race, ethnicity, we found huge differences, the biggest differences between Caucasians in green and African Americans in red than we found between Chinese, Latino people who had just immigrated to the United States and Caucasians and Latinos. So it was not geography, but it was a specific ethnicity itself, right? That was the difference. So then what happens when you shift from eubiosis, a healthy state, to dysbiosis, which is a state of disease? What we found was you shift from aerobic pathways to fermentative pathways, anaerobic environment leading to fermentation. Fermentation releases a ton of byproducts. Fermentation, you use fermentation for breakdown. For every molecule of glucose, you only release 4 molecules of ATP. So it is a lot of hard work. These bacteria are doing a lot of hard work, right? Shromic labor. They're not doing simple work. They're doing very hard work. So because of that, they produce a ton of byproducts. They also, because that environment starts becoming very complex, they don't break down simple carbohydrates and sugars, but they break down more secondary metabolites. They break down very complex molecules, large organic molecules, right? And because the bacteria that are capable of breaking down these kinds of molecules, proteolytic, iron-loving heme breakdown, hemolytic bacteria, those kinds of bacteria also have lots of glycans, lipids, flagella, lipopolysaccharides, etc., right? And so these are capable of living in oxidative stress, high oxidative stress environments, elicit a huge immune inflammatory response. Together, when this microbiome picture changes from a smart work, energy efficient community to a hard work, energy poor, anaerobic, proteolytic, pro-oxidant community, the immune system kicks in, setting up a huge inflammatory response, which then the downstream effect we see clinically as disease. And the more important thing we learned was it is not just the bacteria that are effective in this. The bacteria actually find partners. Archaebacteria seem to be very important partners in this, as are viruses. So viruses and archaebacteria play very important roles in this transformation from eubiosis to dysbiosis. So then we said, okay, this is you. This is you. And yes, genetics plays a role. Yes, your ethnicity plays a role. So then what changes this blueprint? What changes this blueprint? Human behavior. And we started by studying diabetes. So green always is normal for us. So this is a normoglycemic. Blue is your hyperglycemic person. And we found that diabetes does change the microbiome. And when we took this group of hyperglycemics and we put them through, we graded them again based on their glycemic index, we found that the microbiome separates out based on the glycemic index. So certainly hyperglycemia does have a very dose response type of a relationship with a shift in the microbiome. The other thing that is very important is obesity. So here you're looking at yellow is obese individuals, orange are overweight individuals, and green are normal weight individual controls. BMI greater than 25. Here you can see the line of separation. This is my 0 point here. This is my 0 point. Here are the line of separation. So a BMI greater than 25 does make a huge difference. But when you separate the data by males and females, it is definitely seen that BMI greater than 25 is a bigger discriminant in females, but not so much in males. Okay. And the important thing was, this microbial profile of people with a BMI greater than 25 resembles those who have an HbA1c greater than 6.6. So this is already a very pre-diabetic microbiome, right? So the same kind of factors seem to be impacting both of these. Remember my friends, the bariatric surgeons? We did a study with them where we compared the oral microbiome after they had gone through bariatric surgery. And we found that the microbiome, so this is a 0 line, no change, this red line. Each of these dots here is a gene. We did a whole genome sequencing on this. Each of these dots, black dots, is a gene. The red dots indicate significantly genes that are significantly changed, right? So red is significant, the black are non-significant changes. So what you can see is that there's a group of genes that shifts at 3 months. At 6 months, the shift is in the opposite direction. But then at 12 months, the same upward shift persists. So there is a shift in the microbial profile, and the shift happens at 6 months after bariatric surgery and persists through 12 months. So there's a very stable kind of a shift. The other thing we studied was e-cigarettes. And we found, again, green, healthy, non-smokers, non-vapors. Red are smokers, blue are e-cigarette users. We found that e-cigarette changes your microbiome, and this change is not the same as changes induced by smoking. It's quite a different change. And we said, what are the changes induced by this microbiome? We found that the shifts happen in capsule and extracellular matrix, gram-positive cell walls. So the plaque associated with e-cigarette vaping is gunky. It is thick. It is capsule rich. It is slimy. It is more like mucus that comes out from your throat, rather than it is like this one, right? So it is terrible. It has LPS. A lot of iron acquisition products happen there. Functionalities increase. Motility and chemotaxis. So remember the flagella that I talked to you about, the flagella, the pili, which are highly immunogenic. They are upregulated in e-cigarette users. Quorum sensing, biofilm formation, all of which increase pathogenicity. And you would have seen this from Dr. Neelakantan's work. All of these contribute very severely, are very highly overexpressed in e-cigarette users. And this is a high stress environment evidenced by the number of stress response genes. Most importantly, because e-cigarettes are so high in heavy metal compounds, the number of metal efflux pumps is very high in e-cigarette users. And metal efflux pumps play a very important role in antibiotic resistance. And therefore, you can see that there is a huge uptick in antibiotic response in the microbiome associated with e-cigarette use. We also asked if smokers who quit smoking and start using e-cigarettes, or smokers who try to cut smoking by using e-cigarettes, benefit from using e-cigarettes. And we found that No, both these kinds of smokers look very similar to e-cigarette users. So the use of e-cigarettes is a very dominant effect. It's a very dominant effect. Remember I told you the oral microbiome is a very stable, Puri Jagannath kind of a Rath kind of a very stable thing, very slow moving, very stable. It takes this kind of a perturbation, this kind of a huge impact, a perturbation, right? A pressure that changes this microbiome. And so it does change in response to these huge impacts. The other thing we studied was smoking. So here is this is known as the tree of life. Don't worry about what is in the tree of life. Each of these spokes here is a species. So we compared 100 non-smokers to 100 smokers, and we found that smokers have reduction, complete reduction or loss of these species listed in green, and an increase in species that are listed in this orange here. These species, if you ask me what they are species that cause gum disease or species that cause chest infections. So these two kinds of species are highly increased in the oral cavity of smokers. And I'm going to tell you one thing. We studied this further, and then we said, oh, what are these species? We have seen them before somewhere in our life. You know where we saw them? These are species that you acquire when you're before you're 1 month of age. So what smoking does is it reduces that familiar friends that you got when you were before you were 1 month of age. It removes them, it kills them. And because it kills them, your immune system doesn't recognize a benchmark. It doesn't have a baseline anymore. And along with that, it opens up the oral real estate for pathogens to overgrow or to colonize, and therefore it changes the entire microbial structure and it alters the immune benchmark. So it changes both of these things. So the next thing we found was whether you're smoking or you're diabetic or rheumatoid arthritis, the type of insult to this ecosystem is it changes the microbiome in a very specific way. This is the microbiome associated with smoking. When you smoke, you have an anaerobic, gram-negative, antibiotic, heavy metal resistant microbiome. When you're diabetic, you have a fermentative, gram-positive, proteolytic microbiome. When you have rheumatoid arthritis, you have a microbiome that is rich in bacteria that can metabolize arachidonic acid and produce citrulline. So the microbiome is very specific in these changes. And then we said, oh, okay, remember we were playing this game of rock, paper, scissors? Do these environmental factors, human behaviors, impact everyone the same way? And so we started stratifying our data. We took the smokers. Remember I showed you this picture a few minutes ago? So we took these Caucasians and these African Americans and we stratified them based on smoking. Here are non-smokers. You can see the difference between the two groups. But here are smokers. So here are smoking Caucasians in red, and here are those smoking African Americans in these clear circles. You cannot tell the difference between an African American and a Caucasian based once they start smoking. So the ethnicity-based difference is gone when you start smoking. In the game of rock, paper, scissors, smoking wins over ethnicity. Remember when I said with gender, there is no difference between males and females, but when you add smoking to it, here are women who smoke and don't smoke. You see no difference between them, but here are men who smoke and don't smoke. And you can see a clear clustering between the men who smoke and don't smoke, right? So between the game of smoking and gender, gender wins out. Smoking affects the male gender more than the female gender. Remember a few slides ago, I showed you obesity affects the female gender more than the male gender. So this is seen here. Smoking and diabetes. Here we have three groups, smokers, diabetics, and diabetic smokers. And we find that in this game of rock, paper, scissors, diabetic smokers are more similar to diabetics than they are to smokers. So diabetes wins out over smoking. Smoking and rheumatoid arthritis. Rheumatoid arthritis wins out. Are you seeing a pattern? If it's an endocrinological defect or an autoimmune defect, that wins out over the human behavior. Smoking and pregnancy, pregnancy wins out. Smoking and e-cigarettes, hey, no prizes for guessing. I already told you the answer. E-cigarettes win out. Smoking and diet, smoking wins out. Smoking wins out. So changing your diet without quitting smoking does not improve your oral health, unfortunately. So then we took all of these and mapped them, right? Here is a microbial profile of someone who's completely healthy. Here's a microbial profile of someone who has severe gum disease. And this is the halfway mark. Here is 1/4, here is 3/4. We put with all of this on a map, and we said, where does the microbiome of different perturbations fall on this map? When you're pregnant, you're very close to normal. Yes, your microbiome shifts a little bit during pregnancy, but then it falls back to normal once the woman has delivered the baby. If you take a single antibiotic course, it changes your microbiome, but then it comes back to normal. It shifts it a little bit. A single course of antibiotic does shift the microbiome towards about 1/4 of the way, but it doesn't shift it over much. Obesity shifts it about 1/4 of the way towards disease. Rheumatoid arthritis shifts it more close to halfway to disease. Smokers with rheumatoid arthritis, definitely halfway. Smoker. Hyperglycemia shifts it more than halfway towards disease. Smokers on antibiotics shifts it. Hyperglycemic smokers. Obese hyperglycemics. Obese smokers. Vaping. Obese vapors. Smoker who vapes. Obese smoker who vapes. Obese hyperglycemic smoker. So sorry, let me put this. What did I do? Whatever, I lost this. There it is. So you can see that the winner in this Game of Thrones or Game of Hunger Games is the obese smoker who vapes, right? So cumulative human behaviors can change your microbiome much more towards disease. The reason I tell you this story is this map is because the study that we did, this game of rock, paper, scissors, every one of our patients was clinically healthy. These are patients I see in my practice, in my residence practice, and I say, oh, you're doing a great job. Come back and see us. They brush, they floss, they do good things. They see their dentist every 6 months. They're good people. But they still have a shifted microbiome, a dysbiotic microbiome, which tells us that systemic factors have an early impact on the oral microbiome. The impact is measurable. The impact is factor specific, and therefore the microbiome can be an early warning system, like a canary in the coal mine. And that example comes from back in the days when the miners used to go down coal mines, and all of these poisonous gases used to come out, but they were so dark you cannot see. So what did they do? They carried a little canary. The canary will keep on singing. You go cheep cheep cheep, they'll keep on singing. But when the canary's lung is so small, so even the smallest bit of poisonous gas will kill the canary, right? So if the canary doesn't sing, it means that its poison gas is coming, so the coal miners go back. So even the smallest change can be measured by the canary's song. And therefore, we believe that the oral microbiome shifts so quickly and shifts so definitely that it can be used as an early microbial warning system, which means that your patients need personalized care. There is no one size fits all. There is no global mouthwash. There is no global toothpaste that is going to change these things. They need personalized care. The other thing we learned is that the risk for disease might begin much earlier than we think. So this, remember the slide I showed you about when you at what time in your life you acquired these teeth? We asked, where are these bacteria coming from? Not when you acquire teeth, where you acquire these bacteria. So we said, where are these bacteria coming from? We found that the predentate, the dentition, that the bacteria that come in these babies at 1 month of age comes from the mother, directly from the mother. So your mother is your biggest source of bacteria. And so the mother's lifestyle impacts the baby's microbiome. Here is a microbiome of mothers who smoke, in gray, and mothers who don't smoke. So you can see that the two babies have very different microbiomes based on their mother's behavior. So the mother who smokes changes her baby's microbiome, right? And what does she do? She actually enriches the baby's microbiome for pathogens. How does she do this? As we were doing our study, Kristen Agard came out with this beautiful study where she showed that the placenta actually harbors a microbiome. And the source of the bacteria and placenta are from the mouth, from the oral microbiome, not from the gut, not from the skin, not from the vagina, it is from the mouth. Okay, I'm not trying to tell you that the bacteria go from the mouth to the placenta and get into the baby. That is not what happens. What happens is this. This is a paper by Egia Zaura and her group, and this is a very nice review article where they showed that bacteria move from the mouth of the mother to the placenta. The placenta has a blood-brain barrier, correct? It has a barrier. This is the mother's side of the placenta. This is the baby's side of the placenta. This is the mother's side of the placenta. This is the baby side. Sorry, this is the baby side of the placenta. When the bacteria are sitting on the mother's side of the placenta, they train the fetal immune system. Here are the antigen presenting cells. Here are the T regulatory cells, the T17 cells that are developing a fetal immune system. So these bacteria are sitting here training the baby's immune system to recognize them as friends. So when the baby is born and the mother takes a spoon out of her mouth and puts the bit in the baby's mouth, and there's a transfer of saliva, the bacteria from the mother's saliva are easily accepted by are tolerated by the baby's immune system because they recognize them as friends. When the mother carries pathogens and they are recognized by the baby's immune system as friends, the baby now accepts these pathogens that they are allowed to colonize, whereas any other baby would absolutely reject it because it would not have seen it in the womb, right? So that is what happens. So which means that we need to, when we think of a patient who's 20 years old and sitting in our chair, and we're like, oh, you have gum disease, or you have cavities. We never ask them, did your mother smoke? Was your mother on antibiotics? Did your mother have gestational diabetes? We never ask questions. So the risk must have, the disease begins much, much, much earlier, right? So basically what we're doing these days is surgical. Everything we do is either surgical or medical. And we always tell the patient, oh, come to us when you have disease. We will treat you. We will make you better. We are God. We can treat you, right? And so the patient doesn't own their disease. The patient doesn't own their health. And they don't understand it's important to prevent disease rather than to treat it once it comes, right? So these become very, very important. The wellness model is the one that's most important. We need to focus on preventing disease. Primary prevention, don't ever get the disease. But also secondary prevention, if you get the disease once, don't get it again. Secondary prevention becomes very important. And for that, secondary prevention, risk becomes very important, risk assessment. The patient is high risk, you will focus very closely on them, close monitoring, multiple methods, multiple modulatory methods, not just one technique, multiple different things to educate, motivate the patient, and to put the wheels of the wheels back of this car back in the patient's hand to say, you drive this car. Your health is your concern, is your business. I'm only here to coach you. I'm your life coach. I'm not here to fix you, right? So that kind of an attitude to make yourself the practitioner, the monitor, and the patient the driver of their health, right? So this is the entire concept of this new classification system that we developed. A group of us came together, about 139 of us, 29 of us came together in Chicago in 2017, in November, to create this new classification system. And if you're looking for me in this picture, there I am sitting, standing all the way to the back, right? So this is a group of the brightest brains that I had the absolute privilege of working with. We came up with a classification system that truly embraces this concept of risk. That yes, you can have severe disease, but that only measures the history of disease. It doesn't measure how you can come back, how you can bring yourself back, or how badly, what the outcome of your treatment is going to be. And so just like you have for cancer, we have a stage and we have a grade. The grading assesses the risk for future disease and how to best prevent it. So I'm going to skip through these in the interest of time, these. And I'm going to pause here and take any questions that you might have for me. I'm just trying to get to my stop screen thing, and we shall go from there. Give me a second. There it is, stop share. All right, now let's chat.
Thank you so much, Professor Purnima Kumar. So very interesting lecture as always. And I'm fairly certain that the audience are awestruck and shell-shocked looking at some of the evidence that you presented, dentists and non-dentists alike in the audience. So I don't see any questions at the present moment. We have now. Okay, so there's one question. If I understood correctly, the microbiome of individuals converge as they progress to a disease state. And this convergence is irrespective of race, ethnicity, etc. So can you please elaborate the role of personalized medicine as the diseased microbiomes have converged?
Excellent question. Excellent question. Oh my goodness, that is a very deep question. Yes, the microbiomes converge in types of their function. Very good. So the microbiomes converge in the terms of their functionality. But then, again, like I said, the stage of the disease, whether you have early disease or whether you've had this disease for 20 years, or whether you have this disease on a molar tooth versus a premolar tooth, those factors still drive the type of microbiome that you carry, still drive the functionality of the microbiome. So the biogeography of the mouth becomes now the driver, not your ethnicity, not your race, not your gender, but the biogeography of the mouth. So suddenly from the big picture, it becomes a small picture, a little camera that's happening inside your mouth. So that is where we need to start thinking about personalized medicine and understanding if you're a smoker with disease versus a disease in a molar tooth versus if you're a smoker with disease in a front tooth versus if you're a smoker with disease in multiple teeth versus single teeth, the treatments will have to be different. So that is where the personalization begins. And that's a whole new story for another day. I haven't even touched on that topic today, but we will talk about this in the future. But this is an absolutely brilliant question. Thank you.
There's one more question, which is on prebiotics, and I want to add up a bit more content on that from my perspective. So the question is, will prebiotic lozenges stop disease progression? And I would like to add up on the probiotic aspect as well. Do you think prebiotic, probiotic, the so-called symbiotics, can be alternative strategies to what we currently use as antiseptic mouthwashes, for instance?
Antiseptic mouthwashes should be gone. I mean, they should be made illegal, I think. They are, you cannot globally destroy anything, right? Our biggest resources are microbiome. If you're worried about natural resources like diamond and gold and zinc and aluminum, why the hell aren't you worried about a natural resource like an antibiotic? Once it's gone, it's gone. There's no more antibiotics left. I mean, you will die. We'll all die from getting your finger cut by a little nail somewhere, right? Why do you want to die like that? That's a horrible way to die. So we'll start there. Then let's talk about probiotics. Remember what I said? There is more and more and more evidence accumulating that your microbiome is based on familiarity. It's based on immune tolerance. So only species that are already familiar to you will colonize you. Otherwise, every time you eat, every time you drink, why should not bacteria from this glass of water colonize your mouth and a probiotic colonize your mouth? I mean, what is the rule, right? Just because you like a probiotic and you don't like the bacteria in the glass of water, that is no. So fundamentally, your mouth is and that's the most protective thing. This is the gateway to the human body. Like the nose, I mean, less things go through the nose, more things go through the mouth, and you cannot afford fluctuations in the microbiome, right? In the oral microbiome, you should not. And so that is evolutionarily conserved. So a probiotic, there is very little reason for a probiotic to work. Very little reason for a probiotic to work. A prebiotic can change the environment and therefore promote colonization of those familiar friends that you were born with. So a prebiotic definitely will have a role. Definitely will have a role. We need to find the ideal prebiotic and tailor the prebiotic, not just the prebiotic, but also the prebiotic method of delivery for that particular patient with that particular condition and that particular tooth. Absolutely. So I believe the future is prebiotics.
All right, thank you. Do we have time for 2 more questions, Professor Purnima?
Yes.
All right. So one question we have is, how much does the variation of the oral microbiome profile affect the pathogens that play a role in disease manifestation? Are there variations in the key players considering how oral diseases are manifested by multiple interacting species?
Correct. So I'm going to be very honest. Species are not as important as the functions they bring to the table. The reason, when you look at it, we sequenced 4.2 terabytes of sequences with the Human Microbiome Project, and what did we find out from it? No two humans are the same, right? Great. Heterogeneity is intense. But then, when you look at all the functions of these bacteria, yes, 33% of the human community is the same. Yes, 63% of the human community is the same. So functionality, if you're an ecosystem, let's think, if you're a coral reef, what do you have to be? You have to be able to live in salt water. You have to be able to live in a tropical climate. You have to be able to live at 3,000 feet of seawater pressure. You have to be able to coexist you know, Dory and what's his name, Nemo, and all of those kinds of fish, right? So the functionalities, your functionality, you can be any coral as long as you can fulfill these roles. You can be any coral, right? That is important. So same thing, if you're a rainforest, if you're a tree in a rainforest, what do you have to be? You should have big green leaves. You should be able to catch and use rainwater. You should have a tall tree trunk so you can have a big canopy and reach up to the sun, right? So the types, your requirements of fitness, these are called fitness requirements. Fitness requirements of a species drives their presence or absence, which is why when you look at species levels, there's so much heterogeneity. But the functional requirements of the ecosystem will drive the kinds of species that live there. So variations in oral microbiome, the pathogen, there is no pathogen. A pathogen does not classify itself as a pathogen. It is what a human calls a pathogen. And there is no true oral pathogen. They are pathobionts. All healthy people, many healthy people still carry these pathogens. They are insignificant to human health because they are in low numbers and because they are out there. The environmental conditions are not right for them to express their virulence factors, or even when they express their virulence factors, they are in such low numbers that they don't matter. Their quorum signals don't matter. Nothing matters, right? They are insignificant, they're inconsequential. So they're still living in a healthy person, but they're inconsequential, right? They're just aam aadmi. They're just living there. They're not becoming the political neta. They only become the political neta much later on. So there is no pathogen. There is only a pathobiont. And so the species model of disease causation has to go. If we have to make any progress in the microbiome, the species model of disease causation has to go. We have lived too much P. gingivalis. Now that needs to go. I hope
Thank
I
you
answered
so much.
the question.
Perfect. Yes, perfectly. Uh, can oral microbiome dysbiosis act as or can be developed as biomarkers for systemic disease progression?
Yes, I'm going to argue yes. There are lots of studies, some in our own lab, that is really looking at these kinds of things. And yes, they require large scale studies. They require different types of, just like the COVID vaccine, different ethnicities, different geographic locations, everything. So if there is any interest, let's talk. Let's talk about collaborating on a large scale and developing a giant model instead of studying little teeny tiny studies in little communities. Let's do this.
All right. Thank you so much, Professor Purnima Kumar. Thank you for enlightening us today, as always. Thank you very much.
You're welcome. Thank you all. Be well.
Thank you.
Okay. Thank you, Dr. Prasanna. Thank you, Professor Purnima, for this talk. So 4 different talks, 4 different aspects, and everything was amazing in its own way. So let us break for lunch from 1 to 2 and get back to the session. For the second half of session, we have a bunch of entrepreneurs who have showed and proved that anything which we develop in lab can be brought to market with persistent dedication and hard work. So let us all meet at 2 o'clock. We have Dr. Milind joining us, followed by Dr. Asmita Prabhne from different startups. So we'll break for lunch now. I welcome everyone for the second half of this symposium. Now I invite Dr. Shrikant to introduce our session speaker, Dr. Milind. Over to you, Dr. Shrikant.
Hi, everybody. It's my pleasure to chair this session. And I would like to now introduce Dr. Milind Chaudhary, who is actually the founder and CEO of We Innovate Biosolutions Limited in Pune. And Dr. Chaudhary is a microbiologist, and he has finished his master's from Nagpur and also his PhD in microbiology from Agarkar Research Institute, University of Pune. He's a recipient of several prestigious awards like Biotechnology Ignition Grant and also Small Business Innovation Research Initiative Grant, and also several awards like Healthcare Acceleration. He has won competitions like Healthcare Accelerator competition by IIM, and also he's the recipient of AABI Torch Award in 2018 for the best entrepreneurship in Asia. And he's of notable is the point that he's the first person from India to win this award, and he has been in the field for more than 10 years in the field of rapid diagnostic microbiology, and he has been successful in rolling out two technologies like UTI RAP, that's a UTI detection kit, and also DNA RAP, that's a single-step DNA isolation method. And today, Dr. Milind Chaudhary will be talking about SilverGuard, a story from laboratory to market. And I'm really excited personally to know more about the story. And audience, if you have any questions, feel free to post them in the chat box. And I will not take much of the time. I would now pass on the dais to Dr. Chaudhary for his talk. Looking forward to your talk.
Thank you very much for that introduction. And thank you very much for inviting me for this talk. I've been listening to all the speakers since morning, and then I've realized that there is not much of a science in my presentation, though it is from a company point of view. But being a scientist myself, I have actually tried to cover as much as what we have done, and how we actually rolled out a science technology which is mostly onto our lab tables to reach to a level wherein it is now going into the market, and then is going to create a lot of impact in saving lives and preventing infections. So let me start by sharing my screen. And yeah, so I'm the part of We Innovate Biosolutions. We are a company, we are a startup. Basically, just to give you a small introduction about myself, I'm right now serving as a founder and CEO of We Innovate Biosolutions. I call myself an entrepreneur innovator, and I'm very, very passionate about infection prevention, because since the starting early phase of my career itself, I was very much interested in infection prevention practices. I'm a die-hard Indian. I'm an optimistic, and I work in the interface of nanotechnology, biotechnology, microbiology kind of area. As you've already know now, I'm mostly into technology development rather than more in the basic sciences of it. And we've got multiple patents now, few technologies rolled on, few technologies we're already working. And then today, what I'm going to talk about is SilverGuard, which is our upcoming product, which finally now had actually, completed the whole cycle from discussions over coffee table with your scientist friends, and then finally coming out as a product into the market. So just to give you a sense of what We Innovate Biosolutions is, so we are working in the area of infection prevention and control very focusedly with multiple technologies that we are right now developing. So we are actually on a mission of zero infections, and we want to at some point of time work on stopping the spread of AMR, because which we see from infection point of view is something which is now becoming a new era of pandemic. And AMR would be even more difficult to handle than the current pandemic that we have actually seen moving forward. And our vision is to provide technologies to combat infections, and how we can decrease the infections into the settings wherein they are most highly prevalent and situations. So we started in 2016 with two of my friends from my grad school itself. And as I told you, we were actually having a lot of brainstorming across our coffee tables, and then ultimately we're roaming around in hospitals for some problem identifications. And then we reached out with a problem to government agencies, and we got a biotechnology ignition grant in 2017, from where actually the company journey started. So mostly from that point onward, I have been mostly getting government grants to fuel this activity, and now we have raised some VC fundings to actually take the product to the market. So in 2018, a biggest milestone that we hit was inception of our nano AGSide technology. Now, nano AGSide technology, I will explain you about how silver plays a very important role in our day in day out lives, and how this molecule can actually completely revolutionize the infection prevention scenario that we are seeing today. We have been not working as a startup, but working as a complete corporate company since a very, very inception phase. And then we have received ISO 13485 very fast in 2019. And then we got our patent also granted in recently in 2020. And what we do essentially is we work in two ways. Is one is we actually implement our basic technology, which is nano AGSide, that is silver nanoparticles, with multiple clients, and then help them develop antimicrobial products so that the infection prevention can be taken part. And on the other hand, we develop and we work into a specific field of medical devices, because we see that medical devices are one of the major reasons of spread of infections, and if we can control them by making antimicrobial medical devices, that is definitely going to have a substantial impact on the way we are looking at infection prevention control today. So let me give you a sense about something that you already know. Being Indians, we already know a lot about silver. We have been eating in silver utensils since the childhood and all. But why was one of the bigger questions. So silver's story goes back to BC, and people were eating food in silver utensils and all those things, and you have already seen the royal connection in the silver, because people knew that okay, fine, this is a metal which actually has a lot of properties, whether it is infection prevention activities, or whether it is immune supplement kind of activities, or any other ways that silver plays a role into our daily routine all around ourselves. Then people started using silver as a treatment in water for purity of water and sanitation and those kind of practices. And then slowly, for a very long time, till the advanced technologies were developed, it was still used into the most conventional way it was possible. And then from 1800s onward, it has actually picked up momentum and started seeing silver into majority of the things that we see around us. So as technology progressed, the use of silver also started progressing very fast. Almost a few decades ago, people have started exploiting the silver's role into medical benefits, and then where they started using silver into multiple products like immune supplements and all those other things. So what essentially silver does is, what we were interested into is, silver has a very good and broad spectrum antimicrobial activity, and this antimicrobial activity needs to be exploited to a level wherein it can actually be an industrially applicable solution which can be implemented directly into the market. So we started studying silver from multiple point of view, and then we found that there are silver ions. Metallic silver also works very well. But then when we came when we got introduced to nanotechnology, and nanotechnology can reduce down the size of any kind of metal, and then what essentially it does is actually increase the efficiency at a decreased dosage. Because of a larger surface to volume ratio, the ion release is much faster, and it gives you multiple fantastic activity. So this is the basic science behind silver. Now, silver has a role in antimicrobial activity in terms that it non-specifically kills the bacteria by either binding to its respiratory enzymes, and there are multiple roles wherein silver binds to the bacteria and inhibits the bacteria. So just to prove that aspect, we actually did a surface topology of AFM. So if you see, the first layer is an E. coli cell, which is actually, we've gone through the smallest detail of E. coli cells on the surface, and you'll see the arrangement of the proteins and all the polysaccharides are very well aligned on the silver surface, on the E. coli surface, and this is at 250 nanometers. And the moment you treat them with silver nanoparticles, imagine what happens to a E. coli cell. It literally punctures the cell and bursts it open, and then you see the surface topology, which was very, very uniform over here, had actually been completely disrupted by the effect of silver nanoparticles. Just to give you a larger sense of it, if you can see, these are silver nanoparticles which are in the tune of 50 to 60 nanometers, and then they are actually inhibiting the E. coli cells, and they're literally disrupting the E. coli cells. But that's a physical disruption mechanism. What silver nanoparticles helps you do is that it gives you a dual mode of action. And in this dual mode of action, one is a physical activity, because it's a hard inorganic molecule which is actually hitting to an organic molecule. So it is a hard to soft kind of interaction. It literally is called as a silver bullet, you could say, which punches open the cells. But on an ionic basis, silver works very well. So an immediate action you get with a hard inorganic molecule, and on an ionic basis, you're getting multiple roles of silver in inhibition of the bacteria. So whether it is being binding to the thiol groups on the surface bound protein, to the respiratory enzymes, then creating a reducing environment. So there are multiple reasons by which silver can act. Now the advantage, once you make a nanoparticle, that you get is the ion release is much faster and a sustainable ion release. So at a very, very small dosage, you can actually take advantage of silver by converting it into nanoparticles. So when we actually tried to study the activity, we said physical activity is happening, and the physical activity can be seen also by the fluorescence microscopy, whether the dye is adhering to the surface, what is exactly happening. So the physical disruption we wanted to prove, which we actually saw by AFM images. And the ionic basis, we wanted to see whether it is actually compromising on the membrane potential, or is it compromising the membrane of the bacteria. And with the fluorescence dye, we could actually see that the dye is able to penetrate inside because of the compromised membrane of the bacteria. And that's how we could actually see that the dual role of silver is working very well, and it can be exploited for a commercial application from an anti-infective point of view. And then we said, okay, fine, let us actually see whether is it going to give you a long-term kind of effect activity. And secondly, a lot of people were very skeptical about resistance issues. So they said any antimicrobial which comes at any given point of time, chances are that there will be some or the other resistance which comes in. However, from papers and background literature, you could see that the resistance to an inorganic molecule is a little difficult aspect for the on the bacteria, again only because of the mode of action that it is working on. Besides, we have seen, and as I've already shown you, the role of silver. It is like thousands of years we have been using silver, and organic molecule we started using only 5 decades ago from the discovery of antibiotics. And then we have seen that in the 5 decades itself, all the antibiotics are right now kind of out of the market, and then all the there are a lot of bacteria have gone resistance to majority of the organic antibiotics that we have seen. So I think it's a good point to actually go back and see why inorganic molecules can be of use and what it can do to prevent resistance in the bacteria. So what we did essentially is we actually kept the bacteria with minimal concentration, sub-MIC concentrations of silver for a longer period of time, up to 100 days. And then we have seen that whether it is still able to kill the bacteria, the same efficacy that it was able to do that. And we were surprised to see that there was no resistance whatsoever in either in gram-positive, gram-negative bacteria. And we could see that the efficacy remains the same. The inhibition zone remains the same for a very long period of time. So up to 200 days also we have studied, and then we could find that the resistance is not something that is very easy for bacteria to gain against an inorganic molecule, and specifically in case of silver for that matter. So we said now, if you want to commercially exploit, we have to actually do a safety study, because when we say nanoparticles, a lot of questions are raised regarding the toxicity aspects of nanoparticles. And majority of the paper that we actually see on the toxicity of nanoparticles are mostly in vitro papers, wherein people are doing toxicity on cell lines. And this question needs to be addressed on a longer basis in terms of seeing whether can we translate the results of a monolayer in vitro activity into a real in vivo activity. And that's why what we did was the first activity that we did. We have seen a lot of in vitro assays, but we said, okay, fine, we could try and give the maximum dosage possible that we could give to the animals, and both the routes, orally and intravenous, and see what happens. So in the oral toxicity, we said we found out that we could actually go up to 2000 mg per kg body weight, which is way below the higher than the efficacy dose of silver. So the activity is in silver's MIC values are somewhere nanoparticles range from 5 microgram per ml to 10 to 15 microgram per ml, and that's max for any type of bacteria, whether it is multi-drug resistant bacteria, gram positive, gram negative. In fact, we have seen in some of the viruses also the MIC value does not go much higher. So we have done the oral toxicity. Then we said there are chances that it can be inhalation toxicities also, because the nasal cavities can actually absorb the nanoparticles, and whether it is going to create any kind of systemic effects or not. So we have not even just seen the physical appearance, but we have also seen the organ weights and the silver distribution into the organ. And then we have found that we can still achieve a maximum attainable dose, which is much more in a comfortable zone wherein the efficacy concentration of the silvers actually work in. And then we did multiple more studies in terms of eye irritation, vaginal, penile. We have seen whichever dermal tissues that we could get, which are very sensitive and can give an easy access to nanoparticles, and what are the effects on it, whether there is some sort of inflammation which occurs or any kind of activities. And we found that fortunately, even if we talk about cytotoxic effect of silver nanoparticles, that does not translate into an in vivo activity, because when the animal is bigger, the biodistribution is different, and there are multiple components which act and then binds and does the actual activity. So even though we were actually interested as a company into only physical activity of silver, not exactly in a drug, in a systemic activity wherein we can actually prescribe antibiotic silver as a replacement to antibiotics, yet we said we have to have a complete thorough understanding of what is happening inside the body. So fortunately with this, we were able to introduce silver nanotech technology, and we have now got a patent. The technology is called as nano AG side technology, wherein we have a unique nanoparticles which are much stable, scalable, affordable, and versatile for multiple applications. Now, another interesting fact is when I have been into silver nanotechnology area since last 10 years, and then we know silver nanoparticles particularly since last 3 decades now, and yet we haven't seen a commercial exploitation of silver nanoparticles to the extent that we should have actually seen. And one of the major reason for that was its activity, that's efficiency, its scalability, cost, and toxicity. So there were three more aspects which needs to be addressed. And what we did, and that's why we could receive a patent, is we have actually looked at from a completely different aspect, wherein how can we reduce down the cost of manufacturing of silver nanoparticles? How can we scale the manufacturing of silver nanoparticles? And I can proudly tell today that we have a capacity of around 300 liters per day capacity in a GMP manufacturing setup at our end. And we could scale silver nanoparticle manufacturing to this level wherein it can now be commercially used and applicable. Besides, we have worked on the stability of the nanoparticles. So nanoparticles are mostly labile to majority of the conditions, whether it is pH, temperature, and all these kind of things, wherein usually they tend to aggregate together and settle down. So the stability was one of the major reasons in majority of the nanoparticles that you see on the shelf today. So we have very well engineered our nanoparticles to actually bring in high amount of stability to the product, wherein now we can say that it is at room temperature stable at around 3 years stability. So the nanoparticles are giving consistent result in terms of small sustained release of ions and giving you a larger efficacy at a very small and minuscule dosage. And now we have its proven efficacy, and we have done multiple clinical trials around the product and around the nanoparticles itself. We have done, in fact, antiviral studies. Now coronavirus study is also going on. We have done the toughest virus to kill, the murine norovirus, we could kill in 1 minute time. So effectivity of silver nanoparticle was already there. Now came the bigger question for us was, where are we going to look forward for the applications of these silver nanoparticles that we have? Because ultimately, if you have to run a business, it needs to solve a real problem, and then it could actually show up as a tangible product which can be sold into the market. So we tried looking for multiple problems, and this was a very big problem of antibiotics and lack of hygiene practices around. and a biggest example of which had actually occurred as COVID-19, and the pandemic hit, and it actually completely revolutionized our pitch in terms of when we were before pandemic talking to people about prevention is better than cure. A lot of people were skeptical about it, but now imagine the situation that all of us are right now following prevention practices, washing our hand multiple number of times. And I mean, what could be the biggest example of prevention is better than cure? So today we are at a center stage wherein superbugs can become one of the biggest threat tomorrow. So we have to rationally start using antibiotics and minimize the uses of antibiotics so that it can be helpful for us for a long-term activity. Besides, hospital acquired infections are now creating a bigger problem, because in hospital settings, majority of the superbugs are found, and they are usually being spread by from the hospital situation. So people going for smaller surgeries come back with bigger infections, and then it takes a very long time for them to treat those infections, and cost burdens and all those things are at a higher end altogether. So a virus, how an infection needs to be taken seriously, something that this pandemic told us is a huge amount of loss that we have seen in the last 1 year. And it gave us an understanding that we need to take infection prevention practices very seriously. Besides antibiotic, we need to be very conscious about how are we going to use antibiotics. So you said this problem of AMR, problem of infections and everything is connecting to only one single dot, which is hospitals. And hospitals needs are the places wherein we are going for treatments, people are going to become better. But if in case you catch an infection, and there are very good chances that you will catch, around 5% chances that you will catch a hospital acquired infection when you are admitted into the hospital. There are a huge amount of fatalities which are already happening because of this specific reason, and a lot of people are being affected. So we said, yes, hospital acquired infection seems to be a very important problem, from both point of view, from an economical point of view as well as from a social point of view. And this is the problem that we need to address as a company. And majority from an economical point of view, majority of the hospital revenues comes in the first 5 days of hospitalization. And besides this, any kind of infection is actually a troublesome thing for hospitals as well. So if you catch an hospital acquired infection, chances are that your hospital stay could actually increase by a substantially higher number, which is going to create a hole in your pocket, and also emotional issue which is already there staying into the hospital. So we said, if hospital acquired infections, what are the areas in the hospital acquired infections that we can address? And then we found that there are multiple infections which are very well termed as hospital acquired infections, and the majority of it was found to be UTIs. So 40% of the hospital acquired infections are UTIs. So we said, okay, so this is a very interesting problem and UTI needs to be focused. Besides, we are working in UTI issues from a very long time. So the hospital acquired infections is a very big problem, and it is causing multiple amount of excess usage of antibiotic. So now we found out that there are these problems and the usage of antibiotics. So AMR is also a problem which is actually being created because of hospital acquired infections. So it's a vicious cycle. You get infections, more number of antibiotic, AMR. So, and if you get an infection again from AMR, it is again going to create a problem. So if you have to break this cycle, we have to actually make medical devices antimicrobial by themselves. So we were actually thinking about UTI, and then we found out that majority of the UTIs are happening in hospital acquired infections are because of catheter associated UTIs. And we all of us know that catheters are usually the tubes which are inserted inside the body for draining the fluid out. Now, these are the medical devices which are half inside the body and half outside the body. Besides, since they are stuck to human body, there is no way to clean them or disinfect them, or there are no sterilization protocols which are available by which you can continuously keep keep a catheter, disinfected for a very long time. So we said, yes, this is a very interesting problem to work and to solve. So catheter associated UTI happen usually when a bacteria climbs up through the catheter inside the bladder, or sometimes it can be intrinsic. So it could be your normal microflora which is actually infecting, invading, and then causing an infection inside the bladder. Interestingly, once this catheter is inserted, it stays there for from 2 days to it can go to up to a month long time. The patients which are usually catheterized are people either immune compromised, geriatric patients, and with some of the other problems with multiple medicine. They're on high amount of dosage of antibiotics or other medications. So usually for an infection to cause, it becomes a very easy and a very common host that it founds, and infections are very common in catheters. Besides, in few cases, the bacteria from the in the UTI actually creates conducive environment for them to actually form biofilms around the catheters, and then ultimately these biofilms along with a lot of ions which are coming in the urine forms encrustation on the catheter, and sometimes the catheter completely blocks, which creates another bigger problem, which is back pressure on the bladder and discomfort to the patient. Sometimes it causes more infections, and then it can go systemic and ultimately can cause fatalities in cases. So it's a very big problem which needs to be addressed. So we said it is inevitable to make these tubes itself antimicrobial. So if we can do that, essentially there are chances that we can reduce down the infection load from which is causing because of the catheters. So we actually worked on a technology called a SilvoGuard, wherein we have worked out, we have used our nanoegicide and we have combined it with the medical devices, and we combined it with the base material of how the tubes are made, and then made a completely antimicrobial catheter. Now, when we are saying completely antimicrobial catheters, we already know that silver nanoparticles are going to give you a long-term activity. Besides, when they're impregnated and become the part of the material itself, they're going to give you a sustained and a long-term activity for a very long time. So that is how SilvoGuard actually came into picture. And then we said, okay, fine, latex-based urinary catheters are something that is the area that we need to focus, because this is the material on which majority of the bacteria are growing and majority of the infections are happening. So how SilvoGuard catheter actually helps in preventing infections? So basically SilvoGuard is made up using a patented technology, which is again nanoegicide that I told you. This has impregnated nanoparticles completely inside the catheter material, and that's how it's going to be antimicrobial till the lifetime of the product. So even if after use you throw the catheter, the disposal is also safe in case of catheters. So once the catheter is inserted in the body, the nanoparticles continuously keep releasing silver ions. So whatever bacteria which comes either in the vicinity of the catheter or sits on the catheter, they're going to be completely killed. So it's not just a preventive activity, but an antimicrobial activity that we were looking on the basis of catheters. So here we have our antimicrobial catheters, which actually gives you a complete antimicrobial activity. Now, when we were making Foley's catheter, now this catheter is called as a Foley's catheter. We said we should also look at what are the more other attributes of catheter which can create problems. And then we realized that this catheter have been used since 20-30 years, and there has been no innovation happened in this design. So today itself, the same design of catheter is being used, which were being used almost 30 years ago. So we try to find out what are the other problems that are because of this catheter, and can we actually do a little design modifications and help them address those problems. So one of the bigger problem that we found in the catheters was the problem of urine retention. So because this is usually a balloon, and I think in the earlier slide I could show you that the balloon is inserted inside the bladder, and it actually holds the catheter inside the bladder like this. And this distance between the tips. So here there is a drainage tube wherein from the urine actually passes into the urine bag. Now in majority of the designs, the distance between the tip and the bladder and the balloon was much larger. That's why there is a problem of urine retention, and the more amount of urine retention, chances of infection increases. So what we essentially did as the first thing is we decreased down the distance and engineered the distance between these two, so that there is a very less amount of urine retention in the bladder, which also decreases the chance of infections. And then we worked out on the physics point of view of the catheter, how it is inserted, how we can actually decrease the pain to the patient, because these are going to be inserted through the urethra, which has a very smaller lumen. and we have actually worked out the ports, different ports in the catheter, which can actually be one of the reason of extrinsic source of infections in the case of UTI. And then the biggest attribute is antimicrobial activity till the lifetime of the product. So with this, we wanted to see whether we are seriously getting the efficacy that we actually are expecting to get from the catheter. So we did multiple experiments, and the experiments are still going on into multiple level. And you could see here that the catheter is very efficiently killing the gram negative as well as the gram positive bacteria. And then we have a whole battery of 20-30 bacterial species that we have tried, including multi-drug resistant species and multiple viruses. And we could see that, yes, there is a good amount of antimicrobial activity that we are seeing, and the antimicrobial activity is sustainable and long-term effect. We have also studied the biofilm, and we could see that in 48 hours itself, the biofilm can grow very well on a catheter surface, because this is latex rubber-based material, which might have some kind of striations and all. And if you could see that this is not a normal uniform material, so it's a very ideal surface for a biofilm to bind onto. But however, the moment silver nanoparticles are there in the catheter, it does not allow the biofilm to form on the catheter. Besides, there is not even a single bacteria seen, and then we have done some in vitro studies also and seen that there not even a single bacteria which remains on the catheter. So it gives you 100% antimicrobial activity as long as the silver is there. Now our long-term experiments are still going on, wherein we are seeing the catheters to be kept for 3 months or 4 months time period, because we have seen till 1 month we are getting very good antimicrobial activity, and it's still a sustainable activity that we have seen in the case of catheters. Then we have done an extensive study which is called as biocompatibility analysis for seeing the antimicrobial catheter. And again, the toxicity issue needs to be solved in the catheter. So fortunately, as I've told you, for the silver nanoparticle, even if it is given orally, the dosage is 2000 mg per kg body weight. And effective concentration that needs to be used in a catheter, which is hardly 10 grams, is not more than 100 microgram of silver. So ultimately, the dosage use of silver is very too low beyond the permissible limits and also the toxic limits that we have already seen. So the problem can be solved in that. Yet we need to do multiple studies to actually understand whether it is seriously happening or not. So we did intracutaneous reactivity test of SilverGuard catheter in the rabbit model, and we found that it is not irritating. Then we have done acute systemic toxicity, and we have found that the dosage are very well appropriately managed into the ranges, and then it does not create any kind of toxic effects. Pyrogenicity analysis, and these are some typical tests that you need to do for catheters material specifically. Besides this, we also did we said, okay, fine, resistance is one of the major issues, and let's address the resistance problem in the final product as well, because we have addressed that problem pretty much to a level wherein in the silver nanoparticles were used, just silver nanoparticles. But in the product also, we have done bacterial reverse mutation studies and found out that there are not going to be any kind of damages in terms of resistance to the bacteria or any other kind of adverse effect which can create the resistance kind of issues, the skin irritation. And there are multiple studies that we have done to understand whether this catheter is safe and can be used in the patients. And finally, we have been able to successfully launch the product into the market. So we have recently received the CDSCO approvals for the antimicrobial catheters, and we have already started using it in some of the patients, and we're getting fantastic results. So as a lot of speakers, I could not speak too much about science, because now I'm more into an entrepreneurial journey. But this was a very important aspect to see whether how a simple innovation that we actually talk about on our coffee tables can actually translate into a product which is now going to have a sustainable effect, a sustainable company, and going to change multiple lives. As a scientist, this whole journey of bringing nanoegicide to SilverGuard as a product, it actually changed a lot of things inside me as a scientist, because for every scientist, it's actually a journey wherein you start thinking about incepting a product in your dreams, and then you thinking about in your mind, bringing onto paper, taking it to the lab, from the lab doing real validation studies, and then publishing a paper around it. For me, the journey actually went 2 steps ahead, because during my whole experience of entrepreneurship, I realized that technology needs to be translated into a product, and the same amount of time which you take to develop the technology is the same amount of time that you will have to spend more for converting a technology into product. It's called as product development cycle. And the product development cycle itself, no matter how small or minuscule it seems from a science point of view, it is still a very important aspect for you to see the day of your technology in the real market. And that's what I have done. So finally, I have completed half of my cycle from incepting the product to actually bringing the product into the market. Now I'm waiting for the rest of the half of the cycle to complete, wherein my product will be used onto the patient. It will make money for me, my stakeholders, for the company, and a lot of other people in the whole ecosystem, and also save the life of people. And the journey and the cycle will complete when one day a person walks in, come to me, and then, hey Milind, your catheter actually saved my father's life. And I think that is the day when I will feel that, okay, fine, my scientist to entrepreneur journey has now taken a full circle, and now I can move on to another technologies to develop. So this is the team I was blessed with, having two of my best friends around all through the time in my journey, and then moving ahead, they are standing strong besides me. Prasad, who is actually the brain behind the technology, and he's continuously doing the technology improvement, and we have been together since almost 8 to 10 years now, working, brainstorming on problems and how we can address. Anupama is an excellent microbiologist who actually gave a lot of perspective about how we can work on infection prevention practices, what needs to be done. Now we are fortunate that a lot of experienced people are joining. So Mr. Munish is actually was the head of marketing for a lot of top companies, and he have seen the medical device market since a very long time. And now fortunately he is now guiding us, supporting us, taking this product into the market, helping us reaching the product to a larger segment. And then Abhishek, a very young person from an investment background, comes in and completely transform the economics in the company, bringing out the perspective of how the company needs to be run, how it can be scalable, how the newer business model needs to be established in the company. And now I'm happy that now a scientist and entrepreneur is actually, as the scientists show, a lot of lab team members. Now I have a huge team wherein I have diversified the people from economics background, finance background, marketing background, and this is how we are taking our path ahead. I was very, very fortunate to receive multiple support from government agencies, and I would be always, I'll be always happy and indebted to Government of India for having organizations like BIRAC, which has supported us extensively, Department of Science and Technology by giving NSTBD grant, Nidhi Prayas, and multiple other grants. We thank IIM Ahmedabad, who actually helped us model our innovation into a real company, and then how to take it forward. Venture Center for giving us incubation support for a very long period of time, and Startup India, which is doing a fantastic role and activity in turning India as an IP rich entrepreneurial ecosystem, as equivalent to the Silicon Valley baby tomorrow. So I don't see the day is too far wherein India will be as mature ecosystem as Silicon Valley is today, and we have potential to, I think, go beyond them, which I think pandemic has given us a lot of frugal innovations, and how the things can be accelerated in the whole regulatory systems also. So I see India moving towards and innovation and IP driven economy very, very soon. So thank you very much. With that, I would stop here, and happy to address if there are any questions. Yes.
Thank you so much for this wonderful talk, Dr. Milind. It was really insightful to see how a product is developed from lab and how it reaches the market. I think you have nicely explained the whole cycle in a very understandable way. So thanks for your talk. There are actually a few questions. Probably we can ask. I'll ask those questions. So first is from Dr. Prasanna. He has asked this question about the hospital acquired infections data. So he asked if the global burden data is like, is it a global burden data, or is it a data that was taken in India? Like, is it just India specific?
This data, so basically we did both the analysis. We have looked at the global data and the Indian data also, and we found that there is no much of a difference. Even if infection prevention practices are very well addressed in the Western world, yet the hospital acquired infection problem percentage wise remains the same. The data remains the same on percentage wise is because again the population difference is huge. So, however, the Indian data is very discrete. So the kind of data that we can actually collect in an Indian ecosystem. See, one of the bigger problem that we are also facing in Indian ecosystem is under-reporting. Uh, Majority of, if you see the healthcare ecosystem completely in India, it's more concentrated into government hospitals and then polyclinics. The more organized corporate hospitals is a very smaller chunk of our healthcare ecosystem, even though it looks on a revenue based on a larger aspect, but majority of the patient population goes to either government hospital or smaller polyclinics. So out of pocket expenses are much higher. Now in both the situations, the reporting is usually not happening to the extent that it should have been happening. That's why the data that we see is just a tip of the iceberg, that I can literally say it's just the tip of the iceberg, because again this is the data which is usually coming from people who are doing studies in the hospital settings, in Indian hospital settings. However, I still think that the infection data would be completely different than what we have reached up to the level. And as we move into the market, we will see only the insertion of catheters in India. I'll just give you a smaller sense on the statistics of it. There are around 4.5 crore catheters inserted in India itself. So this is annual data. And if we talk about 40% of them catching infections, then it actually matches to the data that we see the number of UTIs happening in the hospital settings, because majority of the data also we have collected comes from ICU settings, wherein the people are usually bedridden for a longer period of time, either and usually catheterized. So majority of the infections are coming from that. So I think this data is pretty much in sync with the global and the Indian data. However, Indian data needs to be studied even to a larger extent more.
Great, thanks for answering that question. And there's another question that Foley catheters can actually cause irritation in the insertion site. So, so this person is asked, can it cause what will be its effect on the coating? Will the coating will still be stable, or can it get disintegrated after insertion?
So absolutely, that is why I told that we have done extensive amount of study on this aspect, whether it should be a coating and impregnation technology. And I myself personally advocate about impregnation technology, and that is where we had to actually do a lot of modification on the base technology, nano side itself, because we did not want it a coating technology. As you rightly pointed out, that the coating technologies might have problem. Insertion is only one problem, but we are talking about urine. The change in pH in urine itself can cause a lot of disruption on the coating surfaces, because we have seen in real field situation people with gram negative infections, specifically protease kind of infections, the urine pH goes up to much towards alkaline side, and that can actually hamper either the surface of the catheter per se itself, or the coating that we could actually put onto it. So address to this is, one is it is impregnation technology. So even if one layer is gone, let's say even it as small as 1 micron or 2 micron, there are silver nanoparticles in the complete catheter. So until the end of the catheter, in all the complete material, silver nanoparticles are there. So antimicrobial activity will always be there, even if there are no bacteria or pH kind of things are affecting the base material of the catheter. Second, at the site of insertion, usually this catheter is inserted with a jelly material, which provides a lot of lubrication while insertion. In few cases, when the strictures are very smaller, the chances of some sort of inflammation or some sort of irritation are always there at the site of infections. But with the jelly and lubrication, it actually decreased down to a much larger extent. In all the cases, anti-inflammatory drugs can still be given. Site specific anti-inflammatory drugs are still available. Infection needs to be addressed properly, because right now we are actually in a situation where we have to think about the usage of antibiotic very judiciously. So that's why these technologies are more relevant in these kind of situations today.
Okay, thanks a lot for that. And I do see a similar line of questions, like a lot of people have asked, and I personally also have the same question, because like you have shown like the efficacy of like this silver guard on different pathogens. So how do you think it would affect the normal microflora? Like, is it also gonna kill the normal bacteria in the urinary tract, or have you done any specificity assays that actually shows that it just kills the pathogens and not the normal microflora?
Absolutely. So that's a very, very valid and interesting question. So frankly speaking, silver will not differentiate between a good bacteria or a bad bacteria. There are few case studies wherein gut microbiota, specifically lactobacillus, are little it's little difficult to kill lactobacillus. So that's why at few concentrations of silver where we are getting an activity on a gram negative bacteria, which maybe most of the pathogens are, and we can still preserve some of the lactobacilli kind of a good microflora in the gut. But when we are talking about the catheter specifically, we could interestingly see that the urine ideally should be sterile. The midstream urine should be ideally sterile. This is now a new coming thing, that there are few planktonic cells in the urine. However, in the bladder environment, there is no role of bacteria per se, and it ideally needs to be a sterile condition inside the bladder itself. That's why, at least for my catheter product, I'm not worried too much about whether it is giving me a broad spectrum activity on both the kind of microflora, whether it is good bacteria, bad bacteria. We ideally want no bacteria on the catheter surface per se. Besides, the microflora is not in the urethral pathway. Usually it is on the urethra surface or on the penis or on the vaginal area, which is around. Usually when you're inserting a catheter, you are actually compromising the normal flow of the urine, and that's why it actually helps as an adherent surface for the normal microflora also to get entry inside, which we want to restrict at any given point of time. So that way we can address this issue.
Okay, thank you. And another question is an audience from one person from the audience has asked, what is the cost of this like silver guard? Like, what is the cost?
Yeah. So cost of the silver guard. So basically, silver guard catheter comes at around 750 rupees, and which is much more affordable of the cost value addition that it is providing. So it is cutting down on your antibiotic cost, your cost of hospitalization. One day hospitalization cost can be in the tunes of 2,000 to 5,000, and then it can go to 10,000 also at some point of time. So even if your one day of hospitalization is saved, the amount of money that you will spend on the silver guard catheter is going to be substantially lesser. That's one part. And you asked about cost economics only, right?
Yeah. So the person has asked, what is the cost of the silver guard catheter? Yeah.
Yeah. So and on the nanogicide basis also what we did, and that's the first important thing that we realized, that in the market, use of silver nanoparticle, even though we know silver nanoparticles are being there around for a very long period of time, we have not been able to commercially exploit was cost, because it's a noble metal. It's a very expensive kind of a metal. What we did essentially was to bring down the cost at the first go itself. So our manufacturing process and our patent is also on that, wherein we have actually brought the cost of silver from metal to metal, and there is no costs involved in between, and that's why we can now commercially exploit silver to a larger extent.
Okay, and there is another question they have said that silicone catheters are actually more stable, like temperature. They are also temperature stable, and also like they resist the immune attack. So they have asked if this coating can be used also on silicone-based catheter systems.
Yeah. So basically, right now the kind of catheter we are providing is kind of a replacement to silicone catheters. The use of silicone catheters started when people started addressing the problem with latex-based material. Now, one of the bigger problems is latex-based material is not uniform altogether, and you've seen the SEM image also. It's not uniform, so the chances of bacteria to gain entry and form a biofilm, it's very easy on latex-based surfaces, which we address by incorporating silver nanoparticles into it. So there are no chances of bacteria either adhering to the catheter or forming a biofilm onto it. So one of the bigger reason for which people were shifting from latex-based catheter to silicone-based catheter is what we have addressing latex catheter itself. Second is silicone is an inert material. It's a synthetic inert material. So irritation or immunogenic responses are much lesser in the case of silicone catheter. Now we are trying to now establish a concept wherein we have also seen silver being an anti-inflammatory molecule, because it's already used in wound healing gels very well, very common. I mean, we have seen multiple products on the market and they're being used extensively and giving very good results as anti-inflammatory and antibiotic as well as the same time. So we are also hoping that at some point of time, our silver nanoparticles will also, along with the catheter, give some sort of anti-inflammatory effect, which can reduce down the irritation to a much larger extent. That's one point. Second, of course, we already have the coating technology available with us, and we are going to launch a silicone catheter, which is also an antimicrobial silicone catheter with a coating of silver nanoparticles by the end of this year, definitely.
Sounds good. Thanks a lot for answering that question. And there's another question which says that like, are there any negative impacts or reviews that you have heard so far with the with this product?
So, frankly speaking, the product is being used right now. To whatever patients we have now used the product, we haven't seen any kind of adverse effect or adverse event for that matter. See, antimicrobial activity, we don't have any problem because silver is giving activity. We have done multiple studies in patients also. We are seeing the urine and we are continuously monitoring. We are recently doing a clinical trial around for 200 patients in India, and then there are multiple pilot settings. So by by around November, we will have the patient data of around 500 or 600 patients, which will be substantial enough for us to talk about the efficacy and the safety of the product. But secondly, from a science point of view, we have actually looked at the catheter and we said, let us also, as I told you, the distance between the balloon and the eye and the material itself, we have seen that the catheters which are currently manufactured in India, which are non-antimicrobial and the common latex-based catheter, are made by materials in which the latex quality is not good either, or the processing defects are very higher, because it's a cheap product, it's a commodity product. So people are not very conscious about the quality of the product itself. And that's why, even though latex is good material, and as I told you, around 4.5 crore catheters are inserted every year, that means, I mean, it's still a valuable product which is still going into the market. Yet we have tried to address and minimize the disadvantages of latex to as much as possible, whether it is striation on the surface, whether it is uniformity of the coating, which is finally done on a silicone based silicone coating. So our catheters also have a siliconized coating on the catheter, which gives it a very uniform kind of surface, which helps in easy insertion and does not also affect the antimicrobial activity of the product. Secondly, we have also worked on the tip of the catheter, because usually the tip is the portion which helps in the insertion inside the urethra. So we have also scientifically derived to the shape of the tip, which is now oval, to an extent wherein it reduces any kind of inflammation or any kind of aberrations during the insertion of the catheter, even though the jelly is already providing a lot of lubrication.
That's very interesting. Thank you. I have actually, I was very fascinated by this talk because I was also working on catheter-associated infections during my PhD. So I had this question because I know about also copper nanoparticles and gold nanoparticles, for instance, that also have antimicrobial and antibiofilm activities. So how do you actually say, what is your take on the comparison of efficacy of the silver nanoparticles to that of copper and gold nanoparticles?
Excellent question, and thank you for asking this question, because my company is completely behind silver, and then why silver is one of the biggest question. Now, first of all, when we are nanotechnologists, we can make nanoparticles of any metals. We have copper nanoparticles, also gold we can make, silver we are already making. Gold will not give you an antimicrobial activity, because per se, as such, gold does not have any kind of role in the antimicrobial activity. If there is any kind of physical activity that gold is giving you because of the size, it is a different story. But otherwise, gold will not have an antimicrobial activity. Silver and copper does. Now, the problem with copper is copper has toxicity at even smaller segments, concentrations. And second is the problem of reduction. So copper is very easily reduced, and the concentrations of silver that you work on MIC level, you will not be able to achieve with copper. So copper MICs are much higher than the silver MICs. Second important aspect that silver gives you is the easiness of working. So silver nanoparticles, like I told you, the MIC starts from 5 microgram, which is much lower concentration. And again, I'm not telling you 5 microgram with silver is a shape dependent activity. Even if it is a rod shape or a circular nanoparticle, it still gives you a good amount of antimicrobial activity, because you are getting an advantage of dual role. The ionic basis also silver gives you activities. Secondly, when I'm talking about the ionic basis activity, silver had 3 or 4 modalities of working on a bacterial cell. So one is, of course, very commonly known is blocking the respiratory enzyme. Secondly, silver has affinity towards thiol group on the surfaces, so it binds to a lot of surface membrane proteins in the bacteria. Once it gains inside the entry inside, it actually increases toxicity. So whether it is reducing environment or reactive oxygen species generating and disrupting the DNA of the bacteria. So it actually works on a multiple layers of levels and at a reduced toxicity. So with silver, you get both the advantages, and you can scale it to a much easily and on a higher level. Copper is little difficult to still scale and manufacture.
Okay, thank you. That was really convincing. There's another question that's very interesting. So they have asked, what was the time span to translate the lab to make the from making the silver nanoparticles to actually the silver nano AG site technology, basically?
Yeah. So frankly speaking, the time from lab to technology for me was 2 years, and from technology to product 3 more years. Why, I'll tell you, and I love to touch upon this point because I have seen the whole pathway of completely thinking from a brain point of view. We usually as scientists comes with multiple ideas in one day, and that's why we need to focus on one idea and then take it forward till the end, till it becomes a product. I have failed. I have failed multiple number of times, and I have failed every day almost in my entrepreneurial journey. So bringing the product from an inception stage to a technology level stage also depends on, one is the kind of scientific backing that you require, experimentation you require, and thirdly, it's real application into the field. So as strongly you can minimize the cycle, as fast you can move into the market. Now, being scientists, what we usually look into is how strongly I can convince my data into the to the people, and how strongly I can do multiple different type of experiments to prove my hypothesis. And sometimes we to prove one hypothesis, we do at least 5 or 10 sets of experiments, which are really not needed for a idea to actually bring to a technology and to a product. 1 or 2 kind of different experiments from different angles are enough for translating. So we can reduce down the time by not thinking too much about what more we can do, of rather bringing it out into the level that it is. And you can always iterate when you bring something and give it to people for testing. They can give you feedback, and then you can modify your product. It's called as a common concept is called as an alpha version and a beta version. So you usually launch an alpha version, get the feedback, and then you launch an improvised beta version into the market. So this is up to the technology, but from a technology to productization is a very straight, clean pathway. If you're into medical field, the pathway completely depends on the regulatory guidance that you get. Of course, for my biocompatibility analysis, for regulatory analysis, it was mandatory only 3 or 4 studies, but yet I did around 7 to 8 studies. And of course, if I would have been a normal entrepreneur, I would have done 3 studies, but because of the scientists inside me, I did 4 more studies in addition to what. So that increased the timeline. But it all depends on the product. So the first timeline you can reduce down from an idea to technology, but from technology and product is not in your hand. It is in the hand of regulators, because there's a lot of processes for product approval and it takes time.
Totally agree. I think that was a wonderful closing note, because I'm sure as scientists we need to also reduce the timeline of product development. That's kind of very important to be translated to the market. So I think thanks a lot for your wonderful insights, and also letting us know about your story on Silverguard. That was really interesting. And thanks a lot for patiently answering all the questions. And and with this, I would like to close this session. And thanks everybody once again. Yeah.
Thanks, Dr. Shrikant. In the comment in both on YouTube and this, I will just put in my number, email ID, and social media handle. Feel free to get in touch with me. I'm always happy to interact with people. Thank you very much.
Thank you so much, Dr. Milind, and thank you, Dr. Shrikant, for chairing this session. It was quite interesting. From an entrepreneur perspective, it was, I think our audience could have gained a lot of insights. Thank you so much. Next, I would like to invite Dr. Deepita from Shastra to introduce our next speaker for the session. Over to you, ma'am.
Yeah. Thank you, Sahana. Good afternoon, everybody. So we have been through some really exhilarating talks which are happening since morning. So it's an honor to introduce next speaker, Dr. Asmita Prabhune. Dr. Asmita Prabhune is the co-founder and director of Green Pyramid Biotech Private Limited, a tech startup funded by BIRAC DT, awarded by Swissnex. Garnering over 34 years of research experience, Dr. Asmita's research interests include microbial technology, microbial biotransformations, sense imaging, basic and applied aspects, biosynthesis of nanoparticles, nanostructures, and their applications. Dr. Prabhune has obtained her MSc and PhD degrees in microbiology from the University of Pune. Dr. Prabhune has several postdoctoral experiences encompassing working as a research associate at the University of Hull and University of Nottingham. She was a visiting scientist at the University of York, UK. She was DST grad visiting scientist and principal investigator at the University of Darmstadt, Germany. From 1982 to 2017, she worked at the CSIR National Chemical Laboratory, where she introduced the potential of biosurfactant cephorolipid. Her path-breaking work has earned her membership in several esteemed organizations such as the Society of Biological Chemists, Biotechnology Research Society of India, Association of Microbiologists India, British Scholars in India, Indian Women Scientists Association, and many more. She has around 140 publications across esteemed seminal journals in the field of her research. Dr. Prabhune has an overwhelming 28 patents to her credit, including world, US, and Indian patents. Her credentials and achievements are endless, and a 3 to 4 minutes introductory session is insufficient to describe Dr. Asmita and her magnificent body of work. With great privilege and honor, I request you, ma'am, to talk us through your spectacular work. Over to you, ma'am.
Thank you. Thank you so much, Dr. Guha, for such a wonderful introduction. So without wasting time, I'll immediately go to the presentation. Is that okay?
Yeah, sure, ma'am.
Yeah, I will. So is it visible now?
Not yet, ma'am. I think it's just coming up.
Can you see it?
Not yet, ma'am.
I have already said to share the screen. So
there might be a small lag for a couple of seconds, maybe. Yeah, it is now visible now.
Yeah. Yes. Yeah. Okay, so I'll start with my journey and how I came up with my startup. And it never happens overnight, naturally, because you have to do loads of work, and then you have to gather interdisciplinary the knowledge, and then only you come into something which can be a tech transfer, or it can be a spin-off of whatever work you have done. I find myself very lucky being in NCL because I got a chance to work on interdisciplinary sciences. So being a microbiologist, I had expertise in microbiology, but I did my PhD in enzymes. So I could get knowledge of enzymes and the accuracy of enzymes and the fast reactions of enzymes, which I was not aware of. Then I could do some work in University of York, molecular biology, site-directed mutagenesis, again on the enzymes. And then after I finished my PhD, I started working on biotransformation, especially for the biosurfactants. So the part of my work I will just explain to you. So sophrolipids are biosurfactants, and for me, 20 years back, a sophrolipid was very new name to me, new concept, new molecule. I never had heard about it. But then when I was in University of Hull, there I got chance to work on these molecules, but not for as a biosurfactant, but there I was working for conversion of arachidonic acid into eicosanoids. These eicosanoids are very important in our body systems, and they control our blood pressure. There are so many activities, and to synthesize the eicosanoids.
Dr. Prabhudev, sorry to interrupt you. I think, ma'am, yours is not in the presentation mode. You have to, I mean, because we can still see just the first slide.
Can you see it now?
Yeah, now it is perfect, ma'am.
Yeah.
Thank you.
Yeah. So the eicosanoids are the molecules which control the blood pressure. They reduce, they increase. I mean, they have got a vasoconstrictor activity, depends on the concentration. So we were using this molecule just for conversion of eicosanoids, 20 hydroxy eicosanoid and 19 hydroxy, because this organism which I'm using for conversion of sugar and fatty acid into sulfolipid, it hydroxylates at position omega and omega -1. So we were just pinning that, and we were working around it. When I came back to India, I realized that there are so many aspects. It's not there. Yeah. So we know what are the surfactants. They are just surface active agents. And what is the current scenario of the surfactants? So till I come back from my postdoc, I realized that there are so many synthetic surfactants are in the market. Of course, they are still there, and they have problem. Surfactants, they remain in the sewage for longer time. There is eutrophication, and naturally it is disturbing the whole ecosystem of water. So I was thinking, why can't I take this molecule as just a surfactant, because it has got some very beneficial properties. First of all, it is a biosurfactant, so it is a biodegradable, biocompatible, non-toxic. It is FDA approved also. And the wonderful thing is that the organism secretes the biosurfactant in the medium. So the purification is even easier for that. So, and another important thing is that it is possible to tune the production. It is like a tailor-made. Whatever fatty acid we give to this organism, it converts that fatty acid into sophrolipid. I mean, glycolipid, I can say, because it's a general term, glycolipid. So that time I tried arachidonic acid, which is C20 carbon molecule. But usually this candida, which I have used, it prefers C16 to C18 molecules. And when you add in the medium, what this organism needs, the primary carbon source is glucose, and the secondary carbon source is any fatty acid. So the surface tension is increases. And to reduce the surface tension, this organism produces the surfactant to reduce the surface tension. And there are some reports that sometimes it produces the surfactant as a storing food material. So these are some physiological role of this organism for making this biosurfactant. Naturally, we are making use of this biosurfactant because it is completely biodegradable, and it has got wonderful properties, which I'm going to tell you now in my following slides. In my group, we have worked on oleic acid, linolenic, linoleic. We have used mixed acids also. We have used different lauryl alcohol as a hydrophobic source. We have used, in fact, terpenes and terpenoids also for synthesis of production of this glycolipid. And we have got a different wonderful results, and in almost all sectors of biology. So to go ahead, what are the sophrolipids? So these are amphiphilic surface active molecules, consist of dimeric sugar. If you see this dimeric sugar, it has got unusual beta-1,2 linkage. And this is the ether linkage, which is attached to the fatty acid. And sometimes it is in the lactonic form. In fact, we get both the congeners. One is lactonic, one is acidic. The acidic is soluble in a water, and lactonic is not soluble water. So in the crude sophrolipid, whichever we get, it is always like that. The acid keeps helping the lactonic because the lactonic has got wonderful properties, antimicrobial. It has got many antiviral property also. It has already established in, and this acidic form is, it has got a surface tension reduction property. So it works like a soap. So both together they work fantastically well. And here the carbon chain, if you see it, we can keep on changing it. Here we have used oleic acid, but here it can be arachidonic acid, it can be linoleic acid, it can be linolenic acid. And as you keep on changing the fatty acids, we are getting different properties. Right now, my students are working on myrastic acid. She's getting so much fantastic activity that the CMC value is much, much lower than even sodium dodecyl sulfate, which is supposed to be very high surfactant in nature. So different fatty acids, they behave differently, and depends on the double bond also, because in oleic acid there is only one double bond. In case of linoleic 2, linolenic 3, linoleic 2, so it keeps giving different properties. That is also I'm going to discuss in my next slides. This is a short pathway of sophrolipid biosynthesis. The main the person in this synthesis is cytochrome P450. To be very honest, it's a 4A. Cytochrome P450 4A monooxygenases. It takes oxygen from the air and it puts that oxygen to the fatty acid, and the ether linkage and the sugar moiety comes into the picture. Here are the two sugar moieties which get attached to this, and these are UHP sugar oxidases which come into the picture, and at the end we get different two types of sophrolipids. Another important thing in this is that we get almost 7 to 8 congeners, in a sense that since there are two sugar moieties, there are sometimes that monooxylated, monoacetylated, diacetylated, or they are just a plain molecule. So for everything there are changes. So usually for oleic acid, we have got 8 to minimum 8 congeners for and because of this congeners we have got a problem, in a sense that the purity and of this sophrolipid is very difficult. We have achieved some purity by using different cultural condition, that is also possible, but even then the acetylation is very difficult to be controlled. Now they are surfactant in nature. Naturally they are called in fact Bola amplifiers, because in sophrolipid, if you see, there is the in acidic form, there is a COH group also and sugar group also. So it has got two sugar groups. That's why there is a bending, and that bending is seen in self assemblies, and self assemblies of other biosurfactants are different. But in case of sopholipids, we get ribbon-like structures. Spherical micelles are there, but very nice ribbon-like structures I've seen. And these ribbons are again dependent on the double bond or triple bond of the fatty acid which we are using for synthesizing the sophrolipids. So this is also very important for making scaffolds, which I will come in my next slide. Different scaffolds we have made with the help of these sophrolipids. First, I thought that since it is a biosurfactant, so I should do something to replace the synthetic surfactant, because in my first slide I showed that there are problems of the synthetic surfactants, and some point of the other we have to remove this synthetic, or at least minimize this the dose of this synthetic surfactant. So what we did, that we took 4 typical stains like turmeric, coffee, poster color, and oil color. We have used saran wraps. We have used different texture of clothing, cotton, polyester, etc. And we have stained those with these stains. And in my next slide, I'll show you that we have got almost same results the way the synthetic surfactant can produce. Another important thing is that we have used pongamia and jatropha seeds, because these seeds, they fall and they just vanish. Actually, it is a renewable source from where we can use this oil, because there are certain reports that even the the oil which is waste oil from frying or deep frying, whatever is remaining, even that oil also can be converted into sophrolipid. So here we have just collected, my students, they are very enthusiastic. They collected these seeds and then crush, oil has been extracted, and the same oil is converted into a sophrolipid. And the sophrolipid showed fantastic activity as a surfactant. So after that we realized that this is not the application, because there were certain reports readily available on the Google, that some people have used as a biostabilizer, some people have used for increasing bioavailability, etc. And there is a report from chain that it shows apoptosis for human liver cancer. So we thought that why can't we also try in different aspects. So first of all, what we did with one of my student, Ruchira, we started using, at that time we were working on quorum sensing and quorum quenching, and we know that essential oils, they are wonderful quorum quenchers, but the problem is that they are hydrophobic in nature. So unless and until we are making them hydrophilic or making available, bioavailable, they will not be able to show activity. So what we did, that we took the essential oils and we mixed it with sophrolipid, and the activity was multifold increase. So that was Ruchira's work. Um, Another work was done by Kasturi, one of my students. What she did, that she made sophrolipid using linolenic acid, and she could see something very unusual and very different, that the differentiation of glioma cell line was seen. If you see this figure, maybe you can see on my research kit, the picture can be seen clearly. So that was very unusual for us. And then the work is, I mean, some other students are working on that. So in this similar lines, we started working. And then in this slide, you can see this. You can see the glioma cell lines differentiation has started. Then being a surfactant, it is a Bola amplified, and it is amphiphilic in nature, because it is water into oil and oil into water. In both the condition, it works fantastically well. So what we did, that we took sophrolipid, we mixed it with antibiotics, and we tried to reduce the antibiotic concentration, and we realized that almost 1/10 of antibiotic is good enough for showing microbial death. So LD50 was below much level, and sophrolipid is FDA approved. So this is another something good finding that which can be taken forward. In another work, what we did, that we have used lauryl alcohol in pathway, which I have showed earlier, that if you give the activated molecule, because fatty acid, it has no active group, lauryl alcohol OH group is there, so we could get large amount of sophrolipid, which is lauryl alcohol sophrolipid, and then it has fantastic antimicrobial property against Staphylococcus aureus and Bacillus subtilis, and which we have 1 minute, and which we have shown using atomic force images also. And similar work was done with some other fatty acids also. One of my students, he was very enthusiastic, and then he wanted to work on sophrolipid, but in a different way. So what he did, that we have a laser machine in National Chemical Laboratory. So he used these molecules and he laser irradiated these molecules. And to our wonder, what we realized, that we are getting perfect round structures which are fluorescent in nature. First, we used to see under the microscope. We thought that we should put some chromophore there, so that we'll be able to use this as a drug delivery system, and it should have a fluorescence. But then we were lucky that there was a fluorescence. But again, that raised one more question, that why this fluorescence is coming, why this photoluminescence has increased. Then we realized that when these two fatty acids, oleic acid, it has got a single double one. So there is a pi-pi interaction, and because of the pi-pi interaction, there is a fluorescence which could see, and you can see the photoluminescence which has increased a lot. Another thing is that in this, it is a completely porous structure, but even then it is perfectly sphere. Here you can see the fimbrial-like structure, and this porous structure, it has taken Fe3O4 nanoparticles. Fe3O4 nanoparticles we synthesized chemically, and we embedded these nanoparticles, and we could see it that it has started showing the magnetic property. Naturally, they are magnetic in nature. So the whole molecule, which can be, this is with sophrolipid, I mean the laser irradiated sophrolipid structures. And here, when we use the magnet, the whole structure is coming here. So this is another interesting property we could check in. And then when we did the ion beam milling, we could see the inside structure of these micro structures. These are nano to micro. I could get that 300 nanometer to almost 1 micrometer structures, and this could be wonderful as a maybe as a theranostic drug system, or maybe it can be used just as a tagging system for the drug molecules. So this is also one. Some work we have done on sophrolipids. Then we realized that the sophrolipid is very mild molecule, and we know that the quantum dots are very toxic, though they are very small. Their penetration power is fantastically well, but they are very toxic. So what we did that we coated these quantum dots with the sophrolipid, and we realized that the toxicity has gone down, and we could use it because the quantum dot, as it is, they are fluorescent in nature, so they can be used as a theranostic application. So on every sector, we wanted to use sophrolipid, because everywhere, wherever we used to go, we used to find some or the other beneficial property of the sophrolipid. So in our group, we started calling it as a wonder molecule. So here we have used acetyl alcohol for production of sophrolipid. Again, the acetyl alcohol tagged with 2 sugar molecules. Sometimes it has happened that instead of 2 glucose molecules, we could see only 1 glycomoiety, which is attached to the fatty acid. But at the end, it was showing fantastic surface active property. It showed, in this case, acetyl alcohol, we could see fantastic antimicrobial property, and which was again showing oil displacement assay. All other analytical parameters we checked for the presence of acetyl alcohol, for presence of glucose, which is indication of that it has converted into sophrolipid. This is very interesting work which we did, and still the work is, I mean, we have gone ahead with this work. One of my friend was working on silk and silk fibroin, and silk fibroin, it is a body human compatible thing. So we wanted to check whether the gelling can be done, because it is somewhere the report was there that the gelling can be done with the surfactants, but there was no report with the biosurfactants. And we have used this biosurfactant sophrolipid, and otherwise the silk fibroin takes almost 24 hours to gel to give a typical scaffold, but in case of sophrolipid, it gives 60 minutes. And the whole thing also can be expedited by adding the concentration. When we did the scaffolds, I mean, same and time we did with the scaffolds, we realized that they're depending on the sophrolipid concentration, they are helping fibroblasts to go faster. So here you can see we have used L929 fibroblast. As you increase the concentration of sophrolipid, the fibroblasts started growing faster and better. So this was a clue for us, and that's why we started working on wound healing property. Same, again, we did, I mean, very briefly, I'll go through this. Here we have used the cipladine, which is a natural molecule, a natural ointment, which is used for wound healing. Then we have used sericin and sophrolipid. So sericin was another control we kept, and sericin and sophrolipid we kept. And this was the incision model they have used. And to our surprise, what we realized that sophrolipid, not only it helps the fibroblasts, but there was no scar. This is, we could see after the day, after 10th day. So this work also, some of the students are taking it forward. So the sophrolipid gave some indication about the fibroblast activity, and it was perfectly seen in one of our experiments also. Then this is very interesting work we did, that we know that the curcumin is not a water soluble. Whatever curcumin we take, within 15 to 20 minutes, or maybe when it goes into the liver, it gets just degraded and it has no activity at all. And as the whole molecule of curcumin, it has got a tremendous activity, tremendous antimicrobial. There's so many other anti-rheumatic, so many other activities are there, but we are not exploiting that because it is not bioavailable. So since we were working on surfactant, we thought that why can't we try sophrolipid? So I checked we just took the sophrolipid solution, it is water soluble, and added curcumin into that. And I just kept it on the table, and I thought that maybe tomorrow we can come and see if it is going into the solution. To our surprise, it was going into the solution immediately. And it was a very homogeneous preparation. And when we see the SEM and TEM images, we realize that there is a nanomerization of curcumin is taking place. As we know that as it is, curcumin is fluorescent in nature, the fluorescence has increased many fold when it is converted into nano form. Then we did animal studies as well. As we know that within 30 minutes maximum, it gets degraded. So what we did, we fed group of rats with curcumin, which is solubilized in sophrolipid, and curcumin just like that. And what we realized, we took the plasma samples from rat blood, and then we injected to HPTLC and other fine analytical instruments. And what we realized that even on the HPLC, we realized that the curcumin is intact even after 2.5 hours. So it was like we could say confidently that this is definitely remaining as it is, and then the property of this sophrolipid can be availed, property of the curcumin can be availed fantastically. After this work, what I realized that those days there used to be a show on the TV, Satyameva Jayate, and then one of the program was, it was showing that there is a full trains with all infected, because of the pesticides, the all farmers all are suffering from cancer. The full train is going for treatment. And the whole, there were 2 episodes, and they were showing that these pesticides are so bad and how their problems are there. That's why I thought that why can't I check this molecule, because it's surfactant. I know the pesticides are hydrophobic in nature, and that's why I started working on this molecule. And to our surprise, that it started removing the pesticides from plants and vegetables. We have used different fruits and different vegetables, and here exactly my startup was taking birth. That something is happening, but I was going into the detail. I wanted to work more. Unless and until you establish, you get the result consistent, you can't go ahead. So we worked a lot on that, and we realized that 90 to 99% pesticides have been removed from the fruits and vegetables, and we could get almost the fruits and vegetables which are grown organically. So this is what we eat. This is the problem. There are 2 major concerns of our food system in a green revolution. They are not, if they are not safe to breathe, how can they will be safe to eat? So these are the harmful, I don't have to say, tell you specifically about the pesticides, what pesticides are, etc. But these are the side problems of the pesticides. These are the harmful effects of the pesticides. And another, this is very important figure which I would like to show. These are every year they give the Environmental Working Group from USA, they give this dirty dozens. And every time all these 4, like pear is there, nectarines are there, strawberries are full loaded with pesticides, kale and spinach, and they are always on the top 10, and they are full loaded with pesticides. And just washing never removes, the way just adding water never solubilizes curcumin. Like that, just adding water or just washing with water never solubilizes the curcumin, never solubilizes the pesticides. That is the problem. So we started making the formulation which has got Naturally, our hero molecule is there. And then we, by trial and error, we made several formulations, and it is as usual, as I mean expected, it started giving fantastic results. So this is the product which we are selling on Amazon, B2B, everywhere. This is called SoFab. We always say it is a fabulous product. Sorry, 1 minute. One person has come to take my swab because tomorrow I'm leaving to Germany, and then my swab test has to be done. So he's sitting. So sorry for that disturbance. So
No problem.
this is the product which I'm talking about. This is the spin-off of my one of the patent, which shows that not only it removes the pesticides, not only it removes it kills the microorganism. In my previous slides, you must have shown that the sopholipid has the fantastic property of killing microorganisms. This lactonic form, it kills the microorganisms. And another thing is that being a surfactant in nature, it reduces the contact angle, and naturally it removes the pesticide from the fruits and vegetables. So I'm making use of that, and this formulation has come up. So what is the technology of that? So it, as I said, it lowers the surface tension of water from 72 millinewtons to 25. In case of myristic acid, it goes even lower than that. Then it reduces the degree of angle, as I said. It is amphiphilic in nature. It is a biosurfactant, FDA approved. Another thing is that it is anti-cancerous in nature. It is less harmful to the body. In fact, it is digestible. 5 ml per kg weight is also acceptable if it's taken in the oral route. Then it inhibits and removes the pathogens and increases the shelf life, because naturally when the microorganisms from the surface of this fruits and vegetables are removed, the shelf life is increased. So that is our bonus which we get using this product. So these are the value proposition. Since it's a surfactant in nature, it is anti-corona also, because the spikes and the lipid layer which is on the coronavirus can be literally collapses when it is added with the surfactants. This work is I'm doing with Bratislava University of Virology, with one of my friend, and they have got fantastic results using different types of viruses which has got a lipid layer on their coat. Then it saves water because it is a completely biodegradable, and it is completely acceptable. We don't have to rewash once we wash with this formulation. It increases shelf life, already told, and removes the pesticides and gives almost as if the fruits and vegetables are grown organically. For going into the market, we needed some certification because some people have to believe us. So we got a FSI, we got a green set. There are many products coming up, especially in the COVID. Many products came up in the market for washing, but none of the, I mean, I can say proudly that none of the product has got green cert, or I mean, they must be having FSI, but we have got a green cert assuring that it is completely organic in nature and completely biodegradable. So these are some results our customers they have sent, some results we also did it. So you can see, even I have not seen such grapes, so clean and crystal clear grapes before my formulation was in the market. So when we wash it, we could get this, and we had done studies using through Enable accredited labs, and they also gave us the results that 99% pesticides have been removed. And you can see how much dust and dirt is there when we wash with this. In fact, nowadays they put the cosmetic colors to the vegetables and fruits for capsicum and even for other vegetables. So those cosmetic colors also can be removed using this formulation. This is extend the shelf life. This is my first experiment which I did when I thought of doing something for removing pesticides and increasing the shelf life. After that, I kept on getting fantastic results. This is after 35 days. If you see, this is a tap water wash. These are without wash, and these are my sample. I mean, my formulation wash. Even after 35 days, the tomatoes were just like that. In fact, I have got a recent record of 45 days, and they were as it is. There was no problem with that. And the shelf life was increased fantastically well. This is also extending of shelf life after 14 days. We took the mixture of vegetables together and we could see such a good results. This is the log reduction of we used 4 different index organisms: Salmonella typhimurium, E. coli, Irvinia, chrysanthemi, which are plant pathogens, and Xanthomonas citri. So we could see that there is a log reduction of microorganism. Within few seconds, they kill the microorganisms, and there is a pesticide removal also can be seen. In fact, we got a catalog also, complete catalog from different labs, Vimta labs from Radbox, and they have given the names also, name of the pesticides also, which has shown that they have been removed from the fruits and vegetables. Actually, this is actually a film where they have taken the time lapse photography, and we have seen that even after 45 days, the tomatoes which has washed with the formulation was as it is, and other tomatoes were got disintegrated. So, but now it is not running. I tried several times, but I was not successful. Sorry for that. So this is something about my spin-off, how I started with, and how I now working as a director and co-founder of this my sophrolipid work. So sophrolipid is always will be my hero. In fact, recently we have made sophrolipids using lauric acid, and we realized that it is giving better scaffolds. Not even 60 minutes, within 30 minutes we could get the scaffolds of silk fibroin. And there are other nanotechnology also I worked on. In sophrolipid, we could first time make the cobalt nanoparticles. We have made silver nanoparticles, and even we have made a silica nanoparticles, and that is totally different. But I couldn't put all the things whatever I had done for last almost 30 years. So this is just the brief that how I ended up with my startup, and how now the current situation is. So thank you so much. I'm sorry I went very briefly, but then you can understand. Yes.
Thank you so much, Dr. Prabhuna, for this amazing talk about your hero molecule. In fact, what I get from your talk, which is definitely brief, given the amount of work you've already put into, that sophrolipid is definitely a magic molecule.
Yes, it is. Yeah.
Yeah, I mean, starting from, applications in cancer to increasing shelf life. I mean, you look at any field, I think your compound finds a application there.
Yeah, it goes ahead and it just finds the application. Because recently with Dr. Prasanna, we published one paper. There also we have done some magic with that molecule. You can, it is published in Collides, and you must have seen that paper. So it is really wonder molecule, and then every day we are finding one wonder molecule. Recently, one of my students, she's using sophrolipid for calcium nanoparticles, and she's getting exact calcium nanoparticles which are required for bone repair and all that. I'm not expert in that, but then just day before yesterday, she showed me the result, and Dr. Anuya, they're really fantastic results. I mean, you just think of it and then it does the work, something like that.
Dr. Asmita, we'll just quickly take a couple of questions which are already there. I think there's a question from Dr. Shrikant, and he asks the most pertinent question of the time: has this surfactant been already tested on coronavirus?
Yes, it has been tested because it is a biosurfactant. In fact, recently I got approval from DST for going for no alcohol hand sanitizer. So I'm still working on that. In fact, I got the result, but I stuck with the certification. Unless and until I have a certification, and all these labs are very busy with corona testing, so there is a delay in getting the certification. But it is a surfactant. It has got a better activity than that of the SDS. So it has to show, because it acts on the lipid and it just removes the lipid. And the coronavirus is, it just collapses. It works on that, yes. In one of my paper I have worked on, we made, gold nano clusters of 3 nanometer. And we have put this sophrolipid as a linker, and then we have put the AHL molecule on that. you all are from quorum sensing, I mean, that area, so you can understand what is the AHL molecule is. So that AHL molecule, and we could detect, the difference between the staph, because staph doesn't have AHL molecule receptor and E. coli has. So we were thinking that that paper also has accepted, I mean, published in SCS. So that work also, if someone takes ahead, then it can be like, tagged with the nano clusters, which is dark red in color, and then it can go and it can detect where the virus is. Actually, we were thinking, but there's so much work ahead in front of me. So these ideas are just keeping in my mind. I'm not taking them ahead, but that definitely is going to, because it has got the surfactant property, it has got the tag which is colorful, and it has got the HL molecule. So it is not only it is killing the microorganism like E. coli, but it can kill the virus also. Little bit tuning of this molecule can be useful for that.
Yeah, so ma'am, we all will be looking forward to you, for the current pandemic situation which is going, your hero or your magic molecule is required.
Being just because it is a surfactant, that's all.
Yeah, true. I think the next question which we have here is that have there been traces of the surfactant in the tomato itself after the number of days?
No, this surfactant is biodegradable, completely biodegradable. What happened, why the shelf life reduces? Because there is a microorganisms, they secrete enzymes, and then they start penetrating. And what happens when we coat, there is a thin coat which gets because of, and there are certain other molecules are there, which is again organic in nature. They, and with the sophrolipid, it gets coated, and then that coating helps that microorganism for another attack of microorganism. Because from farm to table, there are almost 20 sets of hands, they handle our fruits or vegetables. So that they carry microorganisms and other things also. So that is taken care using by this formulation.
Yeah. So yes, ma'am. So in addition to this, I think there's a very important question from participant Aparna. So she asks that, does extending the shelf life have any impact on the nutritional value of the vegetable or the fruit?
No, we did it on the pomegranate, and then we checked certain concentration of these enzymes. Basically, the invertase is there in fruits and vegetables, which converts sucrose into glucose and gives more sweetness. So we have checked the different enzyme levels, and other parameters also we have checked with NABL accredited labs. So whatever the concentration, there is no loss of the property, though there is no loss of taste. And another important thing is that my formulation is it is tasteless, odorless, and it is not harsh. There is no acid, there is no alcohol, there is no other harsh chemicals used here. So it is just preserving, as good as it's preserving it in the fridge, but it is preserving in the room temperature.
Okay, that's great. So now one more question which has come our way is that if the sophorolipids can be tailor-made, does the physical and chemical properties change depending on the parent molecule taken to form the sophorilipid?
Yes. As I said, that every molecule changes its property. When you use arachidonic acid, of course the production is very less, because this candida which I use for production of this, it is not used to taking C20 molecules, so it makes it, but the amount is very less. But the sophrolipid, it becomes vasoactive, which is useful in medical field. In olic, I mean, when I use olic acid, it is just surfactant property and this typical properties like antiviral and like a surfactant associated properties that is there.
Okay, yeah. So ma'am, we'll wait for a few more questions. In the meantime, I'm so enthusiastic about this magic molecule. I have a question for you, ma'am. Like it's out of curiosity. Since you showed me a slide, where there was this multi-pronged applications of this particular molecule, which showed that it could induce apoptosis in
it does, but I want someone to take it forward. I feel bad because my student, she did PhD and she went to Boston. So what happens, they work only for that period, and then there is no one to take forward, and the next group of students, they come, they are interested in some other work, so we have to create another domain for them, and they will start working on that. They might not be interested in that. So I'll be happy if somebody would like to grow on. I can provide them the molecules. The way I can send some molecules to Dr. Prasanna.
Yeah, that would be wonderful. Because now, why I asked you this question is because if something is killing a cancer cell, it's very harsh. But on the other hand, I'm saying, like, when I use it as sort of something to increase the shelf life, it's all taking care of everything. So it has sort of a dichotomy, which is very wonderful. Like if a molecule has all this.
Yeah. If you want to know more details about it, my paper has been published in PLOS. Exactly on the cervical cancer cell line, cervical cancer. I have used ethyl alcohol for production of sophrolipid, and all detail, everything has been written in that.
Okay. Yeah, definitely. I'll read it now. And ma'am, perhaps one question as a science enthusiast is like, ma'am, how do you think is it difficult to become an entrepreneur while being a scientist? What are the challenges?
Actually there are difficulties, but there are certain things which are solved when you are a scientist, because unless and until your knowledge, your fundamentals are clear, you cannot go for entrepreneurship, because when you get stuck at that point, then you can't come back. You need to have, suppose anything goes wrong, you should be able to understand what exactly is going on. And another thing is, I always tell my students that try to work interdisciplinary, because it is like working on the same field again and again. Sometimes it is stereotype thinking. You have to know that if something is happening in physics, how you can apply in biology, or biology into nano into. I was lucky that I could work on all sorts of fields, and then maybe my horizon was, it was like bigger. That yes, I know that why it is not happening, because maybe this is the problem. So that is there. And difficulties are, when I entered into entrepreneurship, the problem was I was not aware of the all commerce things, how to the audit, and the language they use, the deep pockets. And I was almost, it took me 1 year to understand what exactly they're talking, the pitching and all. So when the students are young, they there should be one course which is for just for entrepreneurship, like what are the things, what are the compliances they require, what are the how to pitch, other things should be there. So that is the only thing I always, I had a difficulty when I was going into one field into second field.
Yes, ma'am. So, and you definitely paved the path for all of us to feel that, although nothing can be achieved overnight, like you said in your opening statement, but the same thing is nothing is impossible either.
It is impossible,
So
yes.
I think one last question. Yeah. So I just one last question, ma'am, I will take from another student. I mean, Aparna, she only continues that is soporolipid used in combination with pesticides?
Yes, they can be used. In fact, we had decided to go for one project where only pesticides they are taking huge. They are hydrophobic in nature, because they should remain on the plants during rainy season, windy season. So that is the requirement to be required property of pesticides. So what I thought, that reduce the pesticide concentration, make it more bioavailable, so you will naturally need less. And in fact, the sophrolipid itself has got a anti pest activity also. So that, but in really short life, you have so many things to do, but it's difficult. So that is also open. I have in fact ready a project written also with me, but then I'm running short of hands now.
Okay, yeah, that was a wonderful talk, and so patiently you answered all our questions, Dr. Pravune. So it has been really wonderful session, not only listening to your wonder molecule, but listening to a wonderful person as you. So we wish you all the best, and thank you so much for gracing us with your wonderful body of work. Thank you.
Thank you so much.
Thank you, Dr. Asmita and Dr. Deepika for the session. So we will take a short break till 4:30, and then we'll get back with our next two talks for the day. So we'll meet at 4:30. Thank you once again for joining us. Good evening, everyone. Welcome back for the session. So I now invite Dr. Deepika to invite our next guest and chair the session. Over to you, ma'am.
Thank you, Sahana. So it's our privilege to welcome the next speaker, Mrs. Geetanjali Radhakrishnan. Mrs. Geetanjali Radhakrishnan is the CEO and MD of Aduvo Diagnostics Private Limited, a medical device startup that develops point-of-care diagnostic devices and solutions in healthcare. Geetanjali completed her BTech in Bioengineering from Shastra University and earned her MBA degree in Quality Management from BITS Pilani. She was elected as the Fellow for Entrepreneurship and Advanced Leadership at the esteemed Judge Business School, Cambridge. For 4 years, she worked with TCS as a software developer, following which she founded Aduvo Diagnostics. Her work has been widely acknowledged as testified by the long list of awards that she has received. To name a few, she featured as one of the Forbes 2019 WPAR Trailblazers. She received the TIE BIRAC Woman Entrepreneur Award in the same year. In 2020, she was the recipient of the CCMP NBEC 2020 IQVIA Anthem Bioscience Startup Award. She holds 1 granted patent, 4 pending, and 3 design patents. Mrs. Geetanjali's vision of making high-end diagnostics easily accessible to healthcare settings in India makes her a role model for all scientific enthusiasts and budding entrepreneurs. So with great pride and privilege, I request Geetanjali Radhakrishnan to deliver the lecture. Welcome.
Yeah, thank you, Dr. Deepika, for such warm welcome, and happy to be, actually presenting to Shastra audience as well. So thanks again. So let me just share my screen and then I'll start with my talk. Okay, 1 second. Yeah, I hope my screen is visible.
Yes, it is visible.
Okay, great. Right. So again, I'm Geetanjali, founder of Aduvo Diagnostics. So Aduvo means to help or to aid people, and it's a Latin word. So soon after my college, I had begun my journey as a software developer at TCS. I was a bioengineer by background. And back then, there were very few opportunities that were present in India, specifically for an undergrad student. So which is why after 4 years, I thought I need to get back to my core area of bioengineering. And that's when the startup moment was picking up, and there were a lot of support that we were receiving from Government of India as well, from BIRAC. So which is what led me start this company. And the main focus which made me into work in particularly with respect to label-free diagnostics is because I had visited a hospital in Pune in person, met a doctor named Ayush Gupta, who's a dermatologist. And then we had gone through the wards in his unit, where he caters to close to 200 skin and soft tissue infection patients. And what really moved me was many of these patients cannot afford a simple culture test, and they would have been treated with a broad spectrum antibiotic in the point of care setting where they are actually present. And they would come to Dr. Ayush Gupta at a very advanced stage. And what would have happened is it's a misdiagnostics and prescription of generic antibiotic, which would have actually changed the whole microbiome pattern, making the treatment very, very tough and lead to multiple amputations. So this is what really shook me, that I really wanted to do something in the diagnostic space specific to infection. And our company's vision is to make rapid label-free diagnostics to improve the quality of healthcare. So the main focus area over here, because to be in line with the quorum sensing group as well, is how to exactly add a point of care device that can aid in rapid analysis of these microbiome that are present on wound, and how we went about developing the whole thing from scratch. So my team, I'm a bioengineer, as I had mentioned. My co-founder, Dr. Bala Pesala, who had just joined the company 4 months back. He was our mentor all these years and just joined. He's a PhD from optoelectronics in UC Berkeley and an IIT Madras alumni. We have a strong market and sales team because we have just commercialized our device. And we have Mukund, who leads our operations. He's also from Shastra and MBA from Deacon's University. And John King is our lead product developer who takes the device from the start point till the end in terms of embedded systems and machine learning. We have a strong mentoring network where Dr. Sriram Raghavan is a serial entrepreneur. He has led 6 companies and his insights were very useful throughout the journey of my startup. We have a strong clinical advisory board member as well. So Dr. Rajesh Keswan is one of the very prominent Diabetic Food Society executive chairman who's on our board. And Dr. Arun Venkatesan, he's the CEO of Wilgrow, which is a social impact startup in USA. So he's part of our advisory board as well. So as I had mentioned, currently wound is a very broad range which covers from burn incidences to skin and soft tissue infections which are dealt by a dermatologist to diabetic foot ulcers to surgical site infections. And what is very, very important is 600 million people around the globe suffer from wound, which are of any of these categories. And infection is one of the major incident which causes delay in wound healing. So once a doctor can combat infection, he can treat the wound very easily. And what we have seen is every 30 seconds, there is one foot amputated anywhere in the world. And this is what is very important. What is core to us is to combat these infections. And the current method is a standard culture test, which takes 2 to 7 days to come back. And in case of a fungal infection, it takes months for the report to come back. So which is exactly why there is an urgent need for a rapid device that can detect these infections on these wounds. Also, you would have heard of the rise in antimicrobial resistance, where for the past 30 years, we have not developed any new antibiotics. So the judicious use of antibiotics is the key here. And what is very pathetic is in India, still we come across very resistant strains, and it's very, very important to judiciously use these antibiotics. And we have seen increase in resistance with respect to MRSA, the carbapenem resistant Pseudomonas variety, and even in terms of Candida fungus, we are seeing a lot of resistant fungus being emerging. So how do we exactly go about this problem? We have developed a first ever multispectral imaging device. Now, what is a multispectral imaging means? We shine multiple light sources and leverage the natural fluorescence that is exhibited by the bacteria and fungus. So traditionally, these natural fluorescence are considered as a noise when we do a standard fluorescence-based assay under a confocal microscopy. And this noise is what we are actually using to aid in the label-free diagnostics. And on these multispectral images, we have applied artificial intelligence, which is exactly, it looks at a lot of these parameters that are released by these bacteria and fungus, and it categorizes as different types of bacteria. So that's in short about the device and the technology we work on. And this has very multiple applications, starting from wound care and dermatology to even in terms of cosmetology, where it can be used to detect skin hydration, skin melanin, and other label-free detection of these parameters. And in future, our goal is also to aid in rapid antibiotic susceptibility testing without the use of any reagents. So what is very important is, if we really have to focus on a remote location and add a last mile setting value, we really have to ensure that something whatever we developed can be used even by a compounder or a nurse in a primary healthcare center. It must be very easy to use. There should not be a lot of consumables attached with it. Otherwise, it's very difficult to reach a last mile setting. And this is exactly the goal that we had in mind when we started the whole process. So what we see here is the device that we have developed. We have a patent granted for the label-free diagnostics along with the multispectral imaging we have done. It has been tested on close to 1,000 patients. It has close to 90% accuracy with that of the culture test. And what it does is it leverages the golden period of wound healing, where we understand bacteria and fungal presence instantly rather than waiting for the culture test. So this is a prerequisite and a screening device before a doctor sends it for a culture. Yeah. So the device mainly helps the attending physician to look at the wound and analyze where bacteria or fungus is located instantly. It's a simple imaging device, just like how you use your smartphone to capture images. There's an increase in sampling accuracy. It guides the doctors into debride the wound, which means removing these dead tissues very effectively. And also they can give targeted treatment selection as of now. And it also helps in continuously monitoring the wound. What is very important again is it's very difficult for a patient and a doctor to send samples every day to understand the wound load, the microbiome load in that particular region. So that is exactly why this device adds value to continuously monitor and bridge this gap. So what we have essentially done in the beginning of this thing is we have first understood the major clinically relevant bacteria and fungus and the autofluorescence property that are emitted by these bacteria and the fungus. So here you can see that we have taken 4 common gram-positive bacteria like Staphylococcus aureus, Enterococcus, Streptococcus, and MRSA. When it comes to gram-negative bacteria, we have taken Kleb, E. coli, Proteus, Pseudomonas, Morganella, and Acinetobacter. And in case of fungus, we have chosen Malassezia, few dermatophytes, and Candida. So essentially what we have done is we have understood their excitation, emission, fluorescence spectra. So when we excite with different ranges of light, be it UV, be it blue wavelength, we have seen how the bacteria innately releases fluorescence due to the presence of many biomarkers that are inherently present in a bacteria. So what we have seen is the common metabolic biomarkers like NADH, flavins are strongly fluorescent, and when a pathogen undergoes a mechanism to cause tissue infection, they acquire the heme component in the tissues. So this causes porphyrin and pyoverdin release, and this is also strongly autofluorescent in nature. So we have done excitation emission matrix spectra studies on all these clinically relevant bacteria and fungus. What we have also identified is we have also induced a pathogen's mechanism, a disease mechanism to acquire these heme using an aminolevulinic acid, and also identified how these fluorescent parameters are changing over a period of time at different growth rates and different concentration. Currently, we have trained this particular algorithm towards gram-positive and gram-negative detection. In future, we are also planning to do species-level recognition of the same bacteria. So again, what we have seen and what is very interesting is these fluorescent intensity releases actually acts as a metabolic fingerprinting to identify these bacteria and fungus. So as you can see here, gram-positive bacteria has a very strong porphyrin release, the graph on the right side. So which means they have a higher release of fluorescence in the red region, and gram-negative bacteria has a higher release of flavins, which are in the green region and shown as a green autofluorescent region. So what the machine learning algorithm essentially does is it looks at these difference in this autofluorescent intensity and categorizes as the different kinds of bacteria. So the first step, we took clinical samples that are available from different laboratories after getting an ethics committee approval. And then we have conducted these studies on a large volume of these patient samples, which have been identified as a certain bacteria. And then we have kept this as a repository or database, and we have trained to understand what intensity variations we get, post which we did a study directly on a wound in a patient setting. Okay. So what exactly we do with respect to in a wound setting would be, as you're all aware, the wound is a very, very diverse medium for bacteria to grow. It's a very you have a lot of colonies of bacteria present, and identifying the one that is actually causing an infection, it is of utmost need. So this is exactly why we have put in a strong machine learning and an image processing framework, where the device what we have developed captures close to 16 multispectral images. So we have integrated 4 UV and blue wavelength of light source. We have a filter rotation mechanism which captures the autofluorescence in 4 other bands, which can be in the visible region. And then we look at these fluorescence that are captured in these different bands. And the image processing framework first identifies the regions where there are increase in fluorescence, followed by we take intensity value from this identified region, and then we send it to a training model. So we have our first version, we have adopted a random forest based classification, where exactly it looks at all these basic features and then it classifies into gram positive and gram negative. Again, we have close to 1,000 patient data where we have taken sample from the fluorescent region, sent it to a culture laboratory, got the gram type of bacteria and the species or the genus affecting, and then we have labeled those sites and sent it to the training model. At the end of the training model, we have got around 95% accuracy of classification. For fungus, we have additionally also done a feature-based classification, which looks at the skin region, how the textures are changing, and that has also been sent for training for increased accuracy of fungus detection. So the next step was to, we have done this training model, we have a device that has integrated. We did a clinical validation in 4 major centers in India, 2 in a diabetic foot ulcer center, 1 in a dermatology setting, and 1 in a general surgery setting, where they come across various type of post-surgery infections, be it C-section site, be it burns, it could be even bariatric surgery, and many of these places, many of these wound types. So we had applied for a CTRI, a clinical trial registry in India under ICMR, got it approved. We have taken institutional ethics committee approval in these 4 centers, post which we have done a clinical validation. So here, the major intervention is the device, the Illuminate device, which actually shines, collects the images and shows the region of fluorescence. The comparator agent is a tissue biopsy or a swab that is taken from this region and sent to a culture, and where we got the results back. So this is how we compare. And at the end of the result, we have, like I said, greater than 1,000 patient images that we have collected. And what is interesting is when it comes to gram-negative bacteria, Pseudomonas followed by Klebsiella and E. coli are the major causative agent. And in gram-positive species, the MRSA and Staphylococcus are the major causative agent in a wound infection. And the device and machine learning algorithm at the end of the clinical study has close to 90% of accuracy with that of the culture test. And the positive predictive value, the negative predictive value, the sensitivity and specificity are about 80%. And this is fair enough to make it a screening device at a point of care setting, where the doctor can use this device to screen whether it is infected or not, and then he can maybe send it for a culture laboratory to later understand what antibiotics to prescribe. And he can also use this device to monitor over a period of time. Now, here is a simple, here is a video of the device in action. Some of the images, there is a wound image over here. So there's a small disclaimer. If the picture or the image seems gory, please don't look at it. So I'm going to play it as of now.
Patient developed this very hypertrophic ulcer, which is not healing despite multiple courses of antibiotics. So we've taken the image and now the device is going to use its artificial intelligence algorithm. We'll get to know whether the wound is infected, and if it's infected, where it's infected. Before this device, we would actually have to take a biopsy. It might take as long as 7 days to help grow the slow-growing bacteria. This takes a minute. So antimicrobial resistance actually can be attributed to one of the causes, basically could be misuse or overuse of antibiotics, which is not needed. So this device can help in this regard. So this is the baseline image. This green image shows the amount of infection the patient has. And in the span of 40 minutes and over serial images, we can see with the help of debridement how the infection is going down. You don't need to give the patient any antibiotic. And in the span of about an hour or so, he's here, you've cleaned the tissue, he can just have it dressed and go home. The patient.
Yeah. So that is a thing that we have seen there. Right. So here are some case studies that we have seen in the real time scenario. The first one is accurate swabbing. So usually it's identified that bacteria is always present in the center of the wound region, but using this device, we have identified that it's present even in the periphery. So it gives a spatial understanding of where bacteria is present and helps the doctor take accurate first-line treatment protocol accordingly. Second is with respect to continuous monitoring. As you can see, without using the device on the right hand side, the doctor has debrided the wound, but then there is still presence of harmful infection caused by another gram-negative bacteria, which is away from the region of the wound. Again, the doctor can take, he can use this image to see and track over a period of time if that is causing a huge change in the wound healing pattern. And this is also where the device adds value in. Next, with respect to cleaning. So many times these wounds are being cleaned by a nurse, and he or she usually focuses on the region of the wound. But using the device, we have also seen that harmful level of infection are present in between toes, which also needs to be taken care of. So it actually empowers the nurses, the compounders to actually effectively clean the wound. And fourth, with respect to surgical site monitoring, as you can see, post a surgery, there's harmful level of E. coli that's present in the incision region. So which again, it helps the doctors in making decisions with respect to grafting, with respect to closing of suture wound, or prescribing an antibiotic accordingly. Not only this, the device can also detect biofilms, which is again a very big issue. Many of the chronic wound, at least 60% of the wounds are infected by biofilm, which forms a very thin layer, and then they don't let the wound heal. They are sometimes very inconspicuous for a naked eye. So which is why this device again is very helpful in identifying this very tough to grow biofilms in a standard culture laboratory. Otherwise, the only way to detect these biofilms is using a scanning electron microscope, which pretty much none of the hospitals have. So this is in short about what this particular device can do. In future, what we have also identified is the same device can be used towards dandruff application, also to serially monitor the acne bacteria, whether a particular pimple is caused by bacteria or is it hormonal in nature. Again, this is one field where there is a huge misuse of antibiotic, and we see a lot of potential for such device in such settings as well. And what we've also done is we have also progressively tracked how the wound load or the bacterial load is increasing over a period of time. As you can see, we have patched a band-aid supplied by 3M, and we have seen how bacteria is growing in the edges and how it is increasing over a period of time. Again, this can be serially done using this particular device. Now, because it is a medical device, we need to ensure and be compliant that it is safe to use. And we have a ISO 13485 2016 certified manufacturing laboratory in Chennai. And this is a made in India product and a first ever device in the whole world that can not only detect but also distinguish the bacterial gram type. And in future, as I had mentioned, we're also working on various deep learning models where not only you can detect the gram positive and gram negative bacteria, but we can also look at the species level classification of bacteria, and we have close to 89% accuracy. So we will be releasing this deep learning model very soon. We can also categorize major clinically relevant bacteria. In short, this is just to show that there is a huge opportunity that is available in terms of bacterial identification and how rapid you can identify. And this is exactly the space that we have focused on and worked towards from scratch from 2015 end till now. And we are very happy to say that we have currently commercialized and we are seeing increase in acceptance of such a product. We have testimonials from Apollo Chennai, from Jupiter Hospital in Thane. And what was the real impact? What we saw was when we went to government hospitals in Chennai, the one in Omamdurar Hospital, the one in Kilpok Medical College Burns Unit, what we can see is many of these patients cannot even afford a culture test, which costs 850 rupees. And it's a huge cost for them. So many times they don't send it for a culture, mainly because of this cost factor. And the doctors are really happy to use this device to quickly monitor and at least understand if it's a gram-positive bacteria, they have one type of antibiotic and serial tracking will heal the wound. If there is a presence of gram-negative bacteria, then they will send it for a culture and then identify what antibiotic to give. So this is specifically the value proposition we are looking at. And in future, we're also looking at how to detect the antibiotic susceptibility testing, because it's not only enough to screen the type of bacteria, but also close the loop and help the doctors in prescribing the right antibiotics within few hours. So this is where Edivo is working on this rapid AST device that can detect in under 4 hours using the same principle, where we expose the bacteria in various concentration of antibiotics and look at how these autofluorescence decreases or increases over a period of time. Here I have shown some early results where we have tested on Pseudomonas resistant and sensitive strain, and how within 6 hours we are able to see the difference between a susceptible strain and a resistant strain against a meropenem drug. So we are actively looking at developing this model on various other bacteria and antibiotic panel, and we are looking to come up with a commercialized version by March of next year. Again, it takes a long time in terms of getting all these things tested in an actual laboratory. Right. And last, what we would say is we are also working on fluorescence lifetime measurements, where currently if you want to procure a device that does single photon counting, it costs close to 1 crore. And what we have achieved is at 1/100th of the cost, we have developed the first ever fluorescence lifetime imaging device as well, that can look at all these smaller variations that can accelerate these antibiotic sensitivity and resistance pattern using a SPAD array and a nano LED. And this is also currently in development in our laboratory over here. So I think I have a question, just a second. Sorry if I missed that. Where was it tested, I think? Okay, never mind. Okay, fine. So here we are able to see the fluorescence decay in under 1 nanosecond. And that is the kind of thing that we are working towards. And this can bring down the antibiotic susceptibility testing in under 6 hours, which is our dream and what we wanted to achieve in order to close the loop. So currently in terms of traction, we have placed 5 devices and we have one international customer in terms of dandruff causing fungal detection. And we have partnered with a major distributor in India for marketing this particular product. We are also being assessed by major healthcare wound care players in US and in Europe. And we are planning to also introduce in the Burns ward in Chennai and also in Omandurar Hospital in Chennai. And we're also in talks with armed force and medical services to look at battle and military wounds where the device can add a lot of value. So our future is, like I said, we want to work towards species recognition and compare it against a metagenomic study, and also bring in additional wound depth analytics, which can also add in a lot of value in a clinical setting. Our mission is simple. As you can see over here, the person on the left, he's a farmer who was treated 8 years with antibiotics for a simple fungal infection, and and we want to serve patients like them who cannot afford a test, and they can add a huge value, and the device can add a huge value in helping them at a very early stage and to save their hard-earned money. We are recognized by various players like Qualcomm Design in India Challenge. We were winners of Google Launchpad, and we are happy to say we recently launched the device in last year in Global Bio India, under Dr. Renu Swarup as well. So thank you so much for this opportunity. I think I'll be happy to take any queries you may have. Yeah.
Thank you, Gitanjali, for that wonderful talk. And I must say that whatever the device you are coming up with is so timely, and it is going to have such a colossal impact on the overall health sector that this is just the beginning. So I think I'll quickly just start asking with the question which was there in the chat box, which asked where and how was the initial proof of concept tested?
Right. Okay. Sure. So we had, like I said, we had partnered with Dr. Ayush Gupta in the very beginning. He is a dermatologist in D.Y. Patel Hospital. So the pilot study we did there, and then when it came to clinical validation, we did in Dr. Vijay Vishwanathan Research Center in Chennai, High Care for Wound under Dr. Rajesh Kesavan, and one in Valsad, which is a government hospital in Gujarat, under Dr. Janek Parekh. So I came across all these doctors in various conferences, and they showed very keen interest in such a device coming up. So I had directly partnered with them, and they were kind enough to do the study without charging even a single penny to me. So I'm very happy and glad to have such associations. Is there a okay? Is there a minimum bacterial load that is needed in the wound in order to detect? Right. So currently what we see is 10 power 4 or 10 power 5 bacteria and above is considered as an infection. So what we have done is we have set the threshold in the device to detect above 10 power 4 by comparing the results against all these 1,000 patients that we have studied. Of course, with our devices that we are developing using the fluorescence lifetime, the photon counting imaging device, we can also detect cells of lesser than 10 power 2. So it depends on application and the minimum quantity they want to detect. We can fine tune the device accordingly. Whether the device can carry out species level, red color is representing gram positive. How MRSA is differentiated? Right. Okay. So currently, like I said, in a wound, in a normal culture, it's very easy to do the species level differentiation. But when it comes to a wound, there are also other things that are present, like your collagen that is forming on a wound if it's in a closure stage, the various tendons and bones, even your surrounding regions of the skin will cause a lot of noise and interference. So which is why currently we can do only a gram type classification. And in future, that is why we are working on a deep machine level algorithm. So which not only looks at only the bacterial signature, it also looks at all the noise that is present, and it does a more accurate species level classification. So here the key is more the number of data we have, the more accurately we can train. So it is currently in progress, but definitely species level identification is our next step, which we are currently working towards. Difference between MRSA and normal staph. Definitely, in a normal culture sample that we have got, that from a patient sample where, let's say, it can be a urine or it can be a blood sample, we have definitely seen difference between a resistant staph strain and a sensitive staph strain. Of course, in future, we can also do at least between an MRSA and staph, if not other resistant strains. So that is possible. Now, with respect to oral cavity, how the device works, right? So with respect to oral cavity, we have gotten some preliminary interest. And there are recently published articles where they have done a similar approach, multispectral imaging in an oral cavity. And we have seen positive results in those studies. Of course, if we have collaborators who can work in a dental thing, we would be happy to extend the same device for an oral cavity itself to detect infections in dental region, definitely. Often the problem with the learning algorithm is false learning. The algorithm starts to associate other coincidental non-infection related details. How are you able to overcome this false learning? Okay, that's an excellent question. This is something that we are still struggling with. So in our case, it is not overcoming, how do I say, it is not the false learning that is the problem. The problem is we are comparing it against a culture test. So many times culture test is not very sensitive. If you have sent the sample post 2 hours, it's better, you get a better result if you send it as soon as you collect the sample. So we have seen incidents where, because it is sent at, there is a sample transport error that happens. Sometimes the culture laboratory, the fast growing organisms gets picked up first. There are a lot of issues that happens. So what we have consciously does is we have chosen hospitals and clinical validation centers where the microbiology laboratory is nearby. It is NABL certified. And of course, all the false learning that we have done, we have not sent it for a training model. And the deep learning model that we are doing will definitely help towards overcoming these problems that you have just mentioned. And that's exactly what we are looking at, even when we are training using a deep learning model. So that's a very, very excellent question. Next, how come the pathogens which form heterogeneous biofilms can be found which can have diverse microbes? Okay, that's again an excellent question. With respect to biofilms, we only have a very preliminary understanding, and majority of the biofilms that we have discovered on a wound, that we have imaged on a wound is usually of a pseudomonas type. We have just started our talks with Dr. Karishma, who had given, I think, the morning talk on biofilms. So she has made it a wound model, a biofilm milieu model, I believe. And we are very excited to see how this device on her model can perform, so that we can then strongly give all the questions that you're asking on a biofilm. So to be very frank, biofilm is a very unknown field for us, even for the doctors and even for a culture laboratory microbiologist. So which is why I think Dr. Karishma and her expertise will come in very, very handy in this particular segment. Is there any other questions?
Actually, in addition to that also, I think Dr. Alex's next part of the question was in similar lines, where he asked if gram-positive and gram-negative organisms, if they're mixed in the biofilm, would your algorithms have difficulty in identifying that? So I think it was in the same lines of what you just now said.
Yeah, and just to add on, in a wound, let's say there's going to be a lot of commensals also present. But what the device exactly shows is the one that is causing the maximum infection. So our fluorescence gets picked up only on the pathogens that are present in a higher quantity in that particular wound. So that is something that we have found out, that even when there is a mixture, the one that is dominant is what will be picked up and shown in the gram type in our device.
And I think there is another question from Dr. Shrikant, which asks, how about fungal infections in the wound?
Right. So that's another great question. So all the 1,000 patients we have validated, we couldn't send it for a fungal culture. So our next step is we are partnering with a doctor in AIIMS Jodhpur who specializes in fungal infections. So that would be our next algorithm that we will release. So the device, the same device, we keep on sending app after app, just like how you get your iOS or your OS updates. Just like that, we will also send out releases when we train on fungus, when we train between MRSA and staph. And more the number of data, we will definitely train on those kind of population of bacteria, yeah, or fungus, sorry. Are there any other questions? Regarding the device that was showcased, is the detection only at surface level? How deep can the image, a few layers, say infection under the nail if possible? Okay, sure. So yeah, so the limitation of this device, I would say, is it can only penetrate up to 1.6 mm. It cannot go deep enough. But what we have identified again on a population of 200 patients, we have taken a surface swab where the device has shown presence of bacteria, and we have also taken a deep tissue biopsy. What is interesting is the one that is picked up by our device using a swab is the same that's present in the deep tissue. So this is one point. Second is the device is used for serial debridement. So the doctors as of now look at it, they debride the first layer, they see infection, debride the second layer. So that's how we're going about it. But of course, deeper level infections, like you said, we may not be able to pick up. With respect to infections under the nail, I think that can be picked up. If the fungus is expressing all these markers on the surface, it will definitely be picked up. I don't think we have faced any issues with respect to under the nail, but again, I can comment when we have more number of data on this particular segment. But again, thanks for that question.
Yeah, I'm sorry. I think I went into a technical glitch for some time.
No worries. Okay. Are there any other queries that I missed?
So Gitanjali, if I may ask you a question, it's just out of curiosity. This particular device which you have come up with, like, does the result of each of this interpretation based on the emission spectrum, is it dependent on the cell type where the wound is present? Like if it's a wound on, let's say, the joints, or it's a wound on a normal superficial surface, are the data going to change?
Right. So again, that's an excellent question. So with respect to, I would say, the background noise that is being contributed, definitely the algorithm has to be trained accordingly. So for now, the limitations are we cannot detect on a bone or in a tendon or a fat region. So these are the three limitations. But otherwise, the device's learning algorithm has been compensated for any other background noise that is getting contributed. And majority of these fluorescent markers that we are looking at is something that's naturally coming out of the bacteria. And in case of fungus, the chitin, which is one of the major cell wall structure marker, that is also fluorescent in nature. So that is separate. The question from which you are coming at, I think the noise is something that we definitely need to deal with. Yeah.
And Gitanjali, we cannot let you go until unless you share your experience with us in terms of, like, you have been there, you have done that, like starting from a science graduate, and what are the challenges in your path?
Sure. Yeah, I would say this was not an easy thing. It was really, really difficult. It was very gruesome. To start with, when I presented at the BIRAC BIG, which is my very first grant that I got, I was rejected mainly because of the market insights were very poor in this particular segment that we were dealing with. So then I went ahead and understood that particular gap which I need to fill, and then I presented, and I was lucky enough to get the grant, because nobody will trust you with 50 lakh of grant money, specifically when you want to do something that says I can detect directly on a wound. Nobody will believe you. So I'm really thankful to the BIRAC to actually start supporting us on such very new concepts which are very difficult to digest even with a committee who is very well known in microbiology and cell culture. The second aspect is when I started again, you have very low amount of, resources available in terms of conducting these experiments. So even though my company was based out of Chennai, I could only get facility in Venture Center in Pune. So I have to spend at least every month. I used to go there at least for 2 weeks, collect the sample from a hospital, go to Venture Center Pune, process it. Then the main instrument which does the fluorescence study is at MCL laboratory. So I need to walk because though the buildings are nearby, it is like really, it takes at least 15 minutes to walk. By then the sample, you need to ensure the bacteria is still alive, and otherwise your results are really crazy. So I think 9 months I had very bad results, in the sense I was even doubting myself whether this does this actually work. So the 9 months were very very difficult, and then fortunately I got access to Golden Jubilee Biopark in Chennai. So where we had access to all the samples nearby, so we were able to ensure that the bacteria were alive when we were doing the study. So that we overcame. Once we had this again, the 50 lakhs will go in a, it will just evaporate in thin air, because you do a lot of studies, you need to pay a lot of people. Again, securing funds was a very big thing, and I think developing a medical device, it's not very easy. You need to go through the regulatory process as well. So fortunately, I had approached Wilgrow, who's one of the biggest social impact investors. So they understood that such a device is of great value. You really need to work on this, and they had helped me with some initial funding. So that's how I overcame that. And I think the next step is, let's say you have developed a prototype, you do a pilot study. Trust me, it doesn't work. So the moment we put on the wound, we got no results. And I was again doubting myself, what did I get in the laboratory? What is happening on a clinical setting? So then, we had to fine tune, a lot of sleepless nights, and then we go and test it, and then it came out well. And I think the next major hurdle was you have to do a clinical study, and that too a multicentric trial with a reputed hospital. So that's very important. So I think I would have sent close to 100 mails to major hospitals, and fortunately, all the 4 doctors that I collaborated with instantly replied. So again, because it's a new concept, the doctors were very, very excited. So I think that was the kind of initiative I took. It's okay to send 100 mails. It's okay to fail. At least 4 responses will be positive. And I think the last step would be, I have 2 more points in case somebody wants to start their own company. So I think the next step would be, I have a product that is launched. Okay, now you go to the market. So we approached Dr. Arun Bal, who's considered the father of podiatry in Mumbai. And when we went and imaged it on a wound, the device didn't work. So we were really embarrassed. What is happening? Why is the device not working? The machine learning algorithm was not showing anything correctly, but the fluorescent images have come out well. So then we had to excuse ourselves, get some 1 week of timeline, and again take the device back to him, and still it didn't work. It was really embarrassing that 2 times in a row the device didn't work. And the doctor was kind enough to give us a 3rd appointment. Again, we found out that when we go to a commercialized device, there are a lot of supply chain constraints, or rather, when you're buying in bulk, you need to ensure quality in the product the components that you buy. And what we found out was the component that we bought, which was integrated for this particular doctor, was faulty. We changed our machine learning algorithm because the camera that we integrated, it was faulty. So again, that was a key learning for me. So you need to ensure the things that you buy in bulk, you need to do quality testing. Otherwise, you're going to fail and fail. And luckily, the 3rd time it worked, and the doctor gave us very good. He was kind enough to give us a 3rd opportunity, and also he liked the concept. So I think that is also one of the key learning that I went through. And last is, during the commercialization phase, many times when you go to a hospital, the doctors will say, I know it's bacteria. What are you trying to say? Okay, so there are many moments you get dejected. So marketing and sales is totally different ball game. You may convince a doctor, but you cannot convince a purchase committee. You can convince a purchase committee, you cannot convince a doctor. So you need to identify who's your target audience, which we are still doing. But again, I think that is also one of the key learning, because This is not a replacement to an already existing device. It's a new intervention device. So a lot of concept selling is required to, actually propagate such a device. And just now we have started getting all these positive feedback from doctors, specifically doctors who cater to below poverty line patients. And I think that is my insight that I got just now. So it's okay if the doctors who cater to the community where they can afford a culture test is not my market segment. My segment are the ones who are in a low resource setting. So that's also another key learning. I think the last but not the least would be your team is your major support. Many times when you have a team of 15, because I grew from one member to two members to now we are a team of 15, you need to ensure the dynamics between them is very important. So I'm still learning how to effectively manage people. But I think your team is what makes or breaks you. So I think that, in short, I hope it was not a whining session, but it was something useful. I think you can get out of. Yeah.
Absolutely. On the contrary, I'll say this was such an informative thing, not only about the device which you have come up with, but the challenges on the it's more that we should know about the challenges so that we can find out that one silver lining which is out there, and that is enough, to keep you going. So yeah, I think there are two more questions, Gitanjali, for you in the chat box. I think one is when to take the swab is the, yeah.
Sure. So the doctors usually ask the patient the moment they take a swab within yeah. So within 2 hours you have to give to a culture lab. So if that is the question you're asking. Otherwise, when to take a swab is it's based on the doctor's decision. So currently it's only a screening device and it's up to the doctor to interpret based on the fluorescence result that we had given. Next is how do latent microbes be found? Also, how can it detect a new microbe? How do latent microbes? Okay, if it is a latent microbe, then it would not be producing porphyrins or pyroverdin, which is basically a biomarker that is released only when it causes an infection. So because our device is looking at this biomarkers also, we cannot detect latent microbes, or rather the algorithm is fine-tuned to do A, B, and C biomarker present, then you show. So if somebody is interested in a later microbe, then we just need to ensure that A and B is there, you show the microbe. That's how the algorithm can be trained. If it's a new microbe, what we do is we first do the fluorescence study, like the one that I showed you in my earlier slides. Then we first identify what kind of fluorescence spectra patterns it exhibit. Then we feed it into the algorithm that's present in the device. Again, we need to train on wounds where this particular new evolution bacteria is present, and we need at least 20 to 30 images for us to understand and fine tune this new bacteria, especially if we want to do species level classification. I hope that answered the question.
Yeah, I think one participant has raised their hand. So I'm not sure. So if in case a question comes up,
sure.
I'll just read it out to you. Yeah. In the meantime, Gitanjali, let me just tell you that, your talk had been so inspiring to all of us here, and especially for all of us in Shastra, because you are our alumnus. So that adds glory to all this. So I think, what work you're doing is really worth appreciation, not because it's so unique, but also it is devised with an intent to serve the people who do not have that economic, backup. So more power to you, Gitanjali, and thank you so much for that wonderful talk. All the best.
Yes,
Thank
you so much for inviting me. I think I was pleasantly surprised to, again talk to all of you. I hope I could meet in person, but definitely the COVID has taken it in this way, so it's okay. Yeah. But thank you again. It was a pleasure, and I think the questions were very, very good. So thank you again. You've all been a great audience as well. Okay, then I'm signing out now. Have a great day, all of you.
Thank you, Geetanjali, and thank you, Dr. Deepita, for the lively session. So now I call upon Dr. Karthikeyan to invite our next guest. Over to you, Dr. Karthikeyan.
Good evening, everyone. It feels good to interact with all of you. and I'm immensely pleased to introduce the speaker for this evening, Dr. Alex Michael Walm, Assistant Professor of Biology from the University at Albany, State University of New York. Dr. Walm received his bachelor's degree in biology from Bradley University and his master's degree in cell biology from the University of North Carolina, Chapel Hill. Dr. Walm pursued his PhD in pathobiology at Brown University, following which he pursued his postdoctoral training as a Pratt Fellow at the National Institutes of Health. Pratt, which stands for Postdoctoral Research Associate Training, is a competitive and prestigious program targeted at training postdocs in basic biomedical sciences. Presently, Dr. Walm heads a prolific research program with a specific focus on the influence of microbial communities on health and disease. Using zebrafish and human subjects, his group employs molecular and DNA sequencing approaches to understand the role of microbiota in innate immunity and systemic health. His research group also specializes in employing advanced imaging technologies and computational tools to study the spatial distribution and structure of microbial communities. Among several awards, Dr. Walm has recently received the 3M Non-Tenured Faculty Award and the SUNY Albany RNA Institute Excellence in Research Award. Dr. Walm has published extensively in journals of repute such as Nature, PNAS, PLOS, BMC Biology, Journal of Molecular Biology, to name a few. Needless to say, his research is well funded by grants from agencies such as the National Institutes of Health. Today, Dr. Walm will share with us his work on investigating the structure of the human oral microbiome across multiple spatial scales. Without any further ado, let's welcome Dr. Walm with a virtual round of applause. Over to you, Dr. Walm.
Thank you for that wonderful introduction. I'm blushing a little bit here. So let's see, let me just share my screen. Is that right? Do you see the PowerPoint in the right format?
Yes.
Okay,
yes,
great. So, thank you, Dr. Kathikan, for that introduction, and thank you to the organizers for inviting me here. This is a phenomenal opportunity for me to participate in a symposium that's occurring halfway across the world. What a time! And also, I have to say, so obviously I was sleeping for some of this because of the time difference, but I was able to watch some of the earlier talks, and I've never been in a symposium where I think both sides of my lab are well represented, both the biological side, where we focus on the oral microbiome and periodontal disease, and the technology side, especially with that last talk, where we're employing multispectral imaging. So I feel very much at home in this symposium, and I don't think I've ever felt that before. So I'll just launch in then to my talk. So, right, so I always like to start my talk with this image, and I call this the Human Microbiome Project, circa 1690. So many of you may be familiar with this image. I think I saw this first as an undergrad. This was the first direct observation of bacteria. And this was made by Antonie van Leeuwenhoek in the Netherlands in the 17th century. And and what's remarkable about this image is that 300 years later, I can tell what organisms these are. And the reason is I know where he got the sample from. So I didn't know this until grad school, but actually Van Leeuwenhoek had scraped his dental plaque, and that's where he saw bacteria for the first time. And so when I look at this, obviously I see this spirochete here. This is probably a treponema because they're highly abundant in the mouth. These coccoid cells could be a number of organisms, but Streptococcus is just so overwhelmingly abundant in the community. That's probably what it is. This organism here that's doing this curly dance here, I think must be Selenomonas because it's one of the abundant flagellated organisms and so on. And this organism down here, actually, I want to draw your attention to these organisms labeled F here. This one that's boomerang shaped, I know exactly, that's Leptotrichia, anyone who's seen it. under the microscope knows that these are actually two cells that have divided but have not yet separated. And so a couple of things that we learned from this image, we learned that the very first direct observation of bacteria were from those from the human body. So before we knew that there were bacteria in the soil or in the ocean, or we knew that there were bacteria within the human body. As far as we know, Van Leeuwenhoek was healthy. We don't know that he had gum disease. So our first observation showed us that bacteria in the human body are associated with states of health and not with disease. And lastly, what I think is really important, and what you have to sort of think about, you have to imagine yourself as Van Leeuwenhoek back there 300 years ago. He made this drawing, but of course, he must have seen all these organisms living together. So our very first observation of bacteria was of them living in a community, okay, a multi-species community. And somehow that first observation was ignored, right? For 300 years, we've been studying bacteria in monoculture in the lab until very recently, where we've come back to this idea of the importance of the community environment. So one of our earlier speakers very well set up this slide here. So my lab does focus on studying the role of the oral microbiome in states of health and disease, especially in the mouth, and we focus on dental plaque. So just by way of introduction, we think of dental plaque at least two different communities. So there's a community that lives above the gum line, which we call supragingival plaque, and then there's a community below the gum line, which we call subgingival plaque. And as was pointed out, what we understand is that there is a specific community that lives in the mouth in states of health, which we call eubiosis. And this community undergoes a shift. Certain organisms become enriched, and these organisms are associated with periodontal disease or gum disease. And we call this dysbiosis. Right. And we also know a lot of other things. Of course, we know that the gross anatomical and pathological sequelae of the disease. We know that certain organisms, certain bacteria engage with the immune system. We know a lot about the specific cellular and molecular events that are involved in promoting an inflammatory response that is not resolved, and that ends up providing an environment that enriches these organisms that cause or are associated with the disease process. And what's pointed out here on this graphic, we think that there are keystone pathogens. So these are organisms that are able to, like I said, engage with the immune system, promote inflammation, and then accessory pathogens, which are other organisms that somehow are involved in this process. These accessory pathogens are what my lab is most interested in. Now, this is the state of the art, okay, of what we know, a summary of that. And still, there's something missing here, and that's how these microbes are drawn here. So even on the tooth, the bacteria are drawn. They're arranged as if they're randomly distributed in this community. And as a matter of fact, we have strong evidence that they're not, the bacteria are not randomly distributed. They have a specific structural pattern. And so that's what my lab is interested in. And we consider this a factor of scale. So we know that at the micro scale, okay, at the scale of individual bacterial cells, we can identify the molecules that are involved in interactions between bacteria. At the macro scale, we can identify the community that's involved, right? That shift from eubiosis to dysbiosis is characterized at this scale. And what my lab is interested in, though, is this what we call the mesoscale. So this is actually the scale of the dental plaque biofilm itself. And this scale is important because we know that microbes take on what we call emergent properties. at this level of organization. For example, as has been pointed out earlier, antibiotic resistance, as well as resilience in the environment and nutritional cooperation, among many other things. And I would argue that this level of scale is not as well studied as the other two levels, and that's because of a lack of tools. So what I'm going to focus my talk on today is our work developing tools to be able to study this, this kind of structure. And one last introductory slide here. So again, I guess just a cartoon here on the left. Here is the importance of the biofilm interacting with immune cells. And I like this drawing a little bit better because at least it acknowledges the importance of spatial structure. So here you see that these are neutrophils. What we know is that neutrophils are highly abundant in the gingival crevice. They extravasate through the tissue. We haven't seen them though in the biofilm. We haven't seen them phagocytosing cells in the biofilm. So this is just a hypothesis, but we have seen in dog models, and I heard dogs were mentioned earlier, we've seen these immune cells in juxtaposition to the biofilm. And so really what my lab wants to understand is, if we know that there are keystone pathogens, we know that there are accessory organisms, how are these organisms, how is the spatial structure of the biofilm bringing, for example, the keystone organism into direct contact with the host epithelium and its underlying immune cells. And just to show you that spatial structure is important as a general phenomenon in the microbiome, it's not just restricted to the mouth. So here is a biopsy sample of a mouse intestine, and what you see are here are the beautiful cells of the host epithelium. These candy dots here are the bacteria in the gut. And what you notice is that there's a separation between the bacteria and the epithelial cells, this dark area here. We know that that's a mucus layer, and we know that that's important for preventing bacteria from making direct contact with the epithelium. When they do, they elicit inflammation, and that is almost by definition a disease process. In the skin, it's just the opposite. So here is a hair follicle, and there's a biofilm that's growing on the hair follicle within the dermis. What I want to point out here is that a couple of things. So this biofilm is, again, a mixture of bacteria and fungi, which we heard about in the skin wound environment. And here in the skin, what we know is that the microbes actually do make intimate contact with the underlying immune cells, and that contact is important for establishing the correct inflammatory balance in the skin. Okay, so what I've told you so far is that at the macro scale, okay, we can use DNA sequencing to tell us what are the organisms that are present in a community. At the micro scale, there have been many genetic molecular studies that have identified specific molecules that are involved in interactions. And that together, these two types of observations led to this first hypothesis for the spatial structure of the oral of dental plaque, in which you have the tooth surface here. It's coated by salivary proteins, and then a subset of organisms binds. These are called early colonizers. After these organisms bind, other organisms bind. They're middle colonizers, and then late colonizers can come in and bind to those organisms. So there is a process of ecological succession that occurs over time. Our lab is particularly interested in Fusobacterium nucleatum. That's this orange organism here, which if you pay close attention, you'll see is different from most of the other cells in this diagram because it can bind to many different species, not just one or two. And at least from a sort of high level aerial view, what we see is that these early colonizing organisms are those that are associated with periodontal health. The late colonizing organisms are those that are associated with periodontal disease. So at least this first hypothesis tells us that the spatial structure of the community is important for understanding whether this community is going to promote health or cause disease. Now, it pains me to put this bad hypothesis label on here, because this is decades of work that went into this beautiful model. But I do have to remind myself, I tell my students that this was a bad hypothesis when it was proposed, and I ask them why. And the reason is because it was untestable. It couldn't be tested. You couldn't see all of these organisms together in dental plaque with taxonomic or species level resolution. So that brought us to our first tool development, which is developing a tool to be able to identify many different organisms within a biofilm. And so our lab uses fluorescence in situ hybridization to label bacteria. That's because of the wealth of molecular information that's out there. We know the DNA sequences, especially of that 16S marker gene for probably the majority of the organisms in the oral microbiome. So we can design probes. As well, there are many different fluorescent labels. There's probably at least 50, maybe 100 different fluorescent, spectrally variant fluorescent dyes that are compatible with microscopy. So we could label up to 100 different organisms, but that is yet to be achieved. So why is that? So here I'm going to tell you about the two tools that we're developing in my lab. One is that technological tool, the other is a biological. So to get around this problem, okay, I'm telling you that we can design a probe for any organism. We have fluorescent labels for at least 50, maybe 100. So why do we routinely only image 3 or 4 different organisms in a sample? And that's because these fluorescent dyes, as you see in this graph here, have wide spectra, so they overlap, and we can't distinguish them using filters. So as a first approach, our lab uses commercially available microscopes, mostly confocal microscopes, but not exclusively, that allow us to measure the full emitted spectrum of light at every pixel in the digital image. Then we use algorithms to find the best fit of our known fluorescent dyes to that observed spectrum. And with that approach, that greatly expanded the number of dyes that we could identify. I won't go into the details of it, but that got us to, at this point, about 16. Okay, but we want more than 16. So we also used a combinatorial labeling approach in our first iteration of this, where we labeled each organism with a specific, almost like a barcode of different fluorescent dyes. And so we called our approach Classy Fish for combinatorial labeling and spectral imaging combined with fish. As a proof of principle, we first made this artificial community. So these are E. coli. They're all the same strain of E. coli. We just labeled them with the same fish probe, but a different fluorescent label or combination of labels in separate tubes, and then combine them to make this sort of artificial community. And we imaged that. We also developed our own software to be able to go in and, in a high throughput way, identify the cells and identify their combinatorial label. And we showed that that our approach worked, that what we put into the mixture was what we got out from our computational analysis. We then moved on to a natural community, which was dental plaque. And we first identified the 15 or so most abundant organisms at the genus level in the community. And it turns out that these 15 organisms cover over 80% of the community. So we set out to design probes, either identify probes in the literature or design new ones for these different genera. again, we created an artificial community as proof of principle, but this time what we did was we cultured representative species of each of these 15 genera in the lab and then mixed them and then added our probes. And so here we're showing that our probes are specific for their targets and that with our computational analysis, we can identify all of the organisms in the correct proportion. So that we established our proof of principle. Now we went to look at a natural community. We had volunteers come into the lab. We are not at a dental school, so we have our volunteers self-donate. So they take floss, they floss their teeth. And we recognize this to be a mixture of both that subgingival and supragingival community. They floss their teeth, they hand it over to us. We scrape the dental plaque off and apply our probes. And this is what we saw. We were a little surprised. What we saw were these aggregates. We saw these clumps of cells that were made up of different taxa. And so we wondered if these clumps represented actual spatial arrangements that were present in the plaque biofilm before we disrupted it with dental plaque and all of our fish chemistry and all of that. So we did some computer modeling and we did identify all of the statistically significant intertaxon pairings that were present among these 15 genera. And this allowed us to perform this network analysis where each node here represents a genus. The size of the node is the absolute abundance of that organism in the community, and a line connecting two organisms means that these organisms were shown to directly bind to each other, or co-aggregate as it's called in the field, with statistical significance. And so this was the first network analysis of a microbiome that was informed by spatial data. Right, so okay, so we got 15 genera. It turns out that like I said, about 80% of the community is covered by those 15 genera. But if you go down to species level, what you'll find is that there are 700 different species that have been identified in the human oral microbiome, and each individual human host carries between about 50 to 100 different species. So we'd like to be able to identify, like I said, 100 different targets in a sample. We're actively working on this. And so let's see, right, so we want to identify more than 100. So it turns out if we did use this combinatorial approach, that with 16 different labels, we'd get 120 different barcodes. So we developed our own algorithms for identifying, for doing this spectral deconvolution. In the field, it's called linear unmixing. Our algorithm used both excitation and emission information, and it used that a priori information. We realized we could capitalize on the knowledge that we know every bacteria in the sample is labeled with exactly 2 fluorophores. So we restricted our algorithm to it could only find a solution that involved 2 fluorophores. And when we added that constraint to the algorithm, we found that if the signal was very high, if the image was bright, it did not outperform the already existing algorithms without these constraints. But when the signal to noise level was low, when the labels were very dim, which is very common in a natural community, our algorithms was superior to the unconstrained one. And so again, we did a proof of principle experiment, and we showed that we could identify 120 differently labeled E. coli in a sample. And so we're still working on this, actively working. We are now bringing in machine learning. We've got some exciting preliminary results, which I can't share yet because even I don't understand them fully yet myself, but I hope to be able to share those soon. We're also working at applying these technologies, right? So up until now, what I showed you was disrupted biofilms, okay, when people floss their teeth. What we'd really like to do is be able to image biofilms intact. And so we have a couple of strategies. So we have collaborations with clinicians. One of our collaborations, we have a protocol where healthy volunteers wear this appliance in their mouth. On the side of the this dental appliance, we stick what we call enamel chips. These are square cut pieces of third molars extracted from other volunteers. So we've got a tooth surface, an experimental tooth surface on this appliance. And so our volunteers wear these for a day or so, and then the chips come out and they get sent in the mail from NIH to us in Albany, and we can label them. And what we see is we confirm that we see these early colonizers when the volunteers wear these chips for 4 to 8 hours. The magenta color here is Streptococcus. That's one of the early colonizers. We also see cells of Haemophilus, which are probably, Pasteuracea, which are probably Haemophilus. We see Gamella and Rothia, and interestingly, we see this characteristic, I call it a volcano cake. I don't know if you're familiar with that, but it's a chocolate cake, and in the middle is a liquid chocolate. And so here we find these Rothia cells in the middle that seem to nucleate and get surrounded by streptococci. And we saw this repeatedly in our samples, and now other labs are reporting this as well on teeth with different organisms, but also involving streptococcus. So this seems like a structural phenomenon that might be important to streptococcus biology. We also have collaborations where we're looking at teeth. So this is very early data that I'm showing you here, but we collect teeth, okay, from patients who have severe periodontal disease, who have to have their teeth extracted. And those teeth are then sent to us in Albany. And again, here we have this tremendous challenge. We've got a very hard surface, the tooth, and then we've got this extremely soft surface, the biofilm. And we want to be able to fix this sample and create sections, thin sections, so that we can image it on the microscope. That's proven to be a major challenge, but we're working through it. And at least we've got some preliminary results where we can actually section the hard tooth and maintain the biofilm intact. In this case, it helps to have calcified biofilm as well. And so that's what you see here, the calcified biofilm here, which still has within it streptococcus. And what I want to point out is that fish imaging is a proxy of metabolic activity, because our target is, as I mentioned, the 16S ribosomal RNA. We are labeling ribosomes. If the cells are brightly labeled, it means that they have lots of ribosomes. That probably means they're metabolically active. So this is something we're pursuing. I'm not sure if this was well understood, that at least a subset of organisms in the calcified biofilm are metabolically active. And then lastly, we're looking at what we call plaque smears. Excuse me. So these are, in this case, what we do is we have the volunteer floss their teeth for us, but instead of scraping off the floss, we gently lay the floss down on the slide. And if you do this and you scan around, okay, sometimes for an hour or more, you scan around, you see this disturbed biofilm, but all of a sudden you'll find a region that looks like it was undisturbed. And so our colleagues actually first called these hedgehogs. And so what we think is that the tooth surface was down here. This is the apical surface of the biofilm. And sure enough, In this region that's attached to the tooth, we find the early colonizers, Streptococcus and Actinomyces especially. But what's interesting is that we also see Streptococcus among many other facultative anaerobes in this apical region. And so what this has led to is an updated hypothesis, because what's in the middle here in yellow and this light cyan color are Fusobacterium and Porphyromonas. And I'm taking out the Fusobacterium because it's so abundant to show you the Porphyromonas in the middle here. Those are two strict anaerobes. And so it's clear that the community that forms, it's not a linear progression of organisms. There's a dynamic remodeling that takes place as facultative anaerobes create an anaerobic environment that allows anaerobes to infiltrate. And so this has led to an updated hypothesis in which these long filamentous cells, especially of the genus Corynebacterium, are assigned importance in structuring the biofilm. And so again, another area that we're interested in pursuing. Okay, pardon me, I need just a sip of coffee here, it's early for me. So that takes me to part 2 of the talk, which is the development of the biological tool. And so what we are interested in is, so everything I've shown you so far in terms of biology has been observational. We go to a healthy volunteer for the most part, or even a volunteer with periodontal disease. We label the biofilm and we observe what's there. What we would really like to do is be able to test hypotheses, okay? If you think a specific surface molecule is important in binding between two organisms, what is the role of that event in the community? We would like to be able to test those types of hypotheses, and for that we're developing an in vitro model. We did not invent this model, but I'll describe it for you. So it's called a microcosm. In fact, this was invented hundreds of years ago by environmental microbiologists. But basically what we do is we recruit volunteers, again, for us, self-declared healthy volunteers. They donate their dental plaque with floss or saliva, and we take that dental plaque as a mixed species inoculum and grow it. We culture it in the lab, and our early results showed us that if we provide a highly nutritious media, we get nothing but Streptococcus to grow. If we provide a media that's almost water, okay, 25% human saliva is over 90% water, and then some glycoproteins. And so if we restrict the nutritional milieu, here we get a diverse community, which to us suggests that these organisms cooperate metabolically when they're in an environment that's not carbon rich. And so here's our protocol. Like I said, we recruit volunteers. We sequence the initial inoculum. We grow biofilms in microfluidic devices. We label them and image them, and we do parallel sequencing of what we grew in the community. And so our initial results showed us that, so on the left here is a 16S amplicon sequencing of either one donor's saliva or their plaque. And we grew biofilms either using that dilute saliva or an artificial gingival crevicular fluid saliva hybrid medium that we've been developing in the lab. And what we see in both cases, we get highly diverse communities. We were thrilled with these results. Of course, there are some differences, that's to be expected, but I'll draw your attention to this dark red colored band. This is Fusobacterium. So we are in fact growing Fusobacterium in these species-rich communities. And so we're gonna be able to test different hypotheses regarding Fusobacterium. So I skipped a slide here just to get to the real interesting part here. So here's what we see in these in vitro communities. So in this case, we're labeling for six, sorry, I just have to move the camera here. So we've got 1, 2, 3, 4, 5, yeah, 6 different organisms at the genus level. And we see all 6 of them in the biofilm. We have multiple cases. We've done multiple replicates. At this point, we have collected and banked, cryopreserved dental plaque from one donor, which we got right before the pandemic was declared. And we had to stop our collection at that point, although we're ready to begin collecting again. But we see these highly diverse communities, and all the time, Fusobacterium seems to be present where the rest of the community is also abundant. And so I don't have a good example. Maybe you can see here. We see great heterogeneity in these biofilms where there are regions where there aren't many cells. In this case, they're mostly Streptococcus, the early colonizers. But when Fusobacterium is present, we see that all of the other organisms assemble with the Fusobacterium. And this is consistent with that earlier model that I showed. So our data is supporting that model, and we think that we are beginning to grow these hedgehog structures in vitro, which will allow us to test different hypotheses, right? Our goal is to go in and try and perturb, especially Fusobacterium nucleatum, its interactions with other organisms in the community. Here's a good example where you see the Streptococci that sort of live on their own. They're fine to live on their own, but if you've got Fusobacterium, then you seem to recruit many other organisms into a highly diverse biofilm structure. And so other labs have published similar results looking at both in vitro cultures and actual biofilms on teeth extracted from patients, and they see these heterogeneous clumps of organisms. And so we think that our model is recapitulating what's seen in the natural community. So in conclusion, I'll tell you a couple of things. So first of all, our oral microcosm cultures, okay, our model system, recapitulates the natural community extremely well in terms of taxonomic composition. In terms of spatial structure, I give it a plus minus because we do see structures that sort of look like the hedgehog, but it's not an exact replica. We have yet to see that apical cap of facultative anaerobes. We also, like I was telling you, we see regions of low complexity where we call the, this is just my own language here, I'm calling them facultative social taxa. So these are, other people would call these pioneer organisms, for example. These are organisms that are able to live on their own or to colonize the surface early. And then we see these obligate social taxa that are only present if other organisms are there. I draw an arrow here, but we don't know which direction this arrow goes, okay? We don't know yet whether what organism is involved in recruiting the others and what are the dynamic events that are involved in this development. But what we have done is we have recapitulated with a highly diverse community observations that were seen previously with cultured isolates in a more reduced complexity models. And so here's our model. We think that, so actually I have an engineering student in my lab, and I really appreciate this comment that he made one time, and I've heard this before. So there's this saying that that all models are wrong, but some models are useful. And right, that's true, right? So all models are wrong. If the model were right, it wouldn't be a model anymore. It would be the actual system. So we've got a model, an in vitro model, and it's not exactly right. We do see diverse communities, but we don't see this patterned structure that we expected. And that's, of course, because we're missing some of the inputs, okay? Some of the parameters of our model are just not there. And those are especially anything that comes from the host. So any involvement of the immune system is completely absent in our in vitro culture system. However, we do see these dynamic, what we think are dynamic processes of biofilm development. And so we think that our model is well suited to test, especially events that occur early in biofilm development and that are mediated by intercellular interactions, both biochemical and biophysical. Right, so we think we do have a powerful tool here. So what's next for us? As I told you, we're very interested in Fusobacterium nucleatum. It's interesting, other people, well, there's been a lot of interest in Fusobacterium nucleatum. I'll just leave it at that. But why we're interested in it is because we know some of the surface molecules that are involved in mediating its specific interaction with other organisms. And so now we have an in vitro model. If we could go in and perturb these specific surface molecules, we have a way to test their effect on the community, a community level phenotype. So that's what we're doing now. We're using both forward and reverse genetic approaches, as well as molecular perturbations. And so in the end, I have to thank the people that actually did the work. It wasn't me for the most part. It was other people in the lab who do the sequencing, the labeling, and the imaging, and also create just a wonderful environment to be in, contribute ideas. And so I'll just draw attention to Alex Lemus. He's the one who has been imaging these biofilms and developed this microfluidic system for that. So I'll call your attention. You'll be seeing his work soon, I hope. And also thank you to the generous funding that we've received. And hopefully I've left time for questions. If so, I'm happy to answer any questions. So thank you.
Thank you, Dr. Val, for that wonderful talk. In fact, I'm so happy that we got to share this one, because your research is a very good example of how exploratory research could lead to hypothesis-driven research. And that's something that like students and students could actually learn from this particular work. And also the perspective of scale in your work is quite interesting, which is something very unique about your work. So thank you for that wonderful talk and insights. And I'll go over the Q&A tab to read some questions for you. Great. So we have a question from Srini Kesan. What happens to these microbiome when we brush or use oral mouth fresheners? Will they die? What does it say about their resilience? Right.
Great question. That is an area that we are interested in as well. So, right. So obviously, oral hygiene is involved in the development of these biofilms. And so I think the prevailing, the conventional wisdom is that, on a daily basis, you brush your teeth, you floss your teeth, you might use mouthwash, okay? And so what happens is you remove, you complete, hopefully to near completion, you remove the biofilm from surfaces that are accessible. But there are always surfaces that are inaccessible to those tools, and that's why you have to get a professional cleaning periodically. And so to speak to the resilience of the community, we know that the community is stable over time. There are some fascinating studies that have been done through DNA sequencing that shows that the oral microbiome is stable for at least 2 years at the level of the strain. So that tells us that, yeah, there are niches where these organisms are able to sort of hide out whenever you're doing oral hygiene, and the biofilms come back. And that's something we're interested in, is how then do you, if if you've removed the biofilm from, the majority of the surface of the tooth, how does it come back? If you stop oral hygiene, we know that the biofilm will come back. So how does that process work? That's something that we hope our in vitro model can help shed light on.
Okay, thank you. We have another question. So in case of aggressive periodontitis, how do these biofilms affect individuals?
So aggressive periodontitis. So first of all, I'm not a clinician. I am a PhD microbiologist. Aggressive periodontitis is as I understand it, is a particular disease. We know it's mediated by specific organisms, especially an organism called Aggregatibacter actinomycetum comitans. It can affect people at a younger age. In general, the way I'll answer this question is that what we know about especially aggressive periodontitis is that this is there's a feed forward loop. And I think it was in that early slide, I didn't mention this, but what happens is when periodontal disease initiates, when that dysbiosis initiates, there seems to be a window where it can be reversed. And this would be the case of mild gum disease called gingivitis. We understand that this is a reversible process, but if the dysbiosis just pushes too far, I don't know how to explain it well, what happens is there becomes this feed forward loop where the organism, the bacteria initiate inflammation, the inflammation promotes growth of these organisms, and it becomes a vicious cycle that there's no stopping. We don't have an effective cure for periodontal disease. What we want to do is try to prevent that initiation, or at least be able to reverse it at the early stage.
Okay. I have a question. So in the first part of the talk, you talked about the mesoscale and how community structure and shifts in composition in biofilms are seen based on your work. So do you think all of these are like mostly density dependent mechanisms, or do you think there are also density independent mechanisms that are in place?
Yeah, so that's a great question. So, so I have to say we don't know, but that's exactly the kind of questions that we're trying to ask and get the answers to. I think one thing I'll point out in terms of this issue of density dependence is that these in vitro models are grown aerobically, and yet we see the anaerobes present. So somehow these organisms, we know that if they're exposed to oxygen, they die. In the lab, if we expose them to oxygen, porphyromonas, to oxygen, it will die, and we can't revive it. So there is some sort of mechanism where these organisms are protected even in the process of nucleating a new community. So that's one thing that we've seen. Of course, people know that there are density dependent signaling mechanisms, although we're learning that maybe some of the mechanisms that we thought were important might not be as important as newer mechanisms that are mediated by those gram-positive organisms. That's a very active and exciting area of research. We know that the streptococci can produce peptide signals that are both synergistic and antagonistic. So there are density dependent mechanisms for sure.
Okay. And there is another follow-up question. Is it good to brush twice a day based on what you talked about oral habits and biofilms earlier? So is it good to brush twice a day, or is it bad?
Yeah, so I'll take this opportunity. Okay, so some early culturing and molecular-based work showed that and I'm not sure if this was a supragingival or a subgingival community or a mixture, okay? But what these early results showed was that this process of going from the early colonizers to the late colonizers that are involved in periodontal disease takes about 12 hours. So that suggests that brushing your teeth twice a day is the right frequency. I think that's where that notion comes from. Now, someone mentioned earlier about mouthwash, and there are some emerging evidence that the organisms that live on the tongue might actually be important for promoting health in terms of blood pressure. And so these organisms, the tongue is enriched in nitrate reducers, dietary nitrate that you eat gets reduced to nitrite. The body then can convert that to nitric oxide, and that lowers blood pressure. So there's this, again, it's a little hand wavy, but there is a hypothesis at least that the organisms on the tongue are providing a health benefit, and therefore maybe we shouldn't be using mouthwash, or we shouldn't be scraping our tongue. We should let those organisms grow.
Okay, that's quite a bit of an insight. Thank you. And there's one more question. What is the direct application, clinical application of the model that you have developed? More specifically, in case of dysbiosis, how is the developed in vitro model useful?
Sure. So actually, let's see. We have collaborations with commercial companies, actually. So of course, I can't go into the details of that, but think of this: you can have an in vitro model like this, and if you've got there are you can test any chemical that you think might promote a healthy biofilm and prevent a dysbiotic biofilm from growing. So with these in vitro systems, what we hope is that as a first approach, we could test mouthwash, right? What does it do to the community? What does it do to dental plaque, for example? Or we can test specific molecules that we think should be added to toothpaste or any kind of dentifrice. So that's a clear example. I think what we're really interested in from the basic biology standpoint is, what are the mechanisms that maintain these communities that maintain that high level of diversity? And so if we can understand those complex, maybe multifactorial processes, maybe we can discover new avenues, right? A new molecular pathway that we can target that could, like I say, promote the health promoting organisms and prevent the disease causing ones from expanding in the community. That's the ultimate goal. I do think we're going to get there.
Okay. But I have a follow-up question to your answer then. So your work was about the spatial structure and organization, but how do you think you can factor in the temporal component in?
Yeah, so that's a great question. So we can't use fish for that because we have to kill the cells in order to label them. Okay, so there are more, like I said, there are reduced complexities. There are some organisms, a subset, that are genetically tractable, and so there are the fluorescent protein expressing strains of some organisms. To be honest, that's not what my lab is interested in, because we're interested in the whole community, right? So we've got a problem here. Most of the organisms, these wild organisms, are genetically intractable. We know we can't. I hope that you're still seeing me. My internet is going out. We know that these organisms cannot be manipulated genetically. So what can we do? In comes machine learning. That's all I'll say. We're working on ways to use machine learning where we can use other features, I think is what they're called in the field, of these cells that Again, what we get is we don't get taxonomic resolution, but we do get information on the dynamic process because we can image these. We can image these biofilms as they're developing on the microscope stage. So we get some of the information, but not all of it. So we need all the different approaches to sort of bring it all together.
That's amazing. Okay, we have another question. What is the role of commensals in the oral environment other than the ones that prevent colonization by pathogens? If extremely antimicrobial oral cleaning regimens are used daily such that we maintain an almost sterile oral environment, are we losing on some metabolic ability that only commensal bacteria can perform?
Yeah, so that's really a great question, right? And you know, I would open this up to other experts, but I think I mentioned the nitrate reduction, right? And I think that's pretty much the only commensal, that's the only health promoting aspect of the oral microbiome that I know of. So I, you know, in my opinion, okay, I hate to give an opinion, but maybe I'll call this a hypothesis, okay? My hypothesis is that are these organisms commensal? They're commensal in that the mouth is a surface that is open to the environment and can therefore be colonized by other organisms. So we maintain a certain subset of bacteria, mostly bacteria, although now we're learning that there may be commensal fungi that are part of the oral microbiome. But we maintain these microbes and they provide a barrier. I think that's the hypothesis, is that these organisms the commensals are anti-inflammatory. They provide a barrier to infection and colonization by other organisms. And I think the evidence for that comes from, you know, if you take antibiotics, you know, repeatedly for a long period of time, you have a increased risk of fungal infection in the mouth. And so I think that supports that hypothesis. So I would say, I think so far what we know is that the main role of the oral microbiome in a commensal capacity is to prevent colonization by potentially pathogenic organisms.
Okay, there's one more question. So there could potentially be multiple host factors that can influence these oral biofilms, which can be variable between patients, between human individuals. So how do you think specific characteristics of these biofilm structures from your study can be indicators of oral health or some sort of a oral disease? So because you're bringing in machine learning as a function of the structure of the microbiome, given this amount of variability, how do you think these characteristics of these biofilms can inform us about oral health?
Yeah, you know, I think that all I can say is yes, please, you know, you should serve on my grant review, whoever asked that question. I think that, you know, our goal is to try to answer that question, right? No one has looked with taxonomic resolution to the degree that we have, right? No one has seen 15 different genera intact in dental plaque on a single person, okay? Never mind multiple people. And so at that macro scale, we know that genetic background, we know that behaviors, right? Like our earlier speaker mentioned, smoking, for example, can contribute to the community, the microbial community. And so how does that translate to spatial structure? No one knows yet. We would love to find out. We'd love to have a large scale. We are working on this actually with our collaborators. We're working on large scale imaging of plaque biofilms. We're restricted right now to patients with severe periodontitis because healthy people don't like to donate their teeth. I don't know why. You know, of course, no one wants to lose their teeth. We can get third molars if they're erupted, but even those are usually involved with gingivitis because it's difficult to get the toothbrush back there when they erupt. So yeah, the answer is we don't know yet, but to some extent, our process, like I mentioned, is high throughput. Okay, it's computational. The analysis is definitely high throughput. The labeling and the imaging is a little bit low throughput right now, but maybe if we find that if we do find that golden example where the spatial structure of the community is involved in the disease process, then I think we could ramp this up, right, to a more high throughput, commercialized type of enterprise.
Right. Thank you for that. In fact, just to add to that, I would be curious to ask you if, let's say if we did a PCA or a principal components analysis or an NMDS, a nonlinear multidimension scaling analysis on the list of host factors, how they impact these structures, would they reveal like a couple of or 2 or 3 key factors that possibly influence the community structure by and large, setting aside all the variability?
Right. I honestly, I want to ask the other clinicians here about this. I know of these large scale studies for other diseases, right? But what about periodontal disease? Is there a large scale study where multiple host factors throughout time are considered? I'm afraid I just don't know. Yeah, I guess it's kind of a cop out here, but we know. I have a slide. I didn't have it here where I have 3, almost a triangle, right? Where you've got the composition of the community, you've got the genetic background of the host, and then you've got the behaviors of the host, and all 3 of those things combine and promote either health or disease, right? So we do know that. I guess what I would answer this in my area of expertise would be from the spatial, in the spatial realm. And yes, we are doing this. We are actually using animal models. Again, it's too preliminary to show yet. I should say I've only been in this position for 3 years. So it's too preliminary to show yet, but we're comparing our human biofilms to our animal models. And we're using these data transformational approaches, right? Like principal components, cluster analyses, and machine learning to try and see, are there structural features that define disease and health, irrespective of what the organisms are? Are there structural features that are markers of health or disease? And so that's something that we're interested in. Again, the idea is to find out what are these basic molecular and cellular pathways that are involved in either promoting health or allowing disease.
Okay. There's a comment from Dr. Prasanna Neelakantan on the question that you just answered. Not to my knowledge, dental clinician scientists prefer to leave out the host component always because it's complicated.
Yeah.
Okay, that's the end of the Q&A. So if there are no further questions, let's put our hands together virtually for Dr. Alex for this wonderful talk. Thank you, Dr. Alex. It was quite a stimulating talk, and thank you for keeping us engaged. Thank you so much.
Thank you. It was a pleasure.
Yeah. Over to you, Sahana.
Okay. Thank you. Thank you, Dr. Alex, for this insightful talk. Rather, I would say colorful, spectacular talk. Thank you, Dr. Karthikeyan, for chairing the session. Now we call upon Dr. Prasanna Neelakantan, our organizer, to give us the concluding remarks and close the session. Over to you, sir.
Thank you, Sahana. A very good evening to all of you in this part of the world, and a good morning to those in the Western Hemisphere. Thanks for attending the proceedings of today. and making this first Indo-Hong Kong Symposium a grand success. We've had more than about 300 registrants and participants in this lecture. At the outset, let me place on record immense thanks to all members of the organizing team led by Professor Adeline Princey and all the session chairs for their efficient proceedings. Today, we have coursed through some exemplary lectures over a wide range of topics. This is probably the first ever symposium that brought together lectures by basic scientists, clinician scientists, and entrepreneur scientists. The extraordinary content that you witnessed today is discrete and yet strengthens the philosophy that the nexus and synergy between the STEM disciplines is critical for successful understanding of disease pathogenesis, point of care diagnosis, as well as for implementation of healthcare sciences. Such collaboration is inevitable and is required for success in the grand scheme of things with context to public health. Together, let us learn and advance science for the greater good. Thank you all once again. Wish you all a great day.
Thank you, Dr. Prasanna. So with this positive note, we shall end this session. We once again thank all the participants and the speakers and the session chairs to successfully run this symposium. Thanks a lot. Thank you so much.
Thank you, Prasanna.
Thank you, ma'am.
Yeah.